hydrocorals, you said?
Okay, we're live.
All right. Hello, everybody. Welcome back. This is Colleen Hansel. I'm a senior scientist at Woods Hole Oceanographic Institution and chief scientist of this cruise. We had a little bit of a delay today. We had to come up from our last dive at about noon. We were having some technical difficulties with our sensor, SOLARIS. So for the last, let's see, about 5.5 hours, we've been troubleshooting, taking it apart, putting it back together again, running some trial runs, taking it back apart again, putting it back together again. But we think we solved the problem, and so we're excited to be back down on the seafloor and start looking around for some corals and sponges to explore. So thanks for joining again. We are back at the same spot that we left on the last dive, if you were with us for that dive. So back at Desecheo Ridge. We're at about 320 meters or so. And you can see we got a lot of big rocks and a lot of sponges. Lots and lots of sponges. So we're just getting ourselves oriented here.
Do you want me to just get right back over to waypoint 17, or
Yes.
have a look around this sponge?
No, 17 would be great. So the goal for this dive, we're hoping to find a few species of corals that we either haven't measured yet or that we only measured 1 or 2 times. And since the last dive, SOLARIS was misbehaving a little bit, we'd like to go back and remeasure some of those corals. So we're going to be looking for, I think, 3 different species mainly. But if we see anything interesting, of course, we're going to stop. For those of you who have been with us, you know that we don't get very far. We get very excited about just about everything.
It looks like there's a coral up here. Might be up on top of that. I might be that's the one that you were looking for. The Paramaricia. And that top one on the other. Lost contact. Oh yeah, I never turned it on. That would be better.
Are you guys seeing some corals?
Yeah, right there in front.
Oh yeah, there we go.
Looks like a light purple Paramaricia.
If you get a closer look at it.
Yeah, I'll get you in and just.
See, we didn't get very far, did we? We were moving towards our waypoint, but then we see this beautiful coral. So we're going to get a little closer look here. This might be one of the ones that we were interested in.
Yeah, that's the light purple one. Well, we should. Oh, I'm not sure.
Is that a Paramaricia?
Yeah, it's what they're calling a light purple.
A light purple?
Paramaricia.
I believe this is where they fired the squeezer.
At this same location? We fired a squeezer last dive.
What's that?
Or to wait to move forward.
Oh, no, you're good. I'm trying to keep you safe over there while I look at this thing. Just let me know when they're ready for me to come out with it, rock them, and protect it. But if I'm getting too close to the rock for you, I'm going to get too close.
Oh, that's crazy.
That's what I do.
There are a lot of sponges.
Roger that.
10 or 12.
Rockham's coming out.
Is that a coral there with this long white one here?
Yeah,
Yeah.
that's probably an Ellicella.
Ellicellid.
All right, go ahead and just make sure it's like just inside, not sticking out further than Rockham. So do you want to have us?
You can see there's quite a bit of current here again.
Oh, I got the good view, so I think it's.
You need to get a better view of it?
No, I got a good view. It's
Okay.
the light purple, very, very soon. But I don't know if we want this one or try to find
Should we
a
just
higher
keep?
density.
Yeah, okay. Yeah. Why don't we keep going?
Okay, so I'm at waypoint 17, working
Okay.
my way to 18, correct?
Perfect. Yep. We're going to fight the urge to stop and measure and keep going for a little bit, explore a little bit.
Bottom is just all sponge.
This is just,
It's all like a single species.
that's a lot of them.
If this was if we were in a forest, I would
Maybe
call
have a look over there.
that tree rot fungi.
Yeah,
Right? With the,
you see a little where the elbow is, I think.
they're like wavy and
Yeah, it does almost look like those kind of fungi that stick out of trees.
Go off to the left a little bit. You'll get in a good spot. Okay. Just kind of like cut it down here a little bit. There you go, that's good. And then we'll just have to have a look at it right before we come out with the tray of it. That is. I brought a screen.
Ah, thank you.
I'm good for right now, Chris.
All right.
Yeah, 5'8", right at like 3.
Just tell me when to come back.
Do you mind making me a plate? Is there anything good?
Indian night.
Oh, we really a plate?
I can do that. Would you like some?
As you can probably hear, it's dinner time here, so we're doing shifts, taking turns, or having a plate made for us so we get to eat a little later. We didn't want to delay getting in the water any longer after
Start moving the vessel
our long
to the west.
Tell me when to come back.
delay earlier.
8?
So away from our. Yeah, just go pretty much just straight west. Yeah. Okay. Light for you. Bridge ROV. Yes. This is good waypoint. This place is crazy.
Oh, I see a different sponge there, Scott. That one there. Little round sponge there.
oh yeah.
So not a mono species mound here, but oh, there's a couple of them there. I don't see any corals except for those little whip ones. What are those little whip ones, Sam?
Elicella.
Oh, right, Elicella.
Elicella is based
Is that an octocoral?
Some of those,
Yeah,
some of those are also black corals. I mean, not that,
Yeah.
but some, you said some of the spirally ones.
on.
Right?
The curlicue, pigtail ones are black corals.
Yeah. Man.
This place is.
So for those of you who may be interested what we were fighting with earlier with our sensor, so the way SOLARIS works is it pulls water in through the wand, and that water then reacts with a chemical probe that is very specific to the chemical we're interested in, called superoxide. Oh, I see an urchin there. And that reaction is called a chemiluminescent reaction. So it gives off chemical light, and we measure that light in real time with a photomultiplier tube that's within the wand. And that data is then streamed back to us here in the control room, and so we can see it, what's happening. What? Oh, what,
No, it's just
some sort of little shell there?
Try to
What is that?
take a button.
Gastropods.
A little gastropod there?
Yeah.
Bright yellow in a sea of white sponges.
White sponges.
Yeah, so we think that what happened was got a clog in one of the lines during one of our earlier dives. We had a bump with the ground, and we think we got some sediment that managed to lodge its way up into the manifold where all the lines are pumping the different fluids within the sensor. and it just managed to lodge itself in such a way that it was blocking one of our check valves. And so we managed to take it all apart and clean everything out, and it seems to be running well again. So we have our fingers crossed that we solve the problem and we'll have some good measurements this time. Oh, those little yellow corals. Is that the other one on our list?
No, the new one should be a little bigger than that. taller.
Okay.
Purple.
Do you know what that yellow coral is, Sam?
We'll take a closer look.
You need a closer look?
Do you want to take a look at it?
Let's take a look at one, yeah.
Oh, is that a weird sponge there?
Yeah, I think it might be the same kind, just growing.
Is that our dirty marshmallow? It's close to a dirty marshmallow.
Looks like it, yeah.
Or toasted. That might be the toasted marshmallow.
I'm not sure if these are scleractinians or stelasters.
Are those all the same species here? The ones that are
I think it's at least two.
two different species? There's
Maybe three. There's a yellow one, a white one, and there's like kind of like a tan color.
like this little pink one down here.
It's a little more branch.
Yeah, the pinkish one, yeah. And they don't just differ in the color, they look like this, they have slightly different branching pattern too.
Yeah, exactly. Yeah, the branching looks different. So Sam, how much do you rely on color in identification of corals?
Not much.
Not
Well,
much.
To identify if I'm in a new area, but once I realize what species is in an area, then I might use color to find it. Like that purple Mauritia. I wouldn't have known it was Mauritia because it was purple. That I only realized that when we got it up
There.
close.
You wanna go over
And
that?
then once we know it's there, I know that's the purple one.
So
So if you couldn't hear that,
color
Sam was saying that
in a certain particular environment.
you don't rely on color, but sometimes if you're getting familiar with the different corals in a region, then you can kind of use that to help you
Yeah,
kind of
some
hone
sites
in
where
on the same species.
have purple, red, yellow, orange.
Oh, okay, okay, so that happens.
Yeah.
What are we looking at here, Sam?
Oh, wow.
Is that something of interest?
It looks like a red version of the yellow
That-
Mauritia. The yellow Mauritia.
It's coming right out of a sponge, right?
Trying to get you over there, give me a second. There it is.
There are really big polyps, right?
Yeah.
That's a happy coral.
Let's make an observation of that. You compare Mauritia.
Oh, that's beautiful.
Sorry.
It's not clear that that's our,
Let me see if I can find, let me find a way over there. I don't know, rocks.
Wow, look at that.
Terrain.
That is crazy terrain. Is that all carbonates?
Yeah, probably.
I realized we probably should have stopped for that light purple Mauritia because in our notes, it's not there to say we just need one more, so.
Oh, that's all
Is
right.
that the same one?
Yeah, same colony,
Yeah.
the same species.
Because it was at the waypoint where we like, we descended.
Okay, then it would be safe to left.
We'll say that was, we assume that it was the same.
We just started our dive. We'll see another one. This one's super cool, though. We haven't seen one of those yet, Sam, have we?
No, yeah, so we're talking to the bridge.
But
Give me a second, just trying to.
I guess we keep looking for.
Do you wanna have,
area
do you wanna have a
where
good zoom in on this one, or not?
Yeah, I think that would be great.
Okay.
Do you think we're going to want to measure this one? Seems like
Let me know.
it might be worth.
That's a different kind of setup.
Okay.
Yeah, you wanna.
Is that a place where it would be difficult to sit down?
Maybe not.
Because we could keep looking. We can always come back.
Make it work, but, I can get
Yeah,
you
let's
in
just
there.
keep
I
looking.
can get you in there.
Challenge accepted. for the Solaris from here, I think what we need to do is wait until we're seated
Yeah.
and then wait, you know, make sure we're ready to sample and then do a background seawater and then move
Yeah,
the tip.
definitely.
Just because we ran out of reagents.
I know. Snake hole.
Here. Oh, there's another one.
Seen a couple of sponges.
Yeah.
There's a white coral on the bottom right there.
It's really hard to tell
That one.
some of these apart that have really stony skeletons. There's some scleractinians that kind of have that growth form that look like that. There's an octocoral that looks like that.
From the sponges? Or from the...
Like that little white guy on the right. That
Yeah,
little
yeah.
fan. Really stony. He's gonna have a really brittle, really stony skeleton.
Would we expect scleractinians this deep?
Well, I saw one at the very beginning, a small one.
Okay.
I think you can.
Yeah, we can see him this deep.
So we can see them this deep.
Down to over 1,000 meters.
Really?
Like almost. 1,400 meters at least I've seen him. I saw like 4 species at a site at 1,400 meters.
How is the current here?
Not too bad.
So give us a chance to zoom in on it too. Look at those polyps. That is so pretty.
Some type of soft coral.
Since,
The Photogrammetry.
are you gonna log this, Sam, since Nicole's not here right now?
Yeah, I don't
Yeah, I'm logging on that.
Okay.
Let me get a better focus.
I like the soft
Observation.
corals. I like the grammar sand.
Does this help with ID, Sam? Or,
Yeah.
What's that little thing inside the sponge?
oh yeah. There's like a little critter in the sponge right there.
What is it?
Looks like a sponge in a sponge.
Yeah.
A sponge in a sponge.
Am I good to start taking a background sample?
What do you think? Are we in a good spot? We can start measuring? Okay, we're gonna do a background sample first.
Right now, your nozzle's right here and the coral's right there.
Okay.
Is that far enough away?
Yep. That looks perfect. So we're gonna,
Am I right? The current is coming kind of at us.
Yeah. But I want to see it.
A little bit from right to left, yeah.
so maybe we'll wanna go on the left side of the coral.
For people in the comments. I love that. My family like sent me a link that Wendy's Schmidt was on the Today Show. I was talking about like
So we're getting the Solaris instrument fired up right now, and we're gonna collect a signal for the background seawater. Because superoxide and other reactive oxygen species like hydrogen peroxide are produced in the water as well. So from microorganisms and chemical reactions that occur within the water. So what we typically do is get the signal from the background, and then we move the wand close to the coral surface.
okay, we're good to measure.
All right, we're good to move it on in. And then we move the wand to the surface so that we can then see, is it higher or lower or the same at the surface?
Did you just do a calibration? And how did it look?
You just bring that in when you do the sample.
Do you guys have like a file number?
File number? Yeah.
We've been doing the coral number as the sample name, and then the event number is just the event number from Solaris. So whatever the SH number is for logging.
Well, we want to sign an SH number unless we collect the sample, so I guess we'll collect the sample.
Oh, you don't think we would collect one?
No, we can, we should.
Okay.
So
Looks
it
like
would
you're
be,
right over it, for the most part.
that's H8 rolling
Closer to us.
628.
Say SH628.
That's H18,
Do y'all want to touch them? Like, was that, were y'all?
Let's try to get just as close as we can,
Okay.
down current of it, so it looks like it's kind of,
Yeah, not seeing anything yet.
okay. So maybe closer in, like where more of the branches are, like down in this area, because right now we want to get to where there's more polyps.
Okay.
file of this data collection or the event of
I don't know if you change like one to be, is it like a short, like,
No, the
is it a
file name is
suffix
long.
that, but is there like a suffix that keeps track of?
T13922.
How is it looking, Erica?
Just the date.
Yeah.
No signal so far.
Okay.
Same as the background.
Is it event one?
Can we poke it already?
Huh?
Not yet,
Camera
he
keeps
just
just
bumped
like
it barely.
doing like a zoom on it.
They got all polyps are tracking.
Yep, the polyps went in.
Or what is it?
Grab, someone's here.
Anything?
Same as the background.
Yeah, but what you.
Okay. I think we can pull back.
Okay.
It's this one.
Good, good
I
stuff.
believe so.
Pretty good, yeah, I like that.
But it's
And you can turn it on. Turn it on.
it's okay.
Joe, do you know how to turn that monitor on? Is there a trick
All right, so
Stop sampling.
underneath it?
we just sampled this this coral, which we think is Paramaricia. Paramarisid, and same family, and we did not get a superoxide signal off of this one. I don't know, we're taking a look at this this other coral over here.
It's hard to see through the double plane.
Yeah. You can see the water is kind of shimmery because we're in the thermocline.
Is there anything else you want to look at while we're here? Or pick up-
Did you want to take a piece, Sam? Yeah.
We gave it an essay snore, so let's take a piece.
Okay.
Okay.
It's not a very big colony, so we think that whole thing might fit in the program.
It's shaking.
Yeah.
Yeah.
You
can
want
go
to
ahead.
Where do you want to put it? You want to put in one of the covers?
Go ahead, and then I'll go. Okay.
Okay. They're already out.
So we're going to take a piece of this coral that'll help us identify it.
Yeah, and most of the time, why would you do it? And then you told me, you said it's not that hard. And then I did a power side. Are we in trouble? Live clouding. Debate of information.
I support.
I'd like to fix that all the way, but it's.
Do you want to take another? I'll take a bathroom break.
Sam, can you tell me what number this one is?
Sample, but like just get that signal. So, you can look at it again. I can look at the background.
Is it okay if we put it in number 17?
Yes, that's fine.
It's already out.
No, I don't know what the base of this is. But this is like it's dark, yeah.
All right, we're going to take a piece and put it in one of these coral quivers. Good night, William.
Turn, you turn yourself a little bit more sideways so you can see your backside.
Sam, can you tell me the number, please?
18628.
628? Okay.
And you're going to want to try to drag it.
All right. Oh, look at all that fresh water coming out of the coral quiver. We fill these up with water when we're on deck and loading up the basket. And so you can see all that fresh water piling out.
Go up and over that.
So we're going to collect a piece of this and then we put it in the coral quiver and we'll be able to then sequence the DNA, the microbiome that's associated with this coral, as well as look at kind of some of the more ecological factors, you know, the density of polyps, how far apart they are, how big they are. We're trying to understand how some of these factors are playing into whether or not we see the production of reactive oxygen species outside their cells under certain conditions. So some of it is environmental, some of it is, we think, ecological, some of it we think is the microbiome. So a lot of factors at play. So we're just at the beginning of trying to kind of figure out what are the different things that are regulating that. So this is going to be the sample number SH18628 for our records. We're at a depth of about 310 meters. The time is 22:10. Or 6:10 local time. The coral looks like it's quivering with excitement. Bring me home! It is a very positive interpretation. It looks like the polyps might be back out, yeah? No, they're still retracted? All right.
Do you want him to try to get some of the sponge as well, or just the coral? Because
Just the coral.
Yeah.
It's just a coat.
let's get it a little bit higher than the sponge, because the sponge is wrapped around its base.
Oh, okay.
You still need so I can zoom you in with it if you want.
I didn't realize how small that was when we started because we were so zoomed in.
Plus, I can give you sorry, buddy, I got you a little too far away from that. See your view and sit.
What's that?
See your view and sit.
Yep. Good?
Yeah. Start closing and then maybe all to the left a little bit. Don't close all the way right there. You're actually too far now. Like you're just grabbing it with actual T4 jaws. You become pull back a little bit if possible. Yeah, right about there. It's thick. Looks okay. Maybe drift it a little bit farther.
Sorry, our first coral turned out to be a challenging one here. It's a little itty bitty one sitting inside a sponge. We don't want to get the sponge because it'll kind of contaminate the sample that we'll be getting inside the coral quiver.
You shoulder left a little bit right there. Yeah, keep coming. A little bit more left or go left. Yeah, a little bit more. A little bit more. Keep coming left. There you go. Now go down. Go to the left. Yep, start cutting there.
Oh,
You're in it. You're in
looks like we almost got it.
There you go. You got it. No spoons, just coral.
Look at that. Nicely done. You make it look so easy.
Which quiver did you open?
17.
Ah. Oh.
Those already out.
Is that all right?
Oh yeah, we gotta update our sea log again.
Oh, okay. This is going in Coral Quiver 17.
Like push that out a little bit further if you want.
Look how red that looks.
I like it. So one thing I've done in the past is like to actually shoulder right more to give yourself more of the arc to lay it down. If you do you'll block your view if you do this side. You can go straight down in there with this one maybe. Is this a sticky one? Is it a heavy one?
This one should be a little bit easier. It's going to be somewhat heavy. It'll sink on its, should sink on its own,
Okay, so
but
yeah,
it's kind of light, so it might hang on something.
so get it in, like make sure that branch that's about to catch is inside. And then try to coax it in. There you go. So I'll just, yeah. Just try to go under your, the tips of your drill first.
Almost there.
And then just like pump open. And then just give it a little bit of a wiggle, like rotate it. Come up a little bit, okay. Wiggle, keep coming up as you wiggle, okay. To the right.
There it goes. All right. Woohoo! Nicely done. That was a very, very bright color.
Picking up after this and moving around, or is there something
Yes.
else you wanted to see?
I think we're going to move around unless anybody has an interest. No? I should move around a bit. How did the calibration look for Solaris?
It's a little bit up and down, but as expected for the first one.
Okay.
Alright, you good to
think
start with
that
the other
would
camera?
pace them out like at least 2 to 3 hours, given that we
Good job,
won't
Beck.
be able to follow it, so.
Yeah, right. Okay.
But no reason to
Clean
believe
water,
that
next,
the
clean water.
measurements aren't working. So
Okay.
back out.
Okay. All right. Solaris seems to be behaving. Background signal looks good. We got a calibration. So either the coral that we just measured isn't making superoxide for whatever reason. Again, we don't really know why these corals. Well, we know the variety of different reasons why corals make extracellular or extra. So outside of their colonies, these reactive oxygen species, including superoxide and hydrogen peroxide, we know there's a whole variety of reasons why organisms do it. But we haven't had many measurements at all of corals, either in shallow environments or in the deep sea. So could be for a variety of reasons. And some corals, it seems like they're always making it. Some seem to only make it every once in a while, or we don't catch it at all. So there's a lot to learn. We know that it's an important process, a chemical process that
That's a lot of.
organisms do to kind of change their chemical environment around themselves to either acquire nutrients or fight off pathogens. What is that one?
Oh, that's corallium.
Is that a corallium?
That's our target.
So that's a precious coral. And there's like a spider or crab, or I can't. You can clearly tell I am not a biologist. This up there on the left-hand side. Oh, a crab. Yeah. He's definitely running away. Bright lights. Oh, that looks like a nice big pod that maybe we should.
That's one of our targets.
Again, we didn't get very far.
Is that something that's going to be easy, or I mean, that's going to be possible to measure with Solaris and sample?
Let me have a look around. Let me get some level 360 shots of this if you don't mind, and while I'm doing that, I will
Maybe 3D
be
model of this rock.
That is a really, really
I don't know about
pretty.
3D modeling it, because it's in a little
That's insane.
Oh, I think it was
Is this the fan?
really hoping for
What is it?
This is
mosaic.
corral.
So he wants this. Yeah.
And it's pretty different from all the other corals we've seen so far. So it's really interesting to see the next
More
are left.
fan, yeah.
But it's like stony and great.
It's interesting. It has such a thick stalk, right? So really looks very hardy.
Dr. Hansel, you should go eat.
All right, I'm gonna let Scott Wonkel take the reins here. Another geochemist, so you'll hear him saying the same thing. Sam, what's that coral? All right, I'll be back.
Very nice.
All right, sounds great.
Hello, everybody.
You notice there's not that many sponges around this corallium on that rock.
On that back, on the back side of it, there was, but
Yeah, but not on this front side.
yeah. What? Thanks.
I think if I can sit right there, knock 'em on the back side, you'll be able
We're at 310 meters here off this Desecheo Ridge,
to set. Just let's knock 'em on this side.
using a variety of tools to understand the biology and chemistry and ecology of some of these deep sea coral and sponge environments. This is a corallium
Yeah, can I ask you a question? real quick?
colony. Sure.
So you want to be on the down current side, right?
Ideally,
Which would be on the other side of the rock.
but
To the-
it could also be that we position the SOLARIS wand behind it.
Well, I'm just trying to find a parking spot. And
Yeah.
also a good spot for him to manipulate around, which we're going to find. Just trying to get my head around what all we're going to need to do here. So if I do that same thing that we were talking on the other side, maybe that'll be their best bet.
Where that little rock outcrop is.
So it's dinner time here on Falcor 2, curry night, 6:20 p.m.
Yeah, I had a lot more behind me.
We're along the Desecheo Ridge, which we've been exploring here for the last several days.
How do you spell that, Coral?
C-O-R.
The one on
A-L-A-R.
what?
No. You spell. I need to write it
Coral
down
species?
first.
Yeah.
Corallium. So coral, L-I-U-M.
No,
2 L's?
Yes, 2 L's. That's skeleton.
So before I took over for Colleen, she mentioned that there's another geochemist taking over to try to talk about biology. I'm not going to try to talk about biology, except for how fascinating it is. I study, elemental cycles like nitrogen and carbon and oxygen in various types of environments and with various types of tools.
Does Sebastian have like DP?
No,
Jason does?
You have auto depth, right?
you do have that. I don't use it. I don't use it.
Is it different? Or the same?
What do you mean? I
Is the
think,
auto depth separate from the DP?
not really. Like on which way? What do you?
I've got a question in the chat.
Mm-hmm.
Are the photogrammetry and superoxide readings for the same research topic?
Yeah. Yeah, we were going to make 3D models of the corals to understand if any of the differences in
There's a crab.
RS production, RS levels, had to do with the morphology of the coral.
Yeah, so the idea in a The idea being that so what Sam was saying was one of the goals is to try to understand how morphology, that is the shape and structure of the corals, whether that plays any sort of role in where we see the superoxide being produced. Part of that has to do with the physical flow regime around the corals and things.
Are y'all good with this?
This looks great. This looks like a fantastic setup.
All right, do you want to have me zoomed in? Or are y'all doing background? And am I far enough away for background?
The wand tip, yeah.
Should be, right?
Yeah.
You want to bring it up higher or something? And it actually looks like the current's coming right at us here. So
Yeah, kind of to left from
yeah,
left
so
to
I
right.
think I'll be able to get a good reading.
Okay.
All right, I'll get you some nice little beauty shot. It's gonna be nice right there.
Some little crabs. There's little hermit crabs crawling around up there on the. Yeah, so big part of what my group at Hui has been working on is developing chemical sensors. There's a lot of folks at Hui that do that, or a fair number of us. And SOLARIS, which is an instrument that's here on the vehicle, was the development of that was led by Colleen Hansel. I was also involved, as were a couple of engineers, William Pardis and Jason Caput, looking to develop an in situ analyzer of reactive oxygen species, the compounds that really are very short-lived, so you can't just collect a water sample and bring it back up and measure it on the ship. So that's what we're doing with the SOLARIS right now. SOLARIS, Colleen was the queen of coming up with acronyms, is the submersible, oceanic SOLARIS, luminescent analyzer of reactive intermediate species.
Yeah.
SOLARIS. There's a little shrimp.
I'm done collecting the background.
Okay, background is done. Now we're going to position the wand closer in towards some of the pink. Maybe this colony here, this big colony on the left. Maybe in here somewhere.
Yep. You good with that?
And there's enough variety in here of flow regime and growth morphology and things that we can probably try a couple different sites. We'll hang out for a bit.
Are those like bags, those scooper bags, still on the ROV?
Be careful with that. These are also fragments. You want to be on what side?
This side,
This side.
if possible.
Yeah, I think you're just gonna have to get up and around.
I mean, you could also start up here if you want, if that's more convenient.
Better view. How's that? Oh, it's very red. It might need to be back up. I think the white balance might be off.
All right, it's probably my fault.
Are you about to measure the one on the left? Just sticking out? There were two maybe?
Yeah, he's just trying to get in position for it right now.
Is that one you think we can also sample?
The one on the left?
The one that we're about to measure?
Are you coming into this one?
I think that's the one he was going for.
Okay.
It's really
I mean,
tight
we'll
quarters,
figure
yeah.
out a way to do it.
Okay, it's just that we got a stupid complicated system and we have to assign it a name, and if we sample it, we use a certain set of names, and if we don't, then we don't assign a label to it for complicated reasons.
Gotcha.
So So if it's one that we can't, try to sample down, give it a certain name.
If you were to
So
go,
I think we'll do it
y'all
with the
down,
SH number.
or y'all try to go left.
So it's gonna be six,
That's
18,629.
Just y'all left.
Okay.
No, no, just y'all left. Let's bring your pitch back up.
SH number is gonna be
Not asking
18629.
y'all, just y'all left.
629.
There you go.
Correct.
Keep doing that, now switch shoulder right. Get up high up here, slow down, a little bit.
I'm
more,
logging
pass
no
that,
SOLARIS
to the left.
start now.
Okay.
That's my thought. Good shit. Sorry about that. Y'all left.
CYB is Corallium,
These would obviously have to go in the bio box, right?
that ID 2.
Yeah, we're gonna
Go way out.
Yeah, so I'm thinking it's like a scoop on the front.
Yeah.
The second you touch these things, they just fall apart. It's like a really delicate coral, so that's why that bag, scoopy thing,
Okay.
that if you start bumping it from below, branches will fall into it, and they dump that in the bio box. That's how they do it.
Oh, okay. Okay.
You can come up and over as well. You bring your elbow up, way up, and just point it straight down to the level. Yeah, you're about right. Yeah, you're just about to hit. And I mean, if you come up and over, then you might be able to get over to these better, and then it's easier to sample as well. For these things. Really. I need to wipe down. Look at these two cameras.
I was irising down a little bit.
Yeah, no, I mean that. There's no other. You're on the back side of the footwork.
So right now we're just just trying to position the Solaris wand closer to the color, the surface of this corallium. They're very fragile, apparently, and so easily broken. So it's a little bit more challenging than some of the other corals that we've been sampling before. Sorry.
And I'm watching you right now. Your elbow can start going down now.
Yes.
Just your elbow. There you go. Right. And bring your elbow. Once your shoulder right a little bit more, your elbow down a little bit more. Just don't clear the cameras. There you go. Alright. Now you can start rotating and whatnot to get yourself in there.
You can see the pump, the sampling pump there in the Solaris wand at the very top.
These corals are really brittle, they're like stony. So if you touch them, they're probably gonna start snapping. So you're gonna have to keep.
It's amazing. Then how do they stay intact here with all this current pushing them around?
Well, it's ROV strong.
Oh yeah.
They have those thick bases. Hold still.
Your jaws are getting a little close to the top. Okay. Yeah, so I think you're gonna have to keep doing what you're doing. Yeah. And just maybe rotate counterclockwise. There you go.
So it's a delicate dance here with the ROV manipulator positioning the wand.
All right, how close do you need them to be in? Now, I think you can use a little bit of your yaw left, and that'll swing you into it. No, yaw, just yaw. There you go. Hold that, you get close, switch to the top of the thumb. There you go. Before that, yeah, just like that. So now you got a view of your tip right there. And everything else is clear.
All right, Lena, we're getting pretty close. In position here. Hmm?
Yep, I'm monitoring, no changes yet.
Okay, no changes yet.
This is one that we would be able to sample as well. Okay, cool.
With the scoop.
Whatever you want.
Yeah, Sam was explaining that these are often collected just by scooping and letting the pieces break into the bag. They're that brittle.
That's what our little red
Try to
fingers are
perhaps,
for.
like do a little bit of a prod or physical manipulation.
We can. It sounds like that might we might break them.
Didn't you say, like, the tip of this thing's pretty...
We don't want to clog it. Yeah.
Okay.
I mean,
Then I think I'm not seeing a signal, so
I'm gonna try a different position.
right here, maybe close to the face.
Sam, is this a coral that
Gonna bring the sit down just so you know a little bit more.
part of it dies? Can you see like the dead part, or looking at this up close image that we had, like you can see polyps that are out in some, like this section, you can see the polyps out versus kind of go here, you don't have to make polyps.
Retract. It's not gonna be easy to see the skeleton, distinguish dead skeleton and living if it's only first.
Should we try and get it?
So we spent a good deal of time today working to
Really limited where the ROV can put the sensor.
confirm the operation of Solaris, just to make sure that we're when we don't see any signal that we're actually convinced that that's actually what we're seeing in the environment. And
That either died within the week or it's alive. So
Okay.
there's some
I don't think you're like close to the polyp, but I think you're in.
Scott, I think in terms of just being smart about our reagents, I feel like we can call it on this one now.
Okay.
Thanks.
okay, so we can retract the wand away, and
Is this something you're gonna want to sample?
I think Sam, we're still gonna collect.
Yeah, the coral that we did the SOLARIS measurement on, try to collect that one.
Even though wasn't a good sample?
Yes.
Okay. Because the ones down lower might be easier, but that's okay, we will still get it.
But you mean not a good sample, like you think we were in position enough to,
It's, from what I can tell from this camera, like, we were as close as we were gonna get.
okay.
I just think.
If we're as close as we're gonna get, then I want this sample, please.
You want that sample?
Yeah.
Okay. And is it gonna be a coral quiver or a bio box?
This one, I think we're gonna have to do bio box. I think we're gonna have to use that scoop bag.
Hold on real quick.
Oh, sorry.
Help him out with the camera. I got it.
I thought you got clear. Okay.
Yeah, just making sure. We'll just leave it out there for right now. And
I
then
just don't wanna hit it.
Yeah, we'll stow it later. Alright. So it was this top one up here that you were on, right?
Yeah, I was trying to get right inside there.
Yeah, you were you on this? You were on that one.
Yeah, I was like, I was like trying to get in here.
Yeah. I think if you were to go down with your, just grab to the right here. Only grab to the right.
30
How much of it do you need? Just coral cover size, right?
Yeah.
That goes up with the coral cover. Once it's gonna come out. Actually,
Oh, the scoops down, there's bag scoops down in the front.
there are some on there. It's just where that one's at, it's gonna be,
How hard is with the scoop bag?
yeah, not just crumble it straight down. But with our soft grips, I think he might be able to do it.
All right. So despite the fact that we did not measure superoxide, we are still collecting this as a specimen. These guys are apparently fairly brittle, so we're gonna try to collect this with
I just don't want to get you too close to these guys right here.
Is there a position way up there across? I have one around.
with some soft grips.
Just go over and get 3 done.
Yeah, you
Or
might
you
be a
do
bit
biology sample again?
This Carolyn sample is gonna be.
Going into a quiver?
What?
Going into a quiver?
Yeah, we might as well open a quiver And as we grab it, it's very hard to predict it, just to
Yeah, if
get
it's
an
big,
exact
you'll put it in
size.
Velvet.
Yeah.
You just scrape it over.
What's that?
Just scrape it over. I think you put down a big number. Give yourself the best chance.
Which quiver is it?
3.
Thank you.
All right, let me come up with the set.
You might end up putting it in the firebox and depending on how big.
You can use your
Yeah.
cutters.
Okay.
So the RV pilots are coming up with a strategy for approaching the sample collection here without losing too much of the sample as it's collected.
So, all right, let's go right here.
Some of these corals have proteinaceous skeletons, others are a little bit more calcareous and can be more brittle.
Red side on this, cutters on this side. One focus on this right here. You stay super stable when you're there. When you go in, it's grab. But only the right. That makes sense. You don't want your T4 jaws grabbing the cameras are not free. So can you get up and over it? You say? Oh, you don't. You don't have to. You can come down if you want.
Alright. So you're saying face the cutters this way?
Bring you in tighter if you need to.
I believe this is one of the first corallium we've seen
So make your
on this expedition.
stay live.
They normally more deep water.
Yeah, I'm trying to
Yeah.
Yeah.
get it to where I can have some.
It can be like, near the mesophotic, in the Mediterranean.
They can be found up to 2,000
You're
meters.
gonna want
Also down to 2,000 meters.
this would be like little bumps. Like little bumps. Go.
I just looked at that.
I've seen it at 1,400.
If you want me to do it,
In the Gulf?
if you want me to do it, I'll get it.
Some of these have been identified at 1,400 meters in the Gulf of Mexico by Dr. Sam Vosen.
This genus is actually the truest of coral. That was the first thing they coral corallium. When they made Julia, the pink one is the Mediterranean. This Clarktinians, people think that, and I think coral, that's the fake one. It happened because in
Well, the blades are fine.
ancient
You
times,
don't want
all the
this
cool
part
jewelry came from the Mediterranean to be corallium.
Really?
Then later on,
So you
when
need
the northern Europe developed,
You're going for this guy
they
right
wanted
here.
their own like coral jewelry trades. They checked their own waters. They dragged up cold water Clarktinians. They were all white. And they're like, oh, this is a white.
You might as well just pinch close a little bit now because you got that.
They stole. They started stealing the name.
No way.
Not the corals lost it. Now everyone thinks of
Coral is
building corals as the coral.
That's funny.
Keep this centered right
Is
in
it
there.
a corallium?
Rotate
Is
a
that
little
not
bit
the-
clockwise. There you go. Hold close. Pull
You
your
know,
arm
word
up
reef.
just a little bit.
Well, yeah. Word reef's like a Norwegian word for
Coming
structure in the
in.
water.
What is?
They're looking for
What's that?
their own.
Reef?
Yeah.
Come into it. Where are you? Okay, oh, I see. I see what happened there. Right, yeah. There, go to the right. Go this way. Start. So this one right here.
Oh, yeah.
Same thing. Bring your arm up.
So these organisms are filter feeders, of course, so you can see these colonies are perched up on this rock to take advantage of the current blowing food by. You can see that in the video as well.
Yep.
The current is moving from left to right.
Steve Oscars thinks it's stylized. Guess we'll see when we bring it up.
Do you have to use DNA to be sure?
Well, it depends on how obvious the morphology is. If they can see the polyps, if it's corallium, they'll have exactly 8
little bit, yeah.
the tentacles, and each tentacle have like side branches. But if it's
Right, there's a other one behind it.
stylaster, like Steve says, then they'll have a different number of tentacles and it won't be branched. So if we get a good enough look at it,
So optical or not optical?
correct.
Just want the red from there.
It looks like more, doesn't it?
It looks like more than eight paws, or 8 tentacles.
To your left.
Yeah, it's probably stylaster.
Like a little bit of pitching, not much.
The number of tentacles around.
That's pitching.
Yeah.
What crown is that?
Corallian's not for.
No, I can't.
Oh, Corallian.
I don't.
Crying at
That's what I'm
It
good
is
at.
an optical?
So it's a little too much pitching, but we're looking good. So that's
Yeah, Corallian. So there's some debate among some experts in the room and online about what exactly this is. Just pointing out, just emphasizing how difficult it is to visually identify these sometimes. Maybe a stylasterid.
This is
Coral. What?
it is not Corallian, it is stylaster.
Here, yours is also not coral, so it would have the 8 tentacles. But it looks very, it looks quick. Oh, we zoomed in on a picture, and so when I'm zooming in on that zoom in, that's a lot. So octopus means that they're polyp,
I would go
has
for
8
capture right
tentacles. Black corals,
Oh, just go
they
all the
have
way.
6
All the way.
tentacles.
There
They're hexacorals.
you go. There you go.
Oh, it's a lace coral.
There you go.
Oh.
This is right here.
Just start looking right.
So this is 1, 2, 3, 4, 5, 6, 7, 8,
Oh, that's good.
9,
Keep going right.
12.
You're doing good. If you drop that one piece, that's okay. You're doing good. There you go. Good job.
Wow.
I'd say yeah.
How many tentacles does stylasters have?
Oh, I don't know.
Use one of those, I guess.
More than 8.
All right, let's try to get over that quiver as quick as possible. And once you're here, hold that, hold also.
You think it's gonna fit in the quiver, or should we put it in the bioline?
It's up to you. I mean, I think the one piece definitely will, the one that's hanging. So
Yeah.
it's your call.
I'd rather put it in the bio box just to keep that
Roger that.
as much intact as possible. That's a really good sample.
Right. Go ahead and just show it around.
So you can see why the manipulator grabbed the coral, that it broke very easily and brittly. It's very brittle.
Larry, go in there.
So it's very delicate
Force
here.
out to you if you want.
Trying to get this sample
Right where you're at.
collected intact without it breaking off or falling out.
Whatever you do, I can actually bring it in there.
Yeah, start
We're
working it
over
in.
it now.
Yeah, start working it in. There you go, almost up.
So as not to let this break, we're gonna collect it in the bio box.
You looking good? Just go down. Down with just your elbow and pitch out at the same time.
Oh, that's bioline 1A.
Going down.
All right. Well,
A little bit after the right.
This is still SH.
And I would let it
629.
go.
Okay, so I'm
We're
gonna
gonna
follow
change
No.
species to slash.
There you go.
nice. Well done.
Great. That's a really good sample.
Yep. Great. That was a good sample. You can see most of it's all still intact at the bottom of the bio box there.
Do you want any more of it while we're here, or is that?
Barbox 1A.
Only if we decide to measure some more. This was one of the targets, because I think we just misidentified it from the video as Corallian. But
Okay.
If we can SOLARIS, another colony that we
Well,
can
then-
sample.
Here at this site.
Yeah, what we're trying to do is get about 4 or 5 colonies of a few species.
Yeah.
So,
Even if they're all located right at the same.
yeah, it's okay.
Okay.
We're just trying to, this is a good one because, well, I guess we don't have, this is stylized, I thought it was Corallian, but it's stylized. It's not technically showing Corallian as a target, but I think this is what we saw in the videos. And we were just wrong about being Corallian. It's actually stylized.
So you'd like to set up and collect
So I
another?
think we should try, yeah.
Okay.
If we can get like 3 or 4 colonies right here.
Okay.
Um, then I'm gonna
Base, go base coral.
then I'm gonna suggest
I'm
that.
just gonna do white dollars real quick.
Oh, great. That's a good thing right there.
Looks reddish to me.
I'm gonna suggest then if we're gonna take that we do all the Solaris measurements at one time for the different targets, and then sew the wand and then go collect. Does that make sense?
Yeah.
Okay. So once we get down with the white balance here, we're gonna pivot back over to using Solaris. And I don't know if you guys need to, if you feel like repositioning, and so Doug doesn't have to do an acrobatics with the arm, or
Yeah, I think since the current is going that way,
Left
it
to
might be
right?
better on that other side. when we first
Yeah,
do, right?
let's try it now. Definitely get more,
oh, the crab is climbing onto the ram. There's a little crab down here in the lower left,
He gonna go for
climbing onto the ram.
I'm on board. Might cover some, a lot of ground for us.
Let me know when you want to be relieved, Scott.
Okay.
This is like
it.
an output.
Let's see if he stays.
Oh, he's like, yeah, all right, I'll come. Oh no.
This thing's blue, baby.
Look, he's going for a ride.
Who is?
This guy.
I got him. He's my buddy now.
Oh.
He's my friend.
We got a little hermit crab. Oh, and he's even. This is like Mission Impossible stuff for him. Dun 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 dun. He's climbing. He's like, I'm gonna go get on the top here. It should be a little easier to hold on. Oh yeah, this is great. I've dreamt of this my entire life. Always wanted to fly.
Oh, I hope it doesn't go into a shell at the wrong moment. Yeah, exactly.
Just get. Excuse me. I think I'm in the aisle seat here.
Oh boy. You speak crab, Scott?
I don't speak crab. Would be an interesting language to learn. It's kind of amazing that he's still there.
Gonna leave the screen.
He's on the sit cam here. You can see.
See him on that camera?
He's climbing around on the rubber end of the
I'm
ram.
gonna try to rotate around just a little bit more. Get you set up.
I'm flying. No.
He was my friend.
Actually, you know, he was really always a daredevil. He really always dreamt of base jumping
Yes, without a parachute.
without a parachute.
Base jump.
So he was like, "This is my chance, Mom. Don't hold me back. I'm gonna do it."
best impression of Point Break.
This is comedy hour with Scott.
That was so sick. Best jump ever.
So this looked pinker earlier, is that?
Yeah, the white balance was off. I think.
Ah,
It's
okay.
white balance.
It's
So
still
you think
very
that-
pretty.
You think this is a good spot? It shouldn't be too tight in between there.
Yeah. Which way is the current coming? Is it just that way? Okay.
You can see that, yeah.
Nicole, you know what else we could have done with the experimental core is?
Trying to get
Uh,
us
we model the skeleton.
Yeah. We could have.
Scott, can you hand me my coffee, please?
We could still do it in the future. Opportunities there.
I'm not gonna lie, it's fine.
So we're using one of the hydraulic ram. That's not the right word. Hydraulic
Arms.
That one's called sock'em.
stock'em,
Sock'em.
sock'em,
We have rock'em and sock'em.
rock'em, and sock'em. This is
I have been wondering what the railing has been for this past week, and I did not know it was this simple.
this is one of its
feature.
uses. It also you can put push cores and things out there on it, probably, and coral quivers and things like that. The downside of having things on the outside of the ROV is that they're at more risk to getting knocked.
All these corals were- That's the big
Oh,
they're stylized.
yeah.
What did you see, Sam?
I said, I didn't notice how small they were until I see that bar now right close to them. And that was a big clue that it wasn't corallium. I mean, corallium had that dense of branching, maybe like 6 times taller.
Oh,
Oh, okay. So it's a really big one. Corallium?
Yeah, yeah, if it like has that many branches.
Yeah, okay.
You know, if that was like a tree, you know, it'd be like 50 feet tall.
Okay.
If it was corallium, it'd be 2 meters.
Okay.
Now, are we gonna have to sample whatever we measure?
Are you guys
yes.
turning
Yeah.
the?
So what we're gonna pick 2 or 3 different spots to measure, 2 different colonies, right? And make measurements, and then sew the one, and then go sample each of those. 2 or 3? Which,
So think about that.
Well, for species, we want 4 or 5, so 3, aim for 3.
okay. Well, we're gonna do 3 SOLARIS measurements here.
The thing you cut is way up there, so we had to do this sampling from this side and then go back around and kind of do that, but, I mean, I think
Super
these
brittle.
are all bad angles for grabbing.
Like you
right
touch it
there.
and just crumble and do.
This bottom one is doable.
But Doug did a
We'll probably
fantastic job.
break all that stuff.
So here's another question, Sam. Can the same
Maybe.
colonies go in the same bio box? I guess so.
That leads to the almost happened, so
The same bio box.
Yeah, but a different compartment.
I guess.
Okay.
Not on top of the same column.
What's that?
but it's closer.
Yeah, we're just trying to figure out which one to sample, so that if we actually
Yeah,
have
sure.
to physically sample it,
If they're all
we
0.
can get to it.
There we go. We'll see.
I don't really know.
So yeah, so to correct some earlier we were identifying this as a corallium, and we've been corrected now, and I realized that actually with some help from online information here with some listeners and some high-res camera zooming, that this is actually a stylastrid.
Well, it's not corallium, so now it's.
Could you zoom in on the SID as well?
This measurement, it's SH. The SH number just
Wow, no, I said
18,630. 18,630.
That's US versus, I think.
All right, it's nearly 7 o'clock here on Desecheo Ridge. 310 meters, and it's good and close. I like that. Taking our second measurement with SOLARIS, looking for some extracellular superoxide, which has been identified or has been observed with this instrument in loads of different kinds of corals and different deep sea settings. Here we're using it for the first time in the mesophotic and aphotic zones around Puerto Rico. Is it possible to get the one even closer? Not without hitting it.
One thing that it could be is that we're on the wrong side of it.
Yeah.
Depending on the current, it could need to be on the other side. So, but he's out as close as you can get without
Knocking
making
it.
this thing crumble down to the bottom.
Yeah.
And then have to go pick it up.
Okay. Not.
All right, we'll go to the back side.
So this is the first stylastrid that we've measured.
Yep.
So
Figure out the trip angle.
Some new information.
Swing in before you. Yeah, something like that. Maybe on that side. Zoom in. I can see that shadow is pretty tight up, so you were really close.
Polyps are out.
It's about-
Wow.
He is really close. See the shadows?
Yeah.
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 You can even see like shimmer.
Yeah. This is a great perspective where you can see the polyps out.
Cutting off, but it's not on the screen there.
So William was working on the software, that's just the state situation that has it. Starts to get up.
Oh, I see.
So then this back will push that.
Okay.
So that because there's like, there's that position, it's much better.
Yeah. Okay. All right, looks like nothing. So.
Here, pick
stop
another.
So we don't waste all that.
Yeah, we can pick another spot. Nice job, Doug.
Maybe in that really dense area on the bottom right.
Down here.
Yeah, just possible, or a little farther up. It's
Up?
hard for me to tell.
Over?
No,
So,
in between the two spots that you were just circling. Yeah.
Here?
Yeah, in there maybe. Seems a little dense, maybe.
is that here?
Yeah.
Yeah, maybe in between those two,
See what you're talking about?
if that's
Oh,
possible.
sorry, I wasn't.
Trying to like
Here.
sneak into there.
Yeah.
Which,
So this will be the second or the third sample, right?
Third, yeah.
Yeah, the third measurement for satellites.
So this
The
is
hermit
about
crab?
04.
Yep. Several hermit crabs.
And give this SH1E 631.
631, yes. SH 18631.
looks like they're still far away with these segments. Kind of hard to tell.
Yeah, so getting perspective on how to position things in three-dimensional space with flat screens is difficult.
Right now, see where you're
We'll use multiple cameras with multiple angles, try to get that
See my shadows.
sorted out. And we can use the shadows and different lights to try to.
So
Oh.
you're in there. He's weaved in there right now.
Yep. All right. I think that looks like a pretty tight position. Whoa.
No.
What is
It had
that?
been turned off. So it's just coming back.
Everybody was like, "What? I see a signal."
What is that?
It was just they turned it off and I turned it back on.
Just trying to save some of their agent, so we're not burning through it.
No.
Yeah. That's what we were wanting to see. And have seen in the past. Corals are just taunting us.
Yes, thank you, Steve Auskovich, online, helping to identify this as a stellar.
Does that mean that's me? Yep. I just fell down with the step. I don't know
I
how
think
you
we're
did
good.
that.
Yeah.
You know that? Well done.
Nice recovery, Doug.
Uh-huh.
Making up for some slack that was dropped there on you.
You guys good? You want to turn it off? Yeah.
Okay.
Yeah, I think we're good. That we can confidently say there was no signal.
We collect those when we measure
Okay, I'll get back in there. I just didn't want anything to get ripped off.
So the ROV is slightly buoyant, and the pilots have to use a little bit of downward thrust to hold position on bottom, and we were doing that essentially with one finger touching the rock,
Is that going in the same pile box?
We can go.
holding onto the ground or this rock here, and it's easy for a current to change and shift the balance in that case.
Branches in the same compartment, it's really hard to distinguish them.
Understood.
Uh-oh. Losing our monitors.
Well, that's new.
No,
Let's
we'll do
do-
that. They'll cycle.
This keeps happening again.
Just give it,
20 seconds? 20 seconds? 30 seconds? Oh, there goes. Did you?
Those are direct.
it'll come back, I promise.
You want to do something crazy?
Yeah, let's do something crazy. Scoop.
There it
You were never worried, not for 1 minute.
is. I was worried the first 3 times this happened, but not anymore.
I
I'm gonna put the pile box.
I think it was the last watch. I counted up something like 45 monitors in this room. If you count, the quad is 4.
Oh, yeah.
That's 4, 5, 6, 7.
Oh, I was wondering where you're getting those numbers from. It's like half a
Yeah, but I mean, different. Different. Essentially, those are 4 different monitors, right?
Now we give you all of these.
You could. What?
In theory.
Yeah.
Let's see if we can switch that pile box. Okay. This side is so good.
We're here at about 310 meters on the Desecheo Ridge in Puerto Rico.
I'm gonna try to get that corner of pile box
310 meters is
right underneath.
below the penetration of surface light. So the animals down here rely or on not directly on photosynthetic sources of energy. They do get a lot of food from photosynthetic production that rains down into these depths. You can see the abundance of sponges in the background there on the rocks here. Those are, of course, filter feeders, as are corals. Actually, they're not. Corals aren't, I guess, filter feeders. That sounds.
No,
What do you call them?
Well,
they're.
they are, but not like a sponge is. They do catch little.
They're not like a sponge, or they're not like a mollusk that
Filters water.
that filters through yeah.
Yeah, but they don't actively filter like a net. They
They just-
passively. They're passive.
Yeah, they're kind of passive filters, kind of have their tentacles reaching out and grabbing, whereas the sponges are actively using they're pumping. They can actually actively pump water through and trap dissolved organic and particulate organic matter to feed. So clearly, this area here is a good habitat for sponges. Are you doing?
I'm handling the pictures during this dive, so we don't have to do that.
I was just gonna do
Just
it.
this part I wanted to get done. Yeah.
We're sampling corals with the ROV, literally with the ROV. These are very brittle specimens here, so J-Rod's basically using the ROV to catch broken pieces as we come upward from underneath. These are specimens that we just finished measuring for superoxide. They did not appear to be actively producing superoxide at this point. Which actually, as we know from experience, doesn't mean they might not be doing it at other times.
Just not right now.
Uh-oh.
That's not the one I'm going for. This is.
Oh, is this there something
Oh,
crazy?
wow, those are so brittle.
So little.
I guess I should log
Oops.
the coral collection.
Got another sample.
Sorry, you'll know the difference between that and
Yeah.
Alright.
Okay.
That is the one that you sampled.
Yeah.
With SOLARIS sitting in
Okay, so this is
That was-
the. I got a
18,632.
little bit of extra.
Watch.
630 is the one that
Yeah,
was on in the right.
6:30. Yeah, went to be. Yeah.
And then we got a piece of a colony we didn't measure with SOLARIS, Nicole, and that one's gonna be 32.
Which one is this going in the bin right now?
The one in the
That's
box
H.
B is 30.
630. Next up, $5 more.
Nice. Some smooth operating there, J-Rod.
Do not start singing with Scott.
I wouldn't dream of it, Colleen.
Okay, then I get to make another one.
Prime for a smooth operator.
It was.
I think we're not allowed to sing. Otherwise, I might burst out into song. Maybe an ABBA cover or something.
Alright, are y'all wanting to stay around here or start moving?
I think we start transiting away
Yeah.
upward towards waypoint
Thank you.
18.
It's fun.
I haven't seen any sponges.
I know, we
We're
gotta
gonna
find
abandon
them. We need
that
to
second
transit.
colony that we
There's
measured with
just
SOLARIS.
there's just 7 million of them here,
We're gonna abandon that second.
but we haven't found any barrel
what
sponges.
are we
yet.
gonna
The
do?
second colony that we measured with SOLARIS.
Oh, you wanna get
Oh,
that?
did we,
Do you need
I
that?
didn't know that we didn't sample that one.
Well, I don't know that one was weaved in around a whole bunch of stuff.
Okay.
That was like, we were inside of 3 different corals, that would be, that
Hard.
kind of stuff.
We didn't get it here.
Alright, I'll put a note in the SOLARIS measurement that did not collected, so it's 18 or 6.3.
Yeah, those are some skills.
631 is the one where they can.
Okay, 631 did not collect.
So we have generally seen superoxide produced by various corals in the deep sea. Previous expedition, yes, that listener Steve Auskovich here, I see him online, was also part of on the Atlantis. It was the first time we deployed the instrument and made measurements off the coast of California. The Monterey Bay National Marine Sanctuary, Davidson Seamount, and then further south. And we saw pretty clear signals from sponges and corals in those regions. And it's true, though, however, that was an entirely different ocean with a different chemical composition of its own, different species of corals, different conditions altogether. All of these factors may play into how and why these organisms may or may not be producing superoxide. So it's very much some, relatively simple-minded questions that we're able to now start asking with this new technology.
I'm just using them to,
So we're learning all along the way here.
should be good now.
Yeah, Kate, there's loads of sponges here, but it's all just
I know,
this one
not the-
particular
not
kind.
the
Not the
shape we need.
not the emergent
You want-
sponges.
You want a tube?
Yeah, we'd like a tube, yeah. Or as I call them, they're biogeochemically convenient sponges. That's what we're looking for.
Measurable.
Exactly.
Inhale and exhale easily.
Exactly.
No, we're going.
Was that a?
An example? Somebody doesn't like
And then
sponges.
might as well just
No,
go
That was-
west,
I was thinking that I'll have to communicate to
because I'll be there no time.
the next team, which branches of that SOLARIS or which sample that was, that one part of the biolog.
Oh, you have to draw it or something. That one.
Well, I mean, we got a good look at it, so we got a big piece and then a small.
Yeah, we're starting that towards
Yeah,
the
you
west.
just did a small piece.
So I'm gonna do it.
All right, so we're heading west northwest to the next waypoint, waypoint 18. It's 7:15.
Is that more the same back there? See, you got two corals, you got one off to the
Yep.
right, and then another colony up
Yep.
on that rock over there?
It looks like the same thing, right, Sam?
Yeah, no, this is not last week. This is not last week.
That one is, and then this one over here. Same thing, right?
Oh,
It's the same
Yeah.
stuff.
is that a Primaris though?
Where? The little yellow
Yellow?
stuff? No, it's like a different kind of style.
Okay.
Let's keep looking, maybe we'll.
Oh, there's a little,
find
what is that little, right in the center, the little yellow ball, right in the center.
Oh, I see. I think that's the same stuff, but I will come up with
It almost looked like a, what do you call it? Not
Oh,
Montastic.
varieties?
Yeah.
Ooh, the color looks really green.
Oh, it's a snail.
That's just because I'm still so far away. When I get closer, you'll get its actual color.
Wow, that's so pretty.
We've seen a couple of these.
We have seen a couple of these.
Yeah, I'm just hitting the headache right on the start.
The headache rack.
Yeah, it stinks vertical right here. Very, very good.
I think we can just keep heading up to the next waypoint.
Yeah, I'm just waiting for the vessel to get moving ahead, so I'll just skim around this stuff real quick. If anything I want to stop, let me know.
Okay.
Just slow it down and do that. Yeah, I feel fine.
So wondering if there's anyone online who knows and loves and knows a lot about deep sea sponges, can give us any sort of idea on this. It's almost like a monoculture here, of, to my untrained eye.
Thought we had an idea on that this morning, Scott.
Did we? Okay.
Yeah,
The sponges?
yes, almost there.
Or at least days, she, if I put down a sponge,
Now it seems like they're white pellets.
Alright.
Anymore. It's just
Corallistes.
that.
Corallistes?
Yes.
I have to do the white pellets again. What's that guy
Type
right
of
there?
a sponge.
Oh, that's that one that we looked at earlier, right?
Yeah, so little,
Oh, yeah, one of those pink ones.
yeah.
It's a little pink.
Deep coral. These rocks are cool.
Be a good place for an octopus to hide out.
Mm-hmm.
Maybe
Little
there is,
cave.
and it's blending in.
Oh, that's a big cliff. Floods. Yeah, it's fine. I'll get up and over this real quickly. No, that's good. My locals going this way, not over and stuff, so.
made it into the 200s.
Look at that urchin.
Where? Oh yeah.
Big.
You want to zoom in on it?
Sure, let's get a little zoom on that.
Ah, cool.
Wow, those look like huge spikes.
That
Yeah.
would hurt.
He's walking around.
Is it only?
What about like grid
Sushi?
pattern on the rocks? See that, like below the urchin?
Yeah, yeah.
Yeah.
Yeah,
Because I think the rest of it is
I
like.
think this is all carbonate. But
That's an interesting
There's another
like,
one up there.
grid pattern. Squares.
there's other places I've seen bedding. Yeah, up there you can see.
Yeah.
I don't know if it's carbonate or if this is
That
some
thing
sort of.
hanging down right there.
Oh yeah.
Oh yeah, is that crinoid?
Looks like
Yep.
Oh, it's an interesting one.
That's really cool.
It's upside down.
Is it cheated?
Oh look,
it. Whoa.
I don't know. Because
is that a stalked crinoid? That
A stalk crinoid would have a bunch
Very
more
young.
stalk.
Oh, there's
I think it's
another.
very young.
there's another urchin
Yeah,
there.
very young one.
Yeah, another urchin there.
Before it's stalked as long.
what about that?
Oh yeah, it doesn't look like it's got the.
And then there's little white ones too.
I've never seen one so thick.
It's got really thick.
I don't know what shade it is.
Could it be a carnivorous sponge?
So,
I don't know enough.
What does that look like? All those little tiny white corals there.
Grenade sponge. Hanged it.
Named it.
What did you name this stuff?
He called it a grenade sponge, except that it's a crinoid.
It's a granite.
I respect that name.
Pineapple. Pineapple crinoid.
Oh, it looks like cool. Did you see that?
weird.
I have not seen
What's behind it? I can't tell.
All this stuff hanging out under there. Yeah.
but
Super cool. It's cool how he closed up.
Yeah, he's like, leave it alone. Go
Oh.
away.
Is he gonna open?
Someone in the chat, I think I
Oh.
clarified it.
Oh.
Is he opening?
Yeah,
Holopus crinoid. Yes, this is the same location-ish
oh.
as earlier today. We got a positive ID on the
Yep.
holopus.
Oh yeah. Look
Yeah.
at that.
It's a
Yeah.
kind of.
And do they normally grow hanging from upside down like that? Looks kind of
It looks like
Well,
you
maybe.
might.
maybe. Yeah.
It's a grenade.
That's
It's a
what-
grenade. Also a grenade. Also a pineapple.
Wow.
Related to sea stars.
Is it sessile? Like, is it trash?
Starting to open up, right?
Do
It's
you
like getting used to us.
want to wait for it to open up, or do you want me to keep it?
Let's give it a few seconds, see if it opens.
What kind of crinoid is it called?
Hand grenade.
H-O-L-O-P-U-S. Brazil.
Keep going.
Sure.
Doesn't look like it's gonna do anything else.
Oh, it closed right back up.
Yeah.
That's
Were we just hovering?
No, I just leaned up against the wall real quick.
Oh.
Oh, yeah, that's really
It's like,
steady.
well, that'd be super stable if we were flying.
That was stable. All the way. Another urchin.
Yeah. That was nice.
Are the green dots on the map, the waypoints?
So that we're heading towards waypoint 18 right now, that blue dot.
Oh, it's
And then there's a 19 there.
Oh, crap.
So we're kind of going up this ridge.
See that crab?
Yeah.
Crab.
Oh, there it is. Look at that.
Gal-
Oh yeah.
Galligator? No, it's spider crab?
All right, we are moving upward into slightly shallower depths, 292 meters. Again, along the Desecheo Ridge. Desecheo Ridge. We are in full sponge territory here. Corallastes sponges. With some. Oh, there's another little.
Yep, hand grenade.
Yep.
Nice opioid.
Is that sargassum over there?
There, oh, there's a big snail again.
That's not it.
Y'all see what y'all were looking at?
Colleen, is that sargassum? That's
Where?
Oh, the yellow one?
Oh, I just moved it.
Oh,
This is probably.
right here,
Do you see?
right here, right here. Colin.
Is that sargassum?
Looks like it.
It is.
Yeah,
Why did it move so
Wow.
strongly?
should grab it.
That was a good.
You want to grab it?
Yeah, let's grab it.
You want it in a quiver? Do you want a suction sample? What do you want?
Suction.
Will suction work?
Yeah, let's just slurp it.
Well, there's a separate
You
one.
wanna do it quick?
Good eye, Kate.
Oh,
You're
yeah.
just gonna go by it.
And it-
Somewhere.
and it'd be, this is our second sargassum crab. Third.
Yeah, I mean,
Did
do
we
you
get
want
a
me to-
sargassum one?
With the
Look at
hoses, once I pull it, it's.
I
got one
I
this
never-
morning.
Died.
From
This
the
is
surface,
our
probably
third
better
from the surface.
So it's probably better for me to
Yeah,
just
yeah.
put that right there.
But we're interested in like the
Yeah, the fate.
material that.
Yeah.
So did you give that IND like SARC too?
No. Did today? Did you?
I did. Yeah, I have
So
SARC too.
what we
Okay,
can do
so this
is
would
we
be
can
SARC
put
3.
a
Yep.
jar on one,
I
and
didn't.
then you can go
I
in
didn't
and
log
grab
it
it,
because it was a
and then
surface
put it
sample.
right by the nozzle,
But
and we'll suck it in.
And I will see like the gap too.
So as you may know, there's a very large, extremely large bloom of sargassum, a floating brown algae. Yeah.
brown.
That you're probably very familiar with. Washes up on shores a lot. There's a big giant patch of it out in the Atlantic Ocean. It's quite common and varies in abundance year to year. And some colleagues of ours who we are interested in whether if we see any of the sargassum sinking to the seafloor here, what happens to that, all of that biomass that forms on the sea surface. So Colleen promised to collect some if we saw any. Happy to do that since we're here in the neighborhood.
Can I get to sit up?
Oh, it's looking.
Do we need to sit up? You're all right. You're ready?
Scott, can you write that? Are you writing in this book here?
Yeah.
Just want to keep an eye on- Can you shoulder light? Just trying to watch out for swears.
Where would we like to store this?
We're gonna go suction sample, I thought?
Yep.
Right.
Suction is sounds great.
Section 1?
And we have a SARG number for this.
Right away.
3. SARC 3.
Yeah, go away. Grab it. One, yeah.
Well done.
Sit coming down. All right, you're on jar 1, right? the farm? Not the internal one. Oh, sorry.
Sorry about that.
There you go. Yeah, so we can just see what we're doing right now. I'm gonna come. Rotate? Yeah, rotate a little. I think you're too far in. We can put it up. Yeah. Yeah, put the handle. Yeah, there you go. There you go, you're on. There you go. Go ahead and wake up.
Okay. I'm trying to watch the SOLARIS, or watch the sargassum get slurped up down there.
Someone there.
There it goes.
Can you shoulder right a little bit?
Ooh, ooh.
Yeah, go right. Yeah. I'm just gonna lateral left to help it out. Doesn't want to go all the way.
And we're watching for it to show up in the slurp.
It just went through the nozzle.
And
Where are we looking?
R2.
Oh, in there?
It's starting to come in now.
Oh, I see.
Let me just set up real quick. Going to shut it off and
Did you slurp yet?
yeah,
Which slurp is in it?
it's
go into
chamber
one.
one.
It's just a big thing, so it's gonna give us a little trouble.
And will you log that?
Yep,
SR3?
yep, start 3 and slurp 1.
Who went in the hose?
Yeah, I'm trying to tackle it. Trying to help it.
Come on, come through. There's so many air bubbles in there. That's the problem. We got air in the hose.
Shots in the face with the water. It's not helping anything.
Is it stuck in the tube?
It's stuck in the tube right now. I'm trying to work it.
Uh-oh. Was it a pretty big piece? Yeah.
I thought it would have torn up a little bit, but it didn't.
Maybe it's stuck in the bed. Maybe more down a little bit.
Well, we're trying to get this sargassum sucked up into the chamber. It's stuck in the tube.
Do me a favor. Grab the hose. Just don't grab it and just shake it.
We're gonna shake the hose.
Shamelessly self-plugged. Gotta get the views.
Got a grilling TikTok channel.
All right, I'm gonna
Well-
stop it real quick and start it up.
So I think Scott mentioned there's a large sargassum belt that's moving across the Atlantic. And there's a lot of scientists that are interested in trying to figure out where it's starting and why and where it goes and where it ends up. So some of it sinks to the bottom, but we don't know how long it takes for the sargassum to degrade.
I don't like that heat
back and forth.
Might have to try to pull down on it, get that airboat a little bit.
Apparently, this year is going to be kind of an unprecedented year for how large this sargassum belt is going to be. And I think we might have gotten lucky in terms of timing. So it's supposed to, kind of this large area, it's supposed to reach Puerto Rico pretty soon. And we're seeing signs of it. We see small patches of sargassum. I actually collected some this morning from the surface waters. So there are little patches that go by the ship quite frequently, but
Glass sponge?
yeah. Proliferating so much, and then how proliferating so much, and then how the currents in the ocean, the transport is impacting where it ends up.
That's what all this stuff.
Right.
And ultimately, it's fate. One of the things I'm interested in sargassum is that they tend to accumulate a lot of the metal arsenic. And this is because sargassum does.
That's why you can't use as a fertilizer.
And this is because the arsenate anion, so it's arsenic with 4 oxygens around it, looks a lot like phosphate. So the transporters that pull phosphate into the seaweed, they can't tell whether or not it's arsenate or phosphate. And so when the phosphorus concentrations are low in the waters, the seaweed or sargassum will just start pulling up arsenate or arsenic. And so this happens not just in sargassum, this happens a lot in plants and phytoplankton. So anything that has phosphate transporters, sometimes they can get tricked into pulling up arsenic instead. Most species will kick it back out, have transporters to kick arsenic back out. Some will just, ooh. Look at that fish. Cool.
Ew, what is this light doing down here?
Yeah, so many species can kick the arsenic back out, but some hyperaccumulate. So and this is actually one of the processes that are used to clean up, say, contaminated waters. You get hyperaccumulating plants and you plant them, and then they'll pull up all the arsenic, and then you can harvest the plants. Well, sargassum seems to be one of those hyperaccumulators, accumulates a lot of arsenic. And so areas where sargassum is accumulating, say on the beaches, you're going to have what could be potential accumulation of arsenic within that degrading seaweed.
How much arsenic is just in the open water? It's pretty low. Just slowly accumulates lots.
So now you want to go downhill?
I think we were trying to stay on the ridge, so there was the waypoint kind of to the west. What is that?
Human impact?
Oh,
It's like a flower printed something.
oh, that's too bad.
Just the So live in ice river.
That's gonna be the types of fossils.
It's not gonna last very long.
I don't think it's like got a foam on the inside.
Yeah, exactly.
That is an amazing scene. Just so many sponges.
The plastic disappears by like a trace of this shit. It's how fast. Action figure buried in sediment.
Bridge, ROV, we're starting
Or like Stretch Armstrong, they'll just find his head. The body was
Yeah, they're evolved like
Interesting. I just got into the sand belt too.
Yeah, we have a little
It looks like it's about to
sandy area here.
Oh, yeah.
There's the big. We're gonna get a waypoint 19.
That's the plan, right?
Okay.
But you want me to come to the top, right?
Right.
And then go there, so keep coming up this hill.
Oh,
first.
so yeah, that would be great. Yeah, if we could get up to the ridge and then follow along, that would be great.
There's a lot of AOC in the water.
We're gonna try to get out of the sand here, back to some of the hard substrate where we're gonna find corals and sponges. Someone in the room mentioned that we're feeding cattle seaweed. And the reason for that is it can reduce methane. Some seaweed can, there's been some findings as, feeding some seaweed to cattle can reduce the emission of methane from the animal. So something like sargassum would not be a good choice given how it hyperaccumulates the toxin arsenic. So one of the things we have to be careful about in terms of feeding cattle seaweed, because sargassum is not the only one that is a hyperaccumulator. What is that one there?
This little guy.
He's like waving at us.
You want to have a look at them?
Yeah, just a quick look.
Well, guys, are gonna
He's like, I'm all alone in fields of sponges.
He's a pioneer.
He's a
Oh,
pioneer.
look at that. That's a cool one. Yellow body.
Oh, wow.
Wait, what are we looking at?
There's Ronald McDonald.
Yeah, I see a
I have no idea what you guys are talking about.
Right. Middle to the right screen, right?
Huh?
Right.
Oh, that little guy?
Yeah.
Oh, I see. What is that? Is it
Right.
like a basket star?
Brittle star.
Oh, brittle star.
Without branching arms.
Looks like he's hanging on.
Ronald McDonald.
Yes, it's red, yellow, white. That's common color. Have a business dressing up like that. There's some of the white ones. It was an active world.
Trying to get
Too
you.
small to measure, too few to sample.
All right, so keep going, or?
Yeah.
Okay. What about this bigger one that was over here?
I'll just take a quick look to see if it looks like that little one just passed.
Oh, little purple fish.
Oh.
Look at him ripping in the dragon. Shaking in his boots when
Oh, only a couple calls
Black
are
hole.
out.
but-
Oh, yeah.
Are those zoanthus?
It's a black hole, right?
No, it's an octopus. It has a black skeleton. It's like,
Oh, okay.
Are the rest of the polyps just pulled in?
Some are, some aren't. It's like half and half. Some are partially pulled in.
I think the current is doing that. There's like a snail up on that one tip, right?
Some of them eat the corals. Oh, there's some growing on it, but it's yellow.
How big is that?
Oh, that's about like this big tall, like a few inches tall. Okay, cool, thanks.
Roger that.
We will not measure that poor terrified coral.
You get like a, it's like a, looks like a white paramercian.
There's a giant.
Is that what you say?
I put off the coral, but I can say very much.
It looks like family paramercian.
Wow,
Another one of those, but that one's big.
that's a good size.
Like this, there's the lasers. That's a big old.
Yeah, that's a.
Yeah, it's 10 centimeters across.
You can wear that as a hat.
Almost.
All right, so it keeps going uphill up there. So do you want me to come back down south? If I keep following up, I'm gonna be going.
Yeah, I guess so. Yeah,
So I'll come back down south.
yeah.
Okay, sorry about that.
No, that's all right.
I don't know where they saw all their corals.
I remember.
Now there's some sponges.
Some glass sponges.
This is all I can see the whole video. Oh, yeah.
What do you think with those glass sponges? It might be interesting to see what that flow regime is. They don't have that central oxide, like the big opening, but they do have a.
We could try it.
Might be good to try to get one that's a little more, that has a little deeper,
Yeah.
which we have seen. We just
Yeah.
have to.
Also where there's less, maybe less crazy current, so that it doesn't just like sweep the dye off.
Yeah.
Oh, it's the fluorescent on the RV. Just-
Yep.
Yeah, it is.
Where's the name of these sponges around here? Corallastes?
Coral is.
Listes.
That one is setting price.
Which is a type of that is spongy.
Aha. Okay.
Very good with the south.
20 different species?
Quite a bit of that. Okay, that's me back on like the track line pretty much. Underway 2019. Whole bunch of little stalked things right here. It looks like it, where there's no cracks. Those are too small for you guys, right?
Yeah, that little white sponge there. That looks really small.
Yeah.
I mean, we could take the small ones for the fungal cultivation. We want to have a big mix, but.
We could look around a little bit more.
Yeah, it's just my fault. What's that blotch thing in the middle, kind of, on that rock?
That one?
What is
Is that a glass sponge?
Oh.
Definitely sponge.
Yeah.
Well, actually,
Oh, yeah, that's
It's a sponge. I don't know if it's a glass sponge though.
What's that?
Could be.
What is that?
What, all the urchins?
This-
Spines? Yeah.
Oh, an urchin spine. Oh, cool. So is this like the osculum, Scott, here?
Yeah.
Yeah. And then like other little chambers here?
How big is this one? It's like 10 centimeters, like.
Turn the lights just back on.
Someone says looks like a saccocalyx. Oh, look at this guy over here on the right.
Oh, brittle star?
Yeah. That's cool.
He's coming to attack.
He's coming for us.
Look at the little squat lobster.
Oh, the little lobster.
Where's the squat lobster?
Yeah, Sacrocalyx.
How do you spell that?
He's just peeking out of that little hole.
Alyx.
Wow. This is cool watching him.
This guy's gonna move pretty fast. These guys.
There's a bunch of different types of sponges just in this view here. There's here, and then these little purple guys over here,
Yeah, they're tiny.
and then this little encrusting sponge. More of these like worm casts, right, that we saw. Is that what this is?
Yeah, I saw some of that earlier too.
It's almost like it's a lot of coral rubble too, maybe.
Oh, yeah.
Oh, the what was it? It was some sort of reef. They said cephalid.
Serpulid.
Serpulid.
That was the worms.
No, but this kind of looks the same, right? It looks like a bunch of casts.
I think this is more coral or sponge rubble because they don't
Yeah,
look
that looks like
hollow.
snow ash.
Oh, they don't look hollow? Okay.
That one kind of.
There's one little
This looks like it's half of a cast,
That
right?
one has all the little dots on it.
Little, yeah.
Yeah, that one does.
This looks like,
I think there's a mix of things come from above.
yeah.
Oh, I mean.
Come from above.
If you look up here, like, I mean,
Higher
that's just a wall of stuff that fall down here.
Fall down, yeah. Oh. Ah!
He's like, look at me, look at me.
Why are you staring at that thing? Look how purple I am. Oh, he's going for the fish.
sort of opioids.
Wow, that is so cool. Yeah, a bunch more sponges here. These little guys. This up here.
You could say slag.
Oh, 100%. Yes.
Oh, look at that one.
Oh, a little guy.
Took me a minute to get.
What is all this like, this white kind of webby looking stuff?
Avatar.
Could be
Not
like,
like a
Fungi?
encrusting sponge?
no.
Classic.
That was really thin. Is it some sort of fungal?
You want me to zoom back in on it? Or?
Yeah, that would be great if you don't mind.
Ah, too much. My bad.
That'll teach you the lingo.
Oh, this little cup puller.
There is a cup coral there, yeah.
Oh, this little guy right
Yeah.
here.
Yeah.
Oop.
Is that a scleractinian?
Yes.
One little polyp.
Yeah. Solitary.
Solitary cup.
Oh, pose press.
Wow, he's definitely posing. You show off.
Look at all those little holes in the back.
He's like, that's enough. Huh.
Can you imagine if that brittle star, this was like, he's 80 feet across or something, walking around.
Try to get this sponge right here. Check it. Alright. Y'all ready to keep going?
This is in the whip world to
Yeah.
the left of it.
I'll do that.
Oh, what is that?
Oh.
Sample these guys shallow.
Look, another solitary.
These guys have a left guard.
We're trying to get him to come back south. Pretty bad.
Who's that little purple nubbin down there?
I think I might get another little sponge.
the white thing on top just behind it?
Keep on moving
Yeah,
down.
behind it.
Yeah,
and then
keep on
it
moving
goes
down.
yeah. Oh, there's okay.
What, do you want to break?
I love these close-up zooms.
Think we should measure this, Sam?
Oh, there's a black scroll right here. Did you
Yeah.
notice the style of athletes?
Oh, right there, you guys wanted one of those?
Did you see that? It's black scroll that was smashed right next to it.
Yeah.
Stylopathy's right there.
Yep.
What is
Yeah,
that?
there's so much.
What is that?
Yeah.
Sorry.
What the heck?
See it closer to it again.
There's so much in this view.
I know that view is probably this big.
Can we tell which way the current is? The current going this way?
It's going left to right.
Yeah.
Oh, there's a little shrimp.
Oh, is there something? Is that a little shrimp there?
Yeah.
Yeah, that coral right there is probably this big, right?
Which thing?
Oh, yeah, it's
It's
got
a little
to.
bigger.
Really? Look at the size of it. There's a circular here.
Should we move on, or do we think and look for a bigger?
I think we should move on. I mean, that's a good sign. There might be some other corals in there.
Yeah, we're getting up shallow. We're at 275 now.
Okay. I know it's hard to fight the urge to measure things.
About
That big? It looks huge though on the screen.
Apparently Nicella can appear at depth up to.
All right, we're gonna keep moving on, and we're getting a little shallower here, so
Alright, so just up into the right?
yeah, towards the waypoint there, 19. I think we're.
It would be kind of cool if we see a bunch more Nicella. It would be cool to target it at this depth too, because it kind of directly gets at our depth questions. If it's the same species that ranges this far.
Right, right. So, do you remember what depth we measured it before?
It's like
Like in the
150, I
120s?
think. Yeah, 120.
Okay. Yeah, so that's quite a range, depth range, if that's the same. Well, it looks like we have some other corals here. What are those guys in the wind there, Sam?
I don't know. Maybe Nicella. There's some branch from it.
Some branching.
Interesting.
Look at it shaking.
No, we can keep moving. Yeah, he's really shaking. That current. Is it pretty strong?
Yeah. I'll find out more after yesterday on Instagram. It's really bad.
going to that, we're going to 18?
19.
Oh, 19. Oh. I should move this a little bit. Oh, right. No, like, no. Yeah, move there. It's fine.
So I see a lot of sand over here.
Great job. Just know when you need to do the direction.
There is so much stuff in the water here. Now we're at about 270. Lots of sponges. So we're keeping our eyes out for some corals, some of the species we were hoping to target. A little purple fish. Looks like the guy's following us.
Red fish, blue fish.
If you see a sponge that looks like a cup one, you know, that you want to sample.
Can we find a squirt before the scene? Think Kate would get mad if we did when she wasn't here?
Yeah, right.
She was there.
No, I don't know. She was there for a second. Probably watching from the mess. Wow.
Oh, there's an urchin. There's another urchin right there, I guess. Oh yeah, you can see their current just kicking up sand. Oh, there's another urchin up here. There's a bunch of them. There. Oh, that's okay. Oh, another one there.
Just for a second.
Not seeing a lot of coral. What is this white here? Oh, another sponge. No shortage of sponges right now. Is that another one down here? Like a big blob of a sponge.
Orbit by.
There's this little fish in there. Couple little fish. What is this? This is that a sponge right there too? This,
Yeah, I think so.
yeah.
With this much biomass covered with sponges, you wonder
There's a crab right here.
what's the flux of water through the sponges here, right?
Yeah.
It must be high. How much water is actually
Well, there's a big sponge of an oscillon.
Oh yeah.
Where? Where do you see it?
Okay.
This camera.
In the sit cam right there.
Oh, I don't see it here.
Coming up. Ding.
There it is.
Oh, there it is.
Oh, it's not so big.
The yellow one?
Yeah.
There's a bigger, there's some yellow ones right here, but left. And that's the top
Looks like a glass sponge, right? That's the last one.
What is this here, Sam? This
Some kind of
Or
coral,
Nicole?
Either skylaster or skylaster.
So what makes this area such a sponge haven?
I'm wondering if it's something to do with the high, like there's high productivity
The currents through here?
and the depth is kind of like organic matter sinking out of the surface layer.
particularly interested in.
And
Is there something you want to look at, Scott?
I just want to know what the dissolved oxygen. Can you look at the Sievert, the CDD real quick?
So Steve says
Yours right here.
worth a zoom on that squiggly white coral. Could be madrasis. Is that I think that's the one on the right here. Kind of looks like a spooky coral. Yeah, this one here.
Stone coral madrasis.
Yeah, it looks like it could be.
Yeah, it's got polish all over.
Stony coral.
Is it possible to zoom in any further, any closer?
What's that? Can we zoom in more?
Oh, yeah, that's madrasis.
madrasis.
See if I can get you
Can you make
a
a-
field.
Oh yeah, look at the polyps there.
Yeah.
Square 10 inches.
Nice, Steve. Thank you.
Dr. Steve Askovich, everyone.
Oh, and there's a little critter right here. This little guy.
What kind of, what is it?
See him moving
Oh,
around?
oh,
Little hermit crab.
Crab
Oh,
with a.
so tiny.
He's really tiny. Well, the polyps are all out.
Go pretty deep. That's 260.
Madrasis, myriaster. All right. Well, that looks like one that's worth taking a look at. We're at about 260 meters, yeah. Are we still, can we go back and look at that one?
Sure, one second.
Sorry. I didn't
Anyone really
tell you.
apologize?
I was not clear.
You like that close look at the one on the right there?
Yes.
I'll do that for you. Sorry about that.
Right, Steve, you think? I'm not Steve. Sam, take a reading on that one. The madrasis.
Sure. This would be our first square opinion.
Yeah.
You can do either one. You can do your camera to the inside.
All right, so we're just shy of 260 meters, and we're gonna.
Just hit out on the right
We have this madrasis that's in this field of sponges that we're going to get close to, and then do some measurements. We're getting situated to
For the madrasis?
for the madrasis to do some SOLARIS measurements.
Yes.
So it looks like there's some area with darker polyps and some with lighter polyps. I don't know if that's worth looking at the difference there. Do we know what the depth range is that madrasis lives?
Andrea assumes we'll see more of this as we get a little shallower.
Oh, okay. So this is a stony coral. It has a calcium carbonate skeleton, similar to what you see in shallow corals, stony corals.
The one on the right is good.
Yeah, lower right corner.
Ah, okay. Steve says at a site west of here called Pichinchi. Well, I just completely butchered that. Pichincho? Pichincho. They found a massive wall that had a number of different coral species, including madrasis.
Madrepora, madrasis, and Shermanitrix.
Yep.
I think I know what he's talking about.
Oh, we can see that current just whipping by. This might be a little tricky with the measurement. Are you guys getting a background now? Yep. Okay. Sounds like we're ready to move in.
Give us a signal, come on, the graces. MADRACIS. Where the instrument is left.
Will this be? It'll be full.
Okay, cool. How about we add a comment with the SH number if we decide to see.
That's something I'll do.
Then, because they got to write it.
right?
If it's good, if it hasn't, if it's making ROS.
Get more of a
you're right.
down. There you go.
Probably one.
You happy with that spot?
If we could try to get as close as you feel you can without.
Okay.
This is gonna be brittle like that style asphalt.
But a little thicker. Probably a little hardier.
Not really. You know it's thicker. It's because it's resistant.
So tricky with this current. The wand is just
A little bit of stress.
moving all over the place.
Just how close.
Let's see.
See if we can.
And this says depth distribution of
Closer
this
than that.
coral, she thinks is down to about 1,200 meters.
Oh, seems like we're drifting off here.
Are we hitting it? You might be, so we'll just like bring it up. I would y'all up. I'm responding.
Yesterday, I cut
Alright,
two
shoulder up.
contacts because it fell out twice and I just wore my glasses.
Alright, so I'm lower it.
Okay, what
Just
is
try to
this?
from you right.
Just races.
Is that the?
Genus. MED-RACIS. MED-RACIS. So trying to combine four for the closest
You're not too far here.
So the shallow water, yeah, like most corals make ROS in shallow water.
We're getting nothing.
Yes, I can see why the insurance is like trying to catch it, so that's close.
This is so perplexing.
Now with my contacts are just, my eyes are just rejecting contacts. That's too big. Yeah, at least I brought my glasses.
Nope, I think nothing. So, again, we're striking out on this dive, so nothing from this coral either. I have a feeling the current is kind of causing some problems here with trying to make these measurements, but we have seen in scleractinians or stony corals before that some of them constitutively make reactive oxygen species, so they're making it all the time, night or day, really high concentrations. And then for other corals, they don't. So yeah, I think we could move away. Thank you. Do you want to collect something, Sam? No?
No, we don't need it.
Okay, so no collection.
Need 12.
Yeah.
So I guess we only have two.
Yeah.
Well, no, we have three.
Oh, we could have last ones. I was thinking of traces. Yeah, we got plenty of tissue. Yeah, we can try some of that process.
All right. You just keep heading towards that waypoint. So we're just cruising again here. Moving a little bit shallower along this ridge and looking for some of the targets we wanted to hit on this dive. Finding a lot of sponges. That wasn't the target we were looking for, but
Is that a coral right there?
where? Oh yeah, looks like a big black coral there.
Spin around and put it down like that.
Get a closer look at that. Do the ROV files know what you're pointing at?
Yeah.
What?
Can you tell what that is, Sam?
Not yet. Uh-oh. Might be that one that that was, uh-oh.
It was an uh-oh.
This is one that, I think Jeremy wanted on audio. Can you zoom in, please?
Hey, give me 1 second.
Oh, that's a pretty one.
I think that's a Campopathes.
So a black coral, Acanthopathes maybe. There's a little hermit crab on there. In our last expedition with Solaris, and we were measuring deep sea corals in the Pacific, the one coral that did not give us a signal was a black coral. And similarly on this expedition, we haven't had any positive signals, I believe, with black corals.
Okay. I'm done working out.
All right.
Wait, 1 second.
What?
Oh.
No.
Okay.
I'm getting in trouble with sampling random things.
All right. We're good. I just wanted to get it. Nobody else did.
Ready?
Are we ready? Or do you want to take a screen grab?
I got it. Thank you.
Okay. Now I think we're ready.
Okay.
What are they saying? What are they doing?
All right. Well, we're going to keep our eyes out, keep looking for some more corals. We're kind of moving along this ridge. What is this little brown thing there?
It's good look.
Oh, anemones.
Oh, yeah. Oh, look at that. Yep.
Oh, I remember seeing those in videos, so it's here.
Nice. Yeah, looks like a little anemone. Oh, and something crawling on the ground here. Little shrimp. No, we're good, but thank you.
We're just getting excited by things that are different.
I know. It's like it's something that's not a sponge.
Can't do the forest for the sponges. Which country?
This is sponge country. Kid, don't you know this is sponge? Take your corals and go play elsewhere.
It's also kind of like a monocrop of sponges.
What is this, Sam or Nicole? Do you see this guy here?
Oh.
Yeah. Spotted by the geochemist.
Yes. I'm learning.
It looks half dead.
Oh yeah, it does.
More
All right, we can move on. I'm just proud of myself for spotting it.
Good job.
yeah.
Blends in with all the little orange specks. Or the orange sponges. Oh, there's another one at the very top.
Red
It's
guy.
small.
Let's try to find something big. I want a big coral.
We were a little spoiled yesterday.
Yeah.
Okay.
Oh wait, Mauricia, that's our
Yeah.
target. That yellow
Yeah.
one, I think.
That one right there?
I have no idea what you're looking at. Oh, right here? Is this what you're talking about?
Yeah, the yellow one.
Yeah.
It's hard to see it. It's like
All
just.
these sponges.
Yeah, it's getting full.
Yeah, I was 75, so
Yeah.
maybe I'll try.
Alright. Yeah, we get it. Yeah, keep it right there. This yellow one that's right in front of me.
Those ones are pretty good. Maybe my favorite's not the
Yeah.
Oh, look how much is getting blown over. We're not gonna get a signal off that.
Did like the purple of the rest,
I don't know.
What if the currents will be
I think we should.
a lot less at La Figuera?
They're gonna be, saying it's too, this one's gonna be too
Oh,
hard.
there's a. there's
I think
black
this is
coral
gonna be
above
too
it.
hard.
Okay.
I think we can
Also big,
keep.
yeah, it's also small.
It's small and yeah, and the currents. Just look at it. It's totally laying over the poor thing.
Another little one.
I think we should keep looking.
Wait, almost exactly like the one.
It does.
Yeah. I don't know. Yeah, I just got further in that wall. That's not flying away. So keeping it down.
I saw him in the main lab, like.
Black coral. Looks very center. Oh, there's a couple more. Oh, we've got some bottle brushes.
Yeah.
Oh, yeah, bottle brushes.
Stone patties.
Yeah, we can zoom in. It kind of looks like. What? You said stone
I
trash?
said nothing.
We can zoom. I think it looks like it, doesn't it?
Yeah.
It's
There?
very style with that piece.
Those guys there.
Yeah.
Yeah.
Alright, hold on. I'm getting.
They do look like bottle brushes.
Yeah,
Those are the ones, Santiago ones, right?
Like, I need to get.
no, I got 4 in the last dive, so we'll be good.
That was the three, he had the pictures of, wasn't that one?
Yeah, but he said he got enough in the last few minutes to dive. He got 4. It's fine.
This Wednesday night TV's really Thursday night TV.
Ooh, look at the red fish.
Thursday, I think.
the stylopath.
Yeah, it's a little more branchier.
Oh, that's really cool looking.
Can you see the worm in there?
Do you see a worm?
This is the one that there usually is a worm.
I see something crawling on the bottom.
Very branchy. I would say.
So we've done a look at
Move on.
Okay. Yeah. Not being pulled so hard.
Is that something we're interested in looking at?
No, they got 4 of these in the last dive, and one.
Yeah. Ideally. Are you getting to like
Oh, it looks like it's slightly different bottom brush.
Oh, Eleanor's watching her. Yeah. That's okay. Yeah, they're that way we can.
We don't need it.
So that's what
We don't want it.
Okay.
I think I got it. Measurement on any of them the last time.
You mean like they got no signal?
There you go, SOLARIS.
Yeah.
What's it
Superoxide.
behind it? Oh, that's hydride.
All right, I think we're gonna move on.
Okay.
Well, this definitely is starting to look a little different to me, or maybe that's wishful thinking. Doesn't it look a little different? Like,
Fewer big sponges.
yeah.
Yeah.
So we might be getting into some different terrain here. It's getting a little shallower.
This is wild. It's part of the. See how you can see? Oh.
Oh, what is this here? This guy
Maybe.
and that guy?
Oh, let's take a look at that.
Yeah. Oh yeah. There's
Oh, there's two.
There's 2 good sized ones.
My paper's running all over the room.
There was some overexcitement that we saw found something besides a sponge.
Oh.
That looks like it might be thermoristic.
Yeah.
If that's thermoristic,
This is definitely looking more like different diversity in the cover here. Oh, what's that on top of it here? The
I think
crinoid here on the end.
I think the top one up there. I think that's like a hydroid on the right.
Oh, on the right? Yeah. No, I was talking about on the left here.
Oh yeah, left crinoid.
Oh, and then that other. Yeah. Wow. You want a closer look to see if this is what we were hoping for? Wow, look at that thing.
That crinoid is so cool.
That is so neat.
The one on the right could be a sponge,
We
right?
saw that last night.
No, that's it's a hydroid.
This is interesting.
The hydroid on there?
oh,
Not
yeah.
a part of
Yeah.
this sponge.
That's a cool idea. Okay, this isn't exactly the
Yeah, this is
thermoristic we
different.
were looking for.
These are most droids you're looking
There's two colonies here.
This could be like.
Yes, we should.
I think we should look at it. Yeah.
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 So what do you think it is?
Oh, give me a second. You can pull out a name.
Thermoristic. Yellow.
We have to call it something.
Wow, you think I'm
That
gonna
to like
get a picture of it?
the pyramus guy.
I don't know if it's thermoristic or
Just these-
reflexor.
Oh.
What's got an apron coffee
This
on it?
kind
Um,
we're only researching.
Can you give me a second? Come up with a name.
All right, we'll give you a second.
Oh. Well, we can be wrong. Let's just call it.
Green coral.
Well, it's
All
definitely
right.
soft coral.
We had to, we got in trouble. I gotta come up with a specific name.
It's a soft coral.
Say,
No, it's not softcore.
It's an octocoral, right?
Yes, I think
So
so.
we've got.
some people will say soft coral, but they're not coral.
Oh, okay.
all this yellow, what's the word? No, it's not. I don't know.
It's like a thinner debris.
Yeah, but it's,
It's got that polyp density to it.
Dude, don't worry. I was like, it's moving. I can look over at it for you.
Oh, is the one? I was like, the one is moving.
I would say.
Are you, do you guys want to measure this? All right, so before you get too close, let's do a background.
yellow, hair, my wrist.
Oh, you want that? Okay.
Let us know, Lena, when you're ready to move in.
I was like looking at there, I was like doing math and all of a sudden it's like about a quarter mile.
How slowly you make It looks way close to
Yeah, is it pretty far off the wall?
Okay,
There.
Oh,
Yeah, for your phone or is
Nah, it's okay.
See how the branches are curved?
All right, so we're doing a SOLARIS measurement.
Place over here.
Closer, they're curved somewhat downward. They're more
So we're running the background right now, and then once we collect that and get a stable signal, then we'll move towards the coral, and who knows, maybe we'll get a signal.
At least that's what, like, in the picture we
Sam, what number are we giving this?
I'm not sure we're gonna sample it yet, so I'm not giving you an SH number just yet. Is that okay?
Okay.
Dependent on.
All right, we're ready to move in.
It's on our last. We decided to sample because we got two here.
Event 5. So let's try to get in there, maybe near the middle.
Oh, yeah, big money, big money, big money.
no wind.
Okay, we're still really.
Yeah, we're pretty far away. Is there any way we can get closer to the coral?
Hydraulic?
Yeah.
Yeah, try.
Current is going like the left and we got the backside of that.
Yeah, can we, which way, is it going this way?
The left, yeah.
Yeah, maybe if we don't see anything, maybe tuck around the backside. All right, yeah, let's see if we can tuck around, maybe like tuck around the backside of it. And really where we're pulling in the water is right at the tip, so. That little crinoid's not very happy right now.
Okay.
Such a small scenario.
That was something.
I think it's just maybe from the bump though.
Yeah,
Something
okay.
from the bump.
Oh, okay. Yeah, we could give that a try.
I would suggest we bump this a few times and see. Thanks, Sal.
Deeper.
Way ahead of us.
What was the baseline at, Lena? 12,000?
No.
14.
Oh, 14,000? Well, I got something when we bump it, but other than that, nothing.
Do I do the bumping?
I think it's okay.
Alright, stop the pumps.
All right, we're stopping the pumps.
All right, we're done.
Alright. So,
Sam, do we want to collect anything?
No, you might want to test that colony up there.
Okay.
that one right there.
All right.
That one?
Yeah.
Yeah, I'll do some public. Sorry about that. Happy to.
All right, we're gonna move to that larger colony up a little farther up.
Oh, yeah, no, I see a lot of bottle brushes there.
Oh, look at all the bottle brushes. Yeah,
Oh my gosh.
there's a bunch of them.
So this is gonna put us pretty much more face into the current, so we can put the probe on there. Should be.
What event was that last one, Lena?
It was about 5.
5. All right. Do you feel like Solaris is running well? Seems like okay.
Yeah.
Well, it sure is pretty. Do we know what this is yet?
We're just calling it yellow Marissa.
Andrea has also suggested that.
Okay.
Anything to drink?
Yes. Coffee would be great.
You need coffee?
Yeah. Like a
Put it
little,
in that.
no, just like a little espresso or something, or like a little bit of cream in it.
Mm-hmm. You don't want it in your mug?
Okay.
That way.
Sure.
Spill.
Yeah, thank you. Someone's going on a coffee run.
Oh, no, I should wait until this
Are we ready to start?
I think we're getting ready. Lena, could we do a background and we'll go and measure this one? All right. How's it looking? Stable?
Stable.
All right. So we have a nice background. We'll go towards the surface.
About 6.
And 6.
This is still camera suit.
All right. We're moving towards the surface. And we'll see what we see.
I think you can call this village origin. V-I-L-L-O origin. V-I-L-L-O origin.
Can you tell how close we are? Does it
Pretty
look?
close to the shadow.
Oh, yeah. Okay.
Probably a little bit closer with that picture. A whole lot.
I've just rolled it enough reason.
Okay. Well, it doesn't look like we have a signal again. So, doesn't seem like, at least it's consistent with the other one. Oh, you turned the pumps off? Okay.
Yeah, I just
All right. So I think we're good.
Okay.
Thank you.
Will be alright? I saw Twitch that it's sort of asking so fast. Alright.
All right. I think we maybe can. Was there anything else you guys saw here, or should we just move along? All right, we're up and moving again. See if we can find some other corals, some new species to look at. How is our reagent?
it's good. I've been timing it so for a map and really coming out of the water. I like
Oh, okay. And keeping track of how much we're. Yeah, okay. All right, let's see what we can find. Ah, thank you. Coffee has arrived. Ooh, and a Twix. It's the little things.
Do you guys know what ASMR is?
Yep.
You can do Twix.
Yeah.
Some sand there. Maybe we should try to find a barrel sponge. Scott would like that.
Yes, ma'am.
More sponge.
And, oh, Kate's back. Yep.
Oh, I've been here the whole time.
Oh, you were just quiet.
I'm just waiting for the sponges, man.
Well, there's lots of sponges, but not.
There's snail, I don't know, we might have to get one of these snails.
We're still a little deep.
We're still a little deep, yeah.
So the barrel's running, I think.
Oh, you're still.
Oh, there's a big snail, yeah. A green thing?
Oh, green sponge.
Oh, yeah.
Oh, yeah.
Oh, there's a black coral right there.
That's what I was actually looking for.
Oh.
I don't know where it is. I'm just kidding.
That's
That
another.
was like that kind of
It's green now.
Alright. What does that look like?
The hat over here.
Yeah.
A snail, yeah.
A very cool show.
There's another sponge, a spiny sponge.
Oh,
yeah.
yeah.
You gotta zoom in on that and see if there's an Oscar over there.
It would be really tiny. And an orange sponge there. It's just kind of crazy looking, right?
Oh, I think the colors were a little off a little bit too, so it might not be as green as it looks.
Yeah, these two TVs are off.
Is this that coral?
That's the one soft coral that we've gotten.
Yeah.
Looks like it.
Oh, yeah.
Little guy tucked in there.
This little preteen.
A preteen coral.
Yeah.
It's super moody.
Yeah.
Oh, there's a little fish went by.
Well, you didn't notice. I think it's like in the right most to the one of those.
There's a yellow. Is that a interesting thing here? A little yellow coral here.
Oh, it looks like a different kind of style last year.
Oh, okay.
The highlighter exploded right there.
Yeah.
Nice sound effects.
It's crazy how much stuff is on this rock. Just completely covered.
If you said that we were in an ocean on another planet, easily believe it, right? It just is, it's like it's kind of otherworldly.
Yeah, definitely. Some orange stuff coming up here. No, I assume a sponge.
Probably.
What is this here? Is that like another little anemone of some sort?
Oh,
You see this?
oh yeah, that's a little cup coral.
Oh, it is.
Right there.
This little tiny guy here.
Oh, yeah,
Solitary cup coral.
Huh.
Big giant polyps.
He's just like sticking out.
Yeah. Probably like pea size. Right?
You think that's the size of a pea? Yeah.
Maybe a little smaller.
Wow.
spire here.
Look at that. That's
Yeah.
neat. Huh. Little spires sticking out of the ground here.
Alright, sponge quest.
Yeah, Andrew's pointing out the lack of fish.
So where are we in relation to the last time where we saw a lot of many shapes of sponges?
We're to the southeast of it, so it's up.
That's the green line that we saw a bunch, or the yellow?
What?
Where we sampled the sponges yesterday?
Yesterday we were over in the red,
Uh,
up there in
oh, the
shallow.
top of the,
Shallower, yeah.
top of
we-
the ridge.
Yes. And we're sort of heading that way. I'm assuming moving into shallow water is part of the goal.
Oh, is that a cool clue?
Oh, what is this up here?
Just seeing.
Is this what you're talking about?
Yeah.
Can't tell if that's just rubble or.
Talking about how it's a pile of stuff.
Oh, yeah.
What in the world?
Glass sponge in the middle of it?
That's wacky. Huh?
Feels like you're this is making.
It does,
This
yeah.
sponge made a bird nest with stuff.
it's like
Yeah.
very- Maybe we should get a tighter zoom on that if we can.
More rocks in the woods, sorry.
Thanks.
The wall's breaking on me.
Oh, yesterday was gastropods showing.
Oh.
Oh,
Gastropod?
Interesting. Yesterday's pile, this is.
oh, look at this guy coming for us.
These are branches.
It looks like bits of this that last year, but why would it all be there? Is it getting stuck to the sponge?
Somebody's.
No, somebody's asking, what's the oxygen here?
It's still pretty high.
Where is our oxygen?
It's like 205 micromolar.
Oh.
205.9. So it's very aerobic.
Almost 19 degrees Celsius. 18.8.
It's cool looking. Yeah, it does look like a bunch of coral rubble, right?
Why is it aggregated all right around here?
Yeah, I don't know.
It's almost like somebody
Aliens.
built a nest.
It's always possible. So I don't see any fish around.
Yeah, crazy.
There's a cool rebel pile.
It accumulated right there.
It's like it was a colony that just got squashed or something.
Is that a sponge in the middle?
Yeah. Glass sponge in the middle.
It's coral top left. I think it's another candidate. Or can't get back here.
Where at?
Yeah.
Fatties?
Oh, I think this one they're talking about.
Oh, yeah.
Oh. A bite.
You interested in?
I don't think so. No, thank you.
Shall we carry on?
Yeah.
I think we're Sam, what one were we trying to get live animals for the experiment? Was it the Paramaricia?
Yeah, it's Paramaricia.
The yellow one?
Yeah, it's gonna be yellow. Thicker branches than the one we just saw.
Okay.
Kind of like those white ones we saw earlier, but yellow.
So we gotta try to find those.
Yeah. Or if we see some of those, that like lavender one that we first dove on,
Okay.
some of those too.
The rocks.
No little, is there a little fish? Oh, squid.
Looks like pretty big squid. Oh, that's pretty deep.
243 meters.
That's pretty deep. Oh. He swimming backwards and right into the rock.
Oh, there's a fish. Couple fish. Oh no, that's a squid and a fish.
It's the same squid, isn't it?
Yep. And then some other little guys swimming around in front here. 240-ish meters. Seems like we're starting to lose all those little sponges that we were seeing, right?
They're getting smaller.
Like we're losing that.
Aren't they just the white dots all over everything? They're just like tinier now. I think they're losing the
All that big platey
biomass.
kind of, yeah. So it definitely seems like it's changing.
We're at 243 meters.
Here's a big sponge. Yeah, over here.
Oh, down to the lower left.
Look at the one on the left there.
It's kind of buried in a rock, though.
Yeah, I think we can keep looking.
Big overhang set of the car.
This is getting more bare over here, yeah. That is a really neon yellow. It's bright.
Is that
Right.
on the left?
Oh.
Yep.
Or upper right, upper left?
Upper right?
Upper left.
Okay.
Really?
Gotcha.
I was looking between the screens. and-
Several of us
No,
got in
it's-
big trouble. 'Cause the crew
That
was-
white there, what is that?
I don't know.
They say
Superstitious,
about
huh?
whistling and
No, no.
the
But I'm sure that everyone's whistling, singing.
There's a quick look just to see if this, that purplish color. Wait, what is that?
What is that?
That's really cool.
Yeah.
weird sponge.
I think it's this one.
That is so strange looking.
Your odds are it's a
you guys, yeah, were zoomed in on something like this once when I was in the mess.
I can-
Oh, like a part of it was like
Not
that,
from this
yeah.
angle, I gotta come around the other side. There's a big scary thing right there.
There's a scary thing?
Yeah, I'll leave, yeah, that right there.
Rock.
Oh. This is crazy terrain. Huh?
That's pretty cool though. I like it.
It's gonna get so flat.
Yeah, it's like a paddle. Oh, the other side looks so different.
Oh, it does look really different. It's just that one side. Huh.
I think J-Rock white balance on something far away, so everything closer than that thing is distorted.
You re-white balanced on one of the freezer handles.
I can use that coral to the left above it. Let's see if we can wood balance on that.
Oh, yeah.
Yeah.
This is a crazy looking thing
Got
here, a
this
sponge that has very different look on the two sides.
What's the Solaris wand?
Yeah. Hold on, and it's pretty far away. It's got the port thing to block it, but I'm trying to come in and just see if we can get it away. Try it again. So if you move that arm over, might be able to get it closer.
Oh, yeah, it has the same kind of dots.
Yeah.
Are those dots the sponge itself, or are they like on growing in it?
I think they're fungi.
Really? We can take a piece of it.
Yeah.
Yep. Is the wand okay?
It's fine. We're just getting out of the way so I can get a little closer.
Ah. Nice close up of the wand there.
Alright. You're right there.
Could it be a coral instead of lobster?
The sponge?
Is it a sponge or a stylostrate coral?
I don't think.
No.
It's just,
And there's a stylistic right above it.
yeah.
Oh, so that looks like it's on top.
Are those Zoe?
Yeah, that looks like.
Oh yeah. There's something with little polyps.
Yeah, I think that's what
So
Zoe.
See something on it?
Yeah, there's something on it.
Yeah.
Oh,
Yeah.
Should we sample it?
yeah, it's like a little zone. Zoanthids on it. Yeah, there's some zoanthids that like grow on sponges. They call them parasites, but who knows what they're doing.
Well, I don't know. Is it a little bit difficult to work in this terrain?
Yeah, these little trucker guys are slowly. Ooh.
Stand by. That one, 4 more bounce. Alright, just see the white bounce that carries by you.
I'm almost done.
Oh, wow, look at that.
That's
That was
great
neat.
color, wow. Good. Wow.
Crazy looking.
Tiny, tiny, tiny zoanthids.
Huh. So what is the relationship of the zoanthid with the sponge?
Don't know.
Buddies.
No, I don't know. There's a few species of zoanthids that grow on sponges like this. Thought to be parasites, but I don't really know if they know that well.
Huh.
Grow in the sponge.
Crazy.
You want a piece of it or you want to?
I don't know.
Carry on, carry on.
Did you want a piece of it, Kate? I don't know.
Yeah, we can do fungal cultivation.
Okay.
Add it to the collection for the Smithsonian.
Would they want a piece of sponge?
Yeah, we could give it to Alan Collins who like it.
So do we have to worry about the zoanthids coming off or
No,
no?
just leave them on.
Okay. So are we thinking the whole sponge or
How big is it?
can we tell how big it is?
That's
Like 20.
10 centimeters right there between those 2 green dots. Shoot, I can't get on there very well right now. You can get a little.
Probably this big.
So there you go. That's 10 centimeters right
Ah,
there.
10 centimeters.
So
So it's about 10 centimeters
I
wide.
think it's probably a little wide because we're at an angle and then it's probably like that big.
Okay.
I don't think we can preserve that.
Oh, yeah.
We can dry it.
Yeah.
Drying's okay, the stick goes away. We used to have that.
We also don't have to, we can continue on the search for a big sponge.
It could be a piece, I guess. The whole sponge would be difficult because we have to throw it in the bio box. And if we come across those paramaricia, then we're down to only 3 more compartments to get those paramaricia in.
Yeah, let's just do a piece of it.
Grab a piece? Okay.
Piece and throw it in a quiver.
Piece for the quiver.
All right. So we're going to grab a piece of this sponge.
Well, I'm gonna have to do selection, we gotta watch out.
I'll wait till you figure that out.
Yeah.
Cross that bridge when you get
So
it.
we're at about
You put it into
240 meters right
So
now.
what would the SH number be for this? 1863?
16633.
633.
Yeah.
18633 is our sample number. And we're going to collect a piece and put it in the bio box. Not a bio box, in a quiver.
Quiver?
In a quiver.
Quiver what? Whatever's most reachable.
Whatever the ROV team decides is most reachable. Except for 17, we got one thing in 17, that's the only quiver.
The
with anything in it.
easiest place for us to put it right now is going to be one of those outboard ones on the arm, or on the rail, I mean.
Okay.
Because I don't want to have to move the wand, because it's
Yeah, it's pretty-
In a
safe
precarious.
right now.
Alright.
And I did move the porch out after that, so. Keep going around.
19, coral quiver 19. Are we sitting kind of precariously?
Nah. Not this much space on the rock.
Yeah. Is that all the fresh water coming out?
What's that?
All right, so let's hope that this sponge is gonna just give us a little chunk off the side there. Sometimes they're really spongy and hard to get a chunk off. So in the chat, someone asked if these zoanthids that we're seeing here are similar to what we maybe saw earlier. We had a dive yesterday where we also saw kind of these, I believe, this kind of same reddish.
They look very similar.
Looked very similar. It was a different sponge species. It was at a much shallower depth too, right? That we were at yesterday when we saw that. So we didn't sample that one, so we don't know. But. There's a student here in the control room, and she's interested in the organisms that live within sponges and the interactions between these microbes and fungi and how they're impacting the chemistry of the sponge and controlling the waters, the chemistry surrounding the sponges, particularly thinking about kind of interactions between those organisms. So it looks a little spongy. Kind of reminds me of a pita. Pita.
Totally. There you go.
We're on the same wavelength.
Oh, beautiful.
Good job.
Well
Very
done.
nice.
Nice.
Masterful.
Very nicely done.
Nicely done.
This might be too quick.
Oh, wow. It's a sponge.
Yeah,
I'm just gonna stuff it in there. So our student Kate, she's interested in kind of both sequencing the genomes of the organisms living in the sponge, so looking at the metagenomics, as well as trying to cultivate or kind of grow some of the organisms that are living in a relationship with the sponge, either within the tissue or on the outside of the tissues. So she's going to be taking a piece of the sponge and kind of trying to extract the the fungi and bacteria that are living within that sponge and grow it in the lab so that we can then study the interactions between those different organisms, those different microbes.
What's that?
Shove the jaw straight down there.
Yeah, maybe, or just shake that corner in there and just like we do it.
Yeah, I didn't see, to respond to the chat, I didn't see it turn purple this time. We can kind of, after we get this put away, we'll turn back and take a look at it and see. That took a little bit of time. Is it turning purple? No. So yesterday we took a piece of a sponge, the top of a barrel sponge. And when we did that, it released kind of this black, I don't even know how to explain it, particles or coloration to the water right around it. And then the whole ring of the top of the sponge turned purple. And this time it doesn't, I don't see any on this piece. Ooh, uh-oh, push. And then we don't see it on the piece next to it. So clearly a different reaction.
It's a Doritos kind of flash.
I can't help but think that looks like a piece of pita bread.
Oh, it's a yeah.
I bet it doesn't taste as good.
Hopefully we'll find out.
I'm not gonna try to find out.
Science.
Yeah, in the name of science.
There are questions people have.
All right, this could be a double dog dare. So yesterday when that barrel sponge turned purple after we took a chunk out of the top of it, we went back and we took SOLARIS, our instrument, and measured where we had cut the sponge. And we had a really high signal of this reactive oxygen species, superoxide, when we did that. Which makes a lot of sense. One of the reasons why organisms, including us, produce reactive oxygen species is to repair wounds. So that's called an oxidative burst process. Oh, well, that's okay. She's gonna chop it up to extract the microbes anyway. So,
There you go, that should fall in.
oh, look at some, oh, yay!
Maybe we can sweep the other piece in.
Yeah.
Purple stuff.
No.
Does not seem to be.
Just they put clumsy and stuff.
It's almost like the current's almost pushing it in. Give it a little nudgy nudge.
Put garlic on it.
Yeah, there's a little chat.
Excellent, thank you.
Someone's asking if eating one of our subjects is a time-honored tradition in biology, and I'm a vegetarian, so I'm gonna have to sit this one out.
That tradition has been fading with the new generation. I know somebody who has eaten a piece of Riftia
Oh yes,
tube worm.
Yep, I know several.
yes, we have a colleague who has eaten a piece of a tube worm. That just sounds terrible to me.
Like sulfur bomb.
Experience.
Excellent taste is. You know California,
I don't even want to think about
it's kind of like
it.
a snap.
It's good shapes.
It's like kind of sweet.
Yes, they did live, Steve, and I think that this person's actually done it more than once.
Really?
It's a lot of nasty.
Is that right?
Gurgis?
Yeah, little chunks of.
Peter Gurgis?
I love that
Same
man.
person.
Wait, what is
That man's
Gurgis
a legend
and
in my mind.
Spencer and I want to say Robert probably also.
Wait, Gurgis tried it too?
We're calling them all out.
Childress and Chuck.
I think Gurgis is coming out later this year.
I don't know when he is.
Oh, I think he is.
I
Yeah.
tried,
Yeah.
but I think Loyola is still.
I do not want to talk about my tube worm story. It's still too soon.
Uh-oh, we have a story coming up.
Years ago, but it's still too soon.
That bioaccumulator is.
Yeah.
How did you try it?
We brought a Geiger counter on our last.
Anything
Yeah, yeah.
about
We went to the lab base.
Get it away.
Are you on a beat?
Yeah, people say, well,
So I can see some of the chat. It kind of scrolls up, so I can't see all of it. Someone asked when we're going to be doing this again. Well, we're going to be
Part of that.
Oh, really?
doing dives for the next couple weeks all around the waters of Puerto Rico. We're studying mesophotic to deep sea corals. So we've been at this site for a couple days now. We had originally planned to only be here for one day, but there's just such a rich diversity and a lot of different kind of ecosystems to explore here. So we haven't been able to tear ourselves away, but pretty soon we're going to be heading to the southwest region of Puerto Rico, and we're going to be looking into some more shallow coral reefs, specifically getting up into some of the mesophotic reefs. So it'll look very different. We're going to see kind of more scleractinians, more stony corals. We'll see a coral called agricia, which forms this kind of large plates in the mesophotic region. So eventually, once we can tear ourselves away from this ridge, we will start heading towards that site. And we do 24-hour operations. Of course, we have to bring the ROV up and collect our samples. We also have to replace the reagents within our instrument. And like today, we were down for about 5 hours doing some maintenance and fixing some issues we were having with the sensor. But we try to turn the ROV around as quick as we can so that we can get back in the water and do more exploration.
Stuck. There's a little fish. And he just ate it.
So we'd be happy and love for you all to keep tuning in. We try to answer comments on the chat, but as you can see, we're not good at keeping up with it. But we're trying.
Keeping our eyes peeled.
There's a lot going on in the control room.
Colleen, who gets
Yes.
to name corals that you discovered?
I'm sorry?
The ROV pilot.
Oh, who gets to name the corals?
Yeah.
The pilot, the ROV pilot.
Gets to name the coral?
Oh, that would be wonderful. I don't know if we've discovered a new coral.
Jason Isis.
Oh, okay.
Bamboo coral.
Well, is that after one of the
That's
Jason
the-
leaders?
But if we discover a new coral,
That's the ROV thing.
we'll have to like draw straws, see who gets to name it.
That was a really nice collection.
Oh, like not. I didn't buy it. I thought you meant like a tail of a rat. No, rat tail.
So we should say we would catch on traps. You know, so we just put out rat traps and then you
What's that?
I was.
Just a movie in the
That
chat.
makes a little more sense. I was like, watery.
Oh yeah. Yeah, someone's asking about shifts. The shifts are different for the different science and crew. Some do 12-hour shifts, some 6 hours, and kind of rotate in and out. Some people are on call.
We're at
Kind of depends on what you're doing.
floofy things on
above
the
us. Yeah.
four things.
In the control room here, we're doing 12-hour shifts,
There's no canthropathy.
but we rotate in.
Yeah.
Let each other stretch your legs. Go get some food. Get some coffee.
Yeah, they are. Don't even look close at them.
But even when you're off shift, you kind of want to keep watching the monitors. It's just too exciting.
Yeah, I can come over to 19 or just keep going up whatever.
I think we should start shifting northwest,
Alright.
don't you think?
If you want. Wanna
Start
do the
coming
big
up.
purples?
So the point of coming up,
Sort of coming back up.
so you're wanting to go up on top of the ridge. I just know like we are trying to stay on top of this ridge to stay at this depth. But
Oh,
are
you
you
wanna stay
wanting?
down here? Okay. It's,
Well, we are trying to collect these corals that we haven't found yet, but
So we come up 50 or so meters since I sat down. You want to go back down to that depth?
no, I think we're good about here. Yeah.
Okay. Which direction do you want to go so I can move best?
I mean, we're almost to the waypoint, right? Yeah, I guess why don't we try to get to the waypoint, see where we're at, and then we could start heading up.
see
Up
some
the ridge.
CCA.
Are we?
Yep.
Yep.
Starting to see, yeah, some crustose coralline algae there, the pink.
30 meters, that's like.
I will then wait.
It's probably a sponge.
You think it's a sponge?
I mean, if that's red algae, we're at like the limit of anything for us. Whoa, it does look like
It looks like it.
Doesn't look like
And it would be, that rock is jutting out, you know, like it's.
Catching sun. What do you want to look
like a
like?
lens.
Oh, if we could like zoom in maybe on this pink here, like we're
It's
just
crazy.
debating whether or not that's a coralline algae, which would imply that we have some light.
It really does look like it. We're pushing like
Yeah.
the deepest.
But it's not even on the top of the rock, it's on the side too,
Yeah, but still kind of.
which would get less light.
We also did we change our white adjustment? Like I wonder if
Yeah, we did when we were at the sponge.
maybe it looks redder than it is.
268 meters is close to the max. Deepest. It is a red algae. The deepest
And
one
we're at
is embedded.
230.
Yeah.
Yeah. What is that blue piece?
Oh yeah, that's really neat.
That does look like algae.
That totally does.
That's it.
Wow. So we're
Wow. Proud of you.
even though no one believed you in the
Always
beginning.
looking for that CCA.
I thought it was right away. Alright, we're happy.
Yeah.
Did you get a frame grab? Let's just.
Oh.
Could we do that really quick, just
Too mesmerized.
to have it on there?
Well, there's a few little cup curls on the very right edge.
Oh,
of it.
wow. Those are like single polyps.
Yeah.
Yeah, those are wow, they must be so zoomed in.
It's like a turquoise sponge.
Oh, look at those.
Man, those have a lot of tentacles.
Oh, look at that.
Those are the septa, the things that pulled in. It's like a little.
Oh, there's a dry zone here.
Are those cut corals on the top, right?
Lacy.
Yeah. Is that? Oop! I went the wrong way. Sorry, I went the wrong way.
Oh, it's funny because I was pulling up the 6 and I went down and I was like,
We were like fighting each other.
I broke the camera.
It's a thin layer of tissue over that. And like the tentacles are retracted in.
I always go the wrong way.
Those ones are kind of cute.
That is really cool.
Oh!
Wait, what was the ooh?
The hermit crab almost tried to eat
Yeah,
maybe
it tried to catch a little,
like a squid or an ostrich.
it's not the little fishies, I think.
Oh, is that up above these little?
Watch, it's hunting. Quick, go back on it.
I don't know where it is. Where?
You're right there.
To the left of the
Colleen, it's right
sponge.
here. Oh, oh. They're like taking
Oh,
those
what
little
is
hunting.
that? Oh, I think it's just an amphipod you get.
This thingy?
Down,
Oh,
down. You
I
got
see it. Oh, I see it. There's just too much to see. Okay.
I don't know what it is.
Oh, oh.
Oh, is it eating something?
Yeah, it's trying to eat the
Some
amphipod.
predator prey action,
Oh.
for sure.
What?
Is that a fish came by?
No, I think it's an amphipod.
The big dark thing, the black thing that came in and out.
Oh, that's a little.
Is it eating one? Did it catch one? No.
Okay.
Alright.
We're getting a little
distracted.
distracted.
Back to the mission.
Yeah.
That's all right.
That's okay. We can appreciate the thumbs up.
That, those 4 corals right there.
Uh-huh.
There are actually 5 there. Would make it really easy to get 4
Oh, look at the
replicates.
fish.
Oh!
Hold on for a second.
Oh, that must have been. That's like an adult of what Andrew was telling us.
Is that a little puffer guy?
Yes.
Do you want this?
Oh, beautiful.
Sorry, I was distracted by the fish.
Those black corals that I told you not to look
Oh.
at.
Yeah.
I'm just asking Colleen if she thinks maybe we
Cute
should hit him.
Because are they on the list?
There's a big
They're not on the list of the things he's targeting, but it is an easy one that we can get 4 of right now, right there.
Okay, that's fine with
And
me.
it would add a coral, like it would knock off a whole coral. We would have the full 4 or 5 reps.
Okay. Yeah, that sounds fine.
Oh, look at on the right in that hole, there's another crabby thing.
So yeah, so those 4 or 5 colonies there, we like to measure them and take
Just
pieces
like
for
right up
quiver.
here.
Okay.
Is that fish stuck?
So there's the one on the left. Do you care about that one? Or
Gaster
just the one on the right?
puffer.
Uh,
See, look
Well, if the one on the left is the same one, then yeah, we can do that. But
Alright, so this one and this.
Yeah.
Alright, I'll set up
Covered
that
in algae.
in that area then.
yeah.
Okay.
What is it
Just
doing?
not sure if
Is
that's a
it?
coral.
Did
or
that
the-
coral
just
just
a
catch
weird
it?
part of that rock.
Let's see what
Oh, there's something back there
Oh,
too.
look at the coral. It got something in its polyp.
I mean, it's shrimp. Oh, never mind. Oh, there's a spider crab
It
behind
escaped.
it. Oh, we could just.
Oh, the
sit
spider
there
crab is
all-
creepy.
Yeah, it looks like a coral.
Action.
Looks like a black coral.
Alright.
Same kind. Oh, yellow decapod, brachyura, crab.
Yeah, there's a lot going on right here. Huh.
Well, you started there asking for the cup corals?
I'm asking, Adrian asked if we could slurp those. I'm asking, what are those she's referring to?
Oh, what are we supposed to slurp?
The cup
She'll probably hear me say it. Yep, cup corals.
Those
Asking-
teensy ones?
Yeah,
And
before I
Adrian's
slurp them.
asking.
Can those get slurped? These little
Yeah,
guys?
maybe. I don't know. Let's see where they are.
Yeah, this one you guys had, knock the cup loose with the edge of the.
Oh, that is a good
It's
point.
true, they're
What's that?
really-
When we pull the suction sample out, it's gonna pull the tube off of the plug and that circle it out. Maybe a little bit. Oh, yeah, okay.
Oh,
The dye tube?
I think it's okay. It might push out a little bit of dye when we pull the suction thing out.
Oh, it might let out a lot of dye, yeah.
Oh, is it really? It's stuck in there?
It's possible it could let out a lot of dye, and it's possible that the sucking could also pull a lot of the dye.
Wait, can you try to
Wait,
get
so we
sargassum out?
can't use the slurp?
Oh, we can.
Are they like intertwined?
Oh
Yeah,
my.
we put the
30
the dye tube on the slurper hand. So when we pull the slurp hand out,
Sample
And we can-
sargassum earlier this time though?
We
You had
didn't
that
pull the
issue?
slurp out, we brought the sargassum to a slurp.
Oh, just turn it on?
Yeah.
It was near it?
yeah.
Oh,
This was ties. It was about that big.
Oh, there's a bunch of them. Look, there's some these here, and then they're down here.
yeah.
I just wouldn't want to create a dye mess when we're not near a sponge we're excited about if it is messy.
Well, that was like two, oh wait, no, almost like two different types or two different color morphs.
Yeah, it looks like that, huh?
If you guys want to do it, it's up to you.
So there's,
I guess, didn't we
Well, if you wanna
slurp the
see your-
sargassum?
Your fluorescent thing.
We didn't move the arm, just like if we brought the sargassum to the arm, and then we turned it on.
Okay, we're gonna have to figure out a different approach then, because we
Yeah,
can't
maybe
not
we'll-
use the slurp.
Yeah, we'll put it on a T-handle next time.
Hear that count? It's
too small to pluck.
Okay.
This is
We've
really
done
hard
it
to do.
before.
I think we,
Okay.
But it's not easy.
Well, I guess we pass on it. Well,
We could try it with a slurper. I just think we would really put dye everywhere, including in the sample.
okay.
So,
All right,
So,
let's just,
so we're talking about slurping something as like dye everywhere.
yeah, we have to change that approach. We have to be able to use the slurp.
You have to put the hose on.
Why can't we use this slurp?
You'll put the dye slurper.
Apparently,
Yeah.
it's tied onto the slurp.
Because we tied it on, put it on.
So the pressure won't, there's no pressure on the
Yeah, you
bag
think-
at all. So you think
right.
that the external pressure keeping it there is the same as trying to push it out.
I'm not sure.
We don't really
Yeah, I'm
know.
not sure that it's gonna just start coming out everywhere.
We just
Because on
don't
deck,
know.
like, it was pretty balanced, like, if you lifted the hose above the bag, like, it was level with probably.
Right, I think it has to do if we can keep it, what if we sampled and the hose was high?
Yeah. So
Then that would work.
so we don't know. what
But we don't know if it would.
How it's gonna,
Why high?
so it could
Because
be
of
nothing,
gravity.
it could pull our seawater back in, or it
This
doesn't.
floor seems less dense than seawater, right?
It's diluted in seawater.
Oh, so it's the same density.
Yeah, but it's a spring loaded.
But the spring shouldn't be under it. It
Yeah.
shouldn't be.
But the springs are pushing.
It should be neutral.
The springs
Could
are
we
pushing.
just sample it in such a way where the scooper is higher than the bag? Or is that,
to sample these, you gotta scrape them with the edge of the
Yeah, you know, just brush them
slurp
off.
tube.
straight
Suck them off as you're
down?
doing it. hard to slope up because you gotta knock them loose with the edges, so, like little pebbles.
I think the fluorescence is just gonna dribble out.
So do you want to try it or no? I don't have a, I have no idea.
Yeah, why don't we just try it and we'll know? Is there,
Well, then we could lose all our dye and not have found a full spot.
but the bag is not under pressure, right?
All right, let's just give it a try.
We're not
It's
squeezing
okay.
it, right?
It's under a little bit of pressure.
How? Why? If it was, then it would be
Basically, if you hold it lower, then it starts leaking.
in air.
Yeah.
You're underwater.
Yeah, yeah,
There's
yeah,
no
yeah.
there's no difference in
Yeah.
Then gravity's working here, but here we're
Well,
all.
maybe we'll suck some of the dye out, but yeah, let's put it on a T-handle next time.
Alright.
Before we do this, I'm just gonna,
Hopefully we don't like cover her sample in dye, but I guess we can.
alright, so that's right there.
What are we sloping?
There's some small cup corals here. There's like
Oh, yeah.
a series here, and there's this little.
There's lots of
She
them.
wanted the 4 on the upper.
That's 4 right
Yeah.
there.
Right in there, yeah.
So that will be somewhat, it'll
They're
be.
pretty hard,
it
right?
won't, it won't be going down, it'll be like this, so that helps.
Xandria saying
She said it's a bummer, so I can,
What's a bummer?
that
about that, so I don't, do
I mean, the dye's not gonna like paint them or something, right?
Well, it could, because they're organic. I don't know, it's supposed to be inert, but.
sponges are organic and they don't paint the
Yeah.
sponges.
Yeah. I think we should try.
All right, I'm asking Andrea which one set she wants. Which sets?
Knock and slurp.
What did Andrea ask for?
The bio group.
There they are. I usually grab this handle, but some more dexterity.
Jeremy's.
So she says any of them.
Usually come to the right and grab it.
But also, she wanted to
Oh, I
fit.
see what you're saying. That's a little there.
She says
any of them with
the striped calyxes.
Oh.
So is that
Oh, are those
these
the
or
ones
those?
Like the ones in the up
Yeah.
higher, the dark ones.
Okay,
I think it's like,
let's go for it. Let's just give it a try.
it's like the picture in the guide with the cup roll that's got the dark red tissue. I think it's like the ones we're after, it's just so retracted that it's, you can see the septa as well.
I can push up.
Squid. Alright, we're gonna pull it out and see what happens.
What's that? Yep.
That's because this slurper is gonna
I'm
pull
gonna,
it down.
I won't go.
Only if it creates a pressure differential, might.
Alright, what's that? I can,
Where's the?
In the biochar.
yep, you're good right there. Perfect.
Oh yeah.
Alright, so we are on jar 1. Do we need to move it or is jar 1 okay? Yeah, there's that coming out.
Yeah, it's just.
Oh, there's that.
Alright.
Remember when
Science.
Jeremy said?
Well, at least it's a slow flow.
You couldn't say a coral species name, otherwise it's like dead, and you can't use it. You see there, Andrea's text versus it.
Then we're back out. See, we're going right there.
So she didn't actually say the full name, she said epithet.
Alright, so,
This is our current indicator.
yeah. See it? See where we're going?
Well, we could take a moment to talk about
The top ones or
fluorescent
the
dye.
small ones?
The top ones,
Alright.
yeah.
So we're gonna go for
Those six are.
these ones right.
Or going to sample sponges that have that look like a barrel or a tube, and the way that they acquire nutrients is they pull in water from the outside at the base, and then it goes through the tissue and then out the top. And so, but these sponges only sometimes are pumping and other times are not.
It's a small, yeah. Alright, so you're gonna have to just
So
scrape them and
Andrea was saying, and a little knock and slurp. So, yeah, so sometimes the sponges are pumping and sometimes they're not. So we wanted to be able to know when they're pumping and when they're not pumping. So we have this dye that we can release and it'll show you that it's pumping because it'll pull it in and then out the top of the sponge. So hopefully we'll get to find a barrel sponge during this dive, and you'll get to see that in action. It's really neat to see.
Is this,
In the meantime, we're just having a bunch of dye pour out for no apparent reason.
slurp canisters that go in it too?
number one.
So next time we'll not have the tube stuck to the slurp so that we can not have to deal with this.
Okay.
I asked the question to change it, so.
Okay. We never did see the sargassum wind up in the slurp, did we? Which means these cup corals are going to get caught
In the sargassum.
in the sargassum.
You're gonna get tangled in sargassum fluorescein.
I mean, that's all right. It doesn't matter, right? In terms of the corals and the sargassum.
If they're all
I
together.
haven't seen
Yeah.
anything go through it all, to be honest with you.
But it would look a little embarrassing on the live stream.
We'll just preserve it for genetics. No stable acid setup, so. There's something not like fine. Probably just hose.
Yeah, probably still gonna close it. The fallout tonight.
Are you guys able to recover that from within the hose?
Yeah, we can get them on the hose definitely.
You can get it out?
Yeah.
Okay. Sneak it out.
How
Do I get some more?
insulatory is the hose?
Well, it's slowing down.
Good.
So was that
Yeah.
not flowing before when it was gonna
We found it in a stall.
It was being.
Okay, so once we pull it, it's
Yeah,
just
then
like.
we pulled.
That's actually.
Are you doing some more?
Find a sponge.
Yeah, Sponge Quest is on, guys. Let's go.
Alright, do you wanna look at those? Alright, so we're done with this?
Did we get we got those corals in there?
I can't say if they've done public review that big.
I think you did.
Yeah, they're trying
I
to
think
get
you
it.
did.
They definitely went in the hose, what they do, maybe they're all the way to the.
Fall out.
I wouldn't think that they would, no.
So I don't know, you probably don't have a lot of options for stowing that slurp, but if you could kind of pinch the tubing, you know what I'm saying? Like if you pinch it off, you can stop the fluorescein flow.
We can set it back in there and like try to grab it with the jaw.
Yeah, we can try to get it just in the soft part of the job.
Yeah. So we can set it right back there?
Yeah.
Alright. We can do that.
Did you do
Yeah,
any
it's hard to
screen grab or
predict.
anything on that collection?
It
Yeah,
did.
okay, we
You
didn't
should add
number
that
it,
it's
did
hard to
we?
predict how tension will affect things. 18634.
18634, okay.
The issue
The tension of the.
of the squish
And
mechanism.
put it in slurp too.
We had it snug.
Okay.
That's the of the bungee.
We had it sort of snug.
Yeah.
But
That was providing enough pushing on it apparently, enough.
Squared up the way it's how it's out.
I wonder if there, yeah, so there's enough, yeah, because the springs on it push it back up, right?
Right, they do.
Yeah. But the bungee, you're holding it down.
Right. So it was really neutral when we sent it down, but again, it's just really hard to predict how much, because bungees stretch a lot in deep water.
there's all these other factors. So it's the same
We'll do
density
those
as what we have,
for
but
the
gravity
top.
is still active.
It's
So
probably,
as we,
probably contract and pull the water, right? You can't do it.
they actually become
It's
stretchier.
single side, it'll fall into the thing though. Bring it back towards
Like
it's
a
just
warm,
touch.
warm.
Yeah, I know.
Just a little bit.
Yeah, rubber.
But,
Really?
so gravity is still acting on it, so if we were holding it,
Should be alright.
basically ideally we have a way to store the
So
tea.
it's not gravity or.
Well, because it's the same density, if we have a way to store the tea that's up here on the robot, then it won't leak at all.
No, so everything is the same, under the same pressure rating, right? But the
Right.
bag is being pulled on by the sponge.
Yeah, but
That's why there's no, like you, we took, you know,
there's a little bit of gravity though.
Nah, it's fine.
Not on,
not at this scale.
No.
Geostrophic flow, like
Yeah.
currents in the ocean,
Okay.
are affected by gravity, but, if this were denser, but I'm pretty sure this, you said you made it with seawater, so
Yeah.
if it was potentially less salty than other, than the surrounding seawater, then I could
I
see
can bring it in, so I
it
got
wanting to rise out.
Right.
It's
That's unfortunate.
here's what's about density.
Yeah.
On the
I mean,
deck,
you can see
definitely
it
gravity is
right
coming
there.
right
Yeah,
out
that's
through
right.
there.
Yeah.
I forgot that the bungee is under some amount of tension.
No, but even when they were holding a little higher up, that was stopping.
Very good. Very
It
good
went
on
back
the-
down, it's slowing more. Lower than that.
So maybe the seawater, you made it with the slightly denser than this down here. You made it with surface seawater from the ground.
Yeah, just from the uncontaminated.
I mean, it's
top.
still pouring out the same it was when it came out. I mean, we have, it's just,
Try closing it a little bit. Good job.
Uh,
There you go.
yeah.
There you go. There you go. Nice and easy. There you, it's slower. All right. Do you want to look at those? One problem is I got a big arm in my way.
Yeah, this is the fluorescent dye.
I don't think it really slowed down much.
Yeah, it's. I mean, it's slower.
I don't know. Is it?
trying to pinch it.
It's more diffused.
All right, you want to go check those out?
Well, I don't know if we're gonna sample and if this is spewing out like this.
Can't keep the arm there.
So how can we
Gotta go for the back.
easily turn it off and on?
What do I?
I mean, ideally, we have a pump.
Where's this coming out?
Take one of the SOLARIS
So
pumps.
we added that mechanism.
That
instead
means
of
creating a
closing
machine
them
that's
on the
electronic and
arm.
controlling the whole mechanical. I think
Go
the
on
mechanical
the,
mechanism that
second
we got is
sample.
great. It just
And
needs
we
a way of
use the second
shutting it
sample,
like
so
a
close
pinch
off this
valve.
little plug. Now it's
What if somehow when the rain, when the
yeah, it's connected to the valve pack.
Is it leaving it on?
It was on, I sat down, so I didn't mess with it. I kind of needed.
Did you give the SOLARIS an SH number? You did.
the SOLARIS.
All right.
I mean, the
Oh.
sargassum, I'm sorry.
Oh, yes,
Sargassum.
it was SARC 3.
Right now we're coming over to here.
Oh, but not an SH
And then I
number.
think we're gonna go this direction.
No, I think
Okay.
we didn't ever give them SHs, did we?
Were you not collecting?
I gave an SH number to the small asteroid that we didn't measure but fell in the basket.
Oh, okay. All right, maybe that's why I'm off.
There's 4 style asteroids that have SH numbers.
Okay.
Good idea. Put it on
Yeah, so if it has its own independent T, and
So it's on independent team, we can, so this is the thing, we have to do all of it, I guess, in one place. Anyway.
well, no, what if, I have some ideas.
All right.
Let me try to link it back up. Like, you could have a little quiver with the wand and it sits in,
If the
and the heavy T handle kind of holds it against something at the bottom.
If the tube is pretty short, that's where we could find, and take it in out of it.
Yeah.
It'd be really visible. You wanna do that?
Yeah, yeah.
Gotta think about it, man.
All right, draw some stuff up,
I'm devastated, it's fine.
I got my bowl of grapes. I'm gonna leave it for you guys to get. I don't know if Ben's not gonna eat grapes.
Those your bowl of grapes. Okay.
You want to take over for a little while? Okay. All right. This is where I left off.
We're just hanging out in this train.
All right. So let's see. We're reassessing here, and if you want to start going up a little bit, I think that's fine.
All right.
Going up in the train. I mean, we were trying to find some animals for aquaria. Like, we've got to keep our eyes out for the yellow, right?
Yellow
Paramecia.
paramecia.
Yellow.
Okay.
So let's not go all the way up on the ridge, you know, but
Yep, thank you. Sorry, no worries.
So yeah, we've been working our way southwest. We're gonna kind of start working our way northwest here. This will be a steep face right here. I'm just hoping that we'll start to see something on the way up. I mean, the Paramecia is usually a little deeper.
Well, I'm not sure where they saw theirs.
They saw them around 330 or something.
Which is the other direction.
The wall over here is really steep, already at 220.
I mean, so we could turn
yeah,
around
someone's
and go
asking in
deeper.
the chat if we need to get permits to sample corals. And yes, we do, and it's a long process. We have to apply to be able to collect both corals and sponges. And there's a limit to how many you can collect, and especially in what regions. So in marine protected areas, for instance, there's kind of stricter guidelines on how many samples you can collect. But even outside of the marine protected areas, you know, we have to submit a permit and say why we need these samples and how many, what species, and where we're going to collect them.
Something like that.
And then, you know, we submit after we're done with the cruise, we submit a report to say how many we collected.
Okay.
And then we tell them what we're going to do with those corals, and then ultimately, and sponges, and then ultimately, you know, what we share that data with the agencies. So it's a long process. We start the permitting process about 6 or so months ahead of time. And you don't always get approved. So, you know, if you don't have a justification, or if there are, you know, species that are endangered, then you cannot get approved to do collections.
Yeah, the Paramecia was 350 meters.
Oh, it was? It was that deep?
Yeah.
I mean, we only saw that one, right?
Yeah, we saw that light purple one at the very beginning. How deep were we at the very beginning?
About 370 or so, wasn't it? Oh, wait, no, it wasn't. It was like 320.
In the very beginning of this time? Sorry.
Yeah. All right. I think I'm interested.
Chief Scientist Colleen Hansel is going to take a break from the microphone for a little while. She's turned it over to your new host, Scott. Hi, Sophie. I'd like to keep things light and entertaining here on Radio Schmidt Ocean. So, Colleen, just to let you know, the plan was we can't move straight north because the ship's in the way.
Okay.
So we're going to move south a little deeper, which will give us access to a little bit of deeper water. And as we do that, we can move Falcor around to our southeast, and then we can start pivoting back up the slope.
Okay.
So we'll going to start moving. Well, we're going north right now.
I'm heading down. I'm gonna head south. I'm just crabbing down so you can see something.
I see.
we're gonna, I'm not sure where the,
So you're
purple permanent
facing north, moving south.
Big, there's like a, the pale permanent here,
Yeah.
and there's like a bright permanent. You over to see it, so I don't
Moving away from the-
Yeah, yeah.
We just swim straight south.
But then we won't see anything.
Won't see anything.
Right.
Right? And black corals and.
Yeah.
There's a big stick.
We start moving.
Looks
away
like,
south.
is it just a big stick? Oh, there's a sponge with with an
bag
orifice.
here?
It looks like a coral. You look at that stick.
You want to look at this stick?
Where's the stick?
This big. Yeah.
What's up? And a Coke can.
It's a tree, right?
Yeah, it's a tree.
Coca-Cola can?
Oh, and a Coke can, yeah.
Perfectly displayed. Yeah.
Okay.
Yeah, I just need to take a walk.
All right. I'll continue backing up downhill here.
Okay, you want to start moving the vessel? Southeast. Forward. Now we're gonna head down, and then we're gonna move the ship around 180, and then we'll head back north. Just to my right. Or no, I'm sorry. Just on me. Just put it on me, and then I'm gonna keep moving to my right. Okay, how quick do you want to go?
We're at 250. So I think over here we were at 300 where we started.
Is there anything interesting over there?
Yeah, no, we don't need to return over there. I'm just thinking, is
You want to start turning this way, turning the boat around?
are you gonna bring the boat around this side?
Yeah.
Okay. So yeah, yeah. So if we, is that a little sponge?
Should we put the bag here since
Yeah, I was wondering, is this
Oh,
a,
look at
this one? Right here.
On the left edge of the screen.
this? What about this one?
Ooh, little belly which one by?
Can I zoom? Or okay. Or never
Is
mind.
that one of those paddly ones?
Yeah, no, you're right. It's not. It doesn't have. Where did you see the?
Up
There's
here,
these little
Scott.
small glass.
Up there's one with a big osculum. It's like kind of bowl shaped.
Where is it?
Yeah, to the left.
Left and high.
It's a little bit up on that. There were two of them. Yeah, look at that one.
Oh, that's a good one, Sam.
Glass.
A little small though. But
Yeah.
I think there's another one on the other side of the ridge.
Oh, up there.
Cup shape.
Oh, I remember we
You have
didn't.
options.
Like this one.
Yeah, that's what I saw.
All right. Either one of these could be an interesting opportunity for us to try to set up and release a little dye and see if we see it pumping.
Yeah, whichever one is easier for you guys to
Yeah.
get to. And then ideally we hold the wand there for just a moment, like around the base of the sponge.
Right.
Because if we release dye everywhere, then we won't be able to see chemical reactions. That would be, the dream is like a little bit of dye that scoops it up and creates a.
Big fat anemone here.
Really?
Has it stopped leaking?
It kind of looks like it.
Wonder if the tension is just tension. That static tension initially is maybe released a little
It'd
bit.
be interesting to see what the volume is right now. So that's how much.
I don't think
we.
okay.
You know, like if we had a camera on it right now, it's like 1 liter-ish.
I don't think we squirted very much out.
I don't think we did either. Oh no, trust me.
We'll definitely see what it looks like when we. And you guys have control of that ram, right, for the dye bag? Do you know what I'm talking about? There's a hydraulic ram that was installed.
Yes.
Yeah, I don't remember which number it was put on, but honestly, the dye might be leaking out a little bit already. Like maybe we just lift it up.
Yeah, we don't
and
necessarily-
see. We maybe
yeah.
we don't even need to put any pressure on it.
But yeah, Mike, if you want to just grab the slurp handle, is that what you're gonna do? Yeah. Yep.
Should we do microbiome stuff or metagenomes on this?
Yeah, the dye doesn't affect the sequencing. That's cool.
So how many boards are there?
3, I think.
How many papers are for?
1,
Were there any?
maybe.
Never mind.
Yeah, I don't think we need any pressure on it. Yeah, if we just hold that around the outside, it's fine. It'll be awesome.
So now we're taking a little trickle of a fluorescent dye called fluorescein near the base,
Side near the base.
near the bottom. Yeah, around.
Current seems to be.
Yeah, maybe on the left side of it. If yeah.
1, despite this thing saying 1. Because we did not see the very first sample.
Yeah.
Why?
Yeah, it's a good target species.
For why? Yeah, I got a multi-electron microscope. This one?
Mike.
Hmm.
We get this, it's a big sample, it's gonna be 635.
Leaking a little bit, but I don't think it's actually leaking that much. Yeah, I'm not sure which one they put it on.
They told me it's not powered.
It's hydraulic.
We powered it on the deck.
Yeah, I mean, they said right now it's not powered. It's just leaking, you know.
Right.
Oh, shoot, could we put a little power on it?
Do you know which one it is? Oh,
You know which one? Which one is
you know
it?
which one it is?
What's that?
The RAM for the dye.
Yeah, it's 1.
It's number 1.
Alright, so I just took the section sample off, so now we'll be pulling it at the bottom.
Okay.
I wondered if it was still on.
Yes, I
Okay.
was watching upstairs.
Where's the power for that?
1.
Science Relay 1?
No. Ram.
Oh, RAM.
Okay. So if we ram, yeah. So I guess we can go out and then back, right?
Squeeze, yes, squeeze.
Just a little bit. With some
Oh, squeeze and unsqueeze.
Yeah.
So let's just do a little squeeze and see what happens.
Squeezing now.
Yeah, there we go.
Okay. Let's
All right, well, I would
stop.
say the sponge is not pumping. That's plenty of dye.
Bridge, are we? Yeah, look, when you get to that position where they put you, is there a possibility to do one shoot? Because we're gonna head up north and back up and there's a big cliff ahead of us. So we'd rather have you south of us.
Not much current right now, man.
So is it still squeezing?
Okay, great, thank you.
I think. Can you? How close?
Unsqueeze.
Yeah. Okay. Unsqueeze.
So it's still squeezing right now.
It was, yeah.
Okay, great.
So now we are unsqueezed. Can you tell how far? You can't tell here, right? Nope. Just sort of unsqueezed all the way.
It stops. That's pretty good.
Okay, stop.
Okay. All right, so we checked. Let's see.
That's great. I feel like that's pretty good, Scott. So if we just fill the bag less, that's a pretty low leak.
And you use the same tension on the
Just
bungee?
leave it, but just put less in.
So when it gets back on deck, we can just drain a little bit of it. Right?
Yeah.
Okay, cool. Yeah. We could also try to come up with a strategy here that
A new strategy,
doesn't-
definitely different arm. So
a
it's not
different
on the
T handle.
slippery. Yeah.
Different T handle, yeah.
Yeah.
Okay. Yeah, I don't think that guy's pumping.
No.
I mean, it's pretty with the giant barrels.
You should
have
really
you
be able
seen-
to see it. And even here, I'm not seeing it even go over the middle of the sponge.
Yeah.
No, it's not pumping.
You can see we're probably that far from it.
But still.
Yeah.
No. Yeah, but that's awesome. Thank you guys. Great job.
Yeah, we can stow that.
Well, that was exciting. Next time we'll find a pumping sponge.
Squeeze and unsqueeze. Well, yes.
I'm gonna be slowly coming towards this point here. Once
Okay.
he gets to this point where I am, he's gonna do a 180, so I'm gonna swing out to the right.
Okay. Perfect. That's fine. Do it. Yeah, that's great. Maneuver where you need to go. We're what's our depth? 260. I think. I'm trying to move them up a little bit. That's what we're gonna find. But they're just like they're trying to. We're you know it's balancing. We're kind of So. And I would like to do that. Yeah, it's already 10, 15, so we're offline. Then sometimes.
Then coral quest comes.
Then cool.
See anything interesting while we're doing this maneuver, let me know.
All right. Yeah, we'll be watching. Science eyes are watching. Looking for yellow paramericiids for some coral incubation experiments. All right, awesome. So the deep sea fluorescein delivery engineering project has been a work in progress, and that was our first test of it. So you've been a little bit of R&D. What we've got is a hydraulic ram that kind of squeezes two pieces of plastic between it, and there's a bag of dye, and acts to kind of gently push on those two sheets of plastic and squeeze the dye out of the tip of the wand here. Colleen, you got lots of thanks on the chat.
Thanks, Colleen. Yeah, look, once you get close to that position, you can go ahead and start maneuvering doing the 180. And I'm just gonna slide on.
We're at about 252 meters.
We're still pretty close to the cliff, so why don't you come out another 30 meters or so?
It's like you're getting to talk.
Keep it the same way.
It's like a shot. That's how we looks at our daughters.
Right, launch and recovery is on the side of the boat. That's why it matters.
They're just asking
So we've got to do a dance system for the RV happens over the starboard side versus, for example, off the back, the stern of the ship with an A-frame, for example. So when things happen over one particular side, with the tether, the cable, and everything needs to be oriented and matched up with where the RV wants to go. So we want to basically we had been transiting to the southwest, and we want to move now to go to the northwest. And so Falco's on the wrong side of us for that. So we're doing a little ship maneuver and RV maneuver to sort that out. And in the meantime, exploring along this steep terrain at 250 meters. Really, much deeper, there was really just oodles of sponge cover. Here we have the same cover of sponges, but seems like they're smaller. There's lots of stuff in the water.
If they want to access like a little bit, do they want to access a little bit right version of the stream? Or do they just
No,
want
I
that
think they just wanted to make make logs. Maybe they saw something interesting. I think they wanted.
I can,
Enter
I'll answer that. If they want me to type, if they want me to type anything, I can.
Sam, there's a coral here, right?
You can type to me and then I can put it in there.
Yeah, with the black coral.
That one, yeah. Is that the Anthopathes? Acanthopathes? Whoops, sorry.
Is there like a genre, a comment that I can?
Yeah.
Like, is there just like a general observation comment that I can add? Is everything very specific?
We were pass on that.
Another one up there. Hmm?
The images that pass on them, sorry. So I'm trying to find the images that go every minute. And I thought they'd be video.
Oh, really? I'm
There's another little glass sponge in the lower left. No, that's okay. And a big urchin up on the top.
Science. Yeah, it was science.
We have not seen any of it, so.
I was having the same problem. One of the computers in there, the main computer lab, the computer is right here, and that's the hallway that comes into this room. I had some things open to try to copy those C-log, and that C-log folder, those images that go every minute. I was trying to copy one of those, throw it in that and see to it, and I just sort of did that directly. And it's giving me that same error on that computer.
Very cool sea urchin.
It would fit now though.
Stairs, so is that,
Yeah.
Furniture right next to that portal. Yeah. What is it called?
Oh, is that a
Coral?
big sponge up there?
It might be.
Might be a big sponge up here.
So it could be more saucy. That's actually the one. Doing it for. Yeah. Oh, it's got one.
Let me try to log that.
Shall we try to
What address should I use?
put a little dye on it?
It
See?
might be 1111.
Want to try to Solaris it first?
Who's the one with
Good,
the
yeah.
green?
Oh, it's the same thing. They're like, oh, wait a second. Can you take notes, Scott, while you're sitting there?
Yep, I have been.
SOLARIS first.
Yeah, I think so.
What is a snot tube? Yes, that is in the wall. It's a sponge.
What are you talking about?
Is that a technical term?
I don't think that's
We're gonna go to Solaris right there.
Yeah, give it a go. Should we do outside the sponge first? Like on the side? I guess we are outside.
You want to do a background CRE?
I'm already.
Yeah,
Okay.
okay.
Stand by and we'll position it. You ready?
No, it's
Okay.
like 20 seconds.
This will be the event. This will be the event. Yes.
Falcor still pivoting? Okay, cool. Okay.
Okay, we're ready to measure.
Okay.
Just ask him to 10 meters to his port.
The jellyfish?
Yeah, go ahead, Luke.
That's the tentacles or something like that.
It was like
probably about
one of those feeding experiments. They
250,
control
250.
feed them with calcium or magnesium free
Or
water.
reciprocal of your last position. Still what he.
But when they did, they should.
Hey Luke, before you do that, can you move to your port about 10 meters?
First and ROS.
Well, it's definitely the same thing.
Yeah.
Roger that.
Morning.
Thank you. Because we're
How's it going?
doing a dive
Looking at a lot of sponges.
on regulated circle.
Sponge land down here.
See, I see. Watching Encore.
Just keep
Uh,
saying
No,
that.
just lateral port, about 10 meters.
Yeah, Nicole did.
Ready to go in?
Yeah, they're ready.
Continue
Okay.
to,
Inside.
Mike, you want to go? Yeah. Put it inside? Yeah.
Fish. Andrew, what kind of fish do you think this is?
Roger that.
Just down to the bowl.
The green dye is really cool. Color.
Some black holes like that. Did the sponge suck it up?
No.
No. That would be
This one's a beautiful sponge.
Yeah. Waft.
We had another idea in the kitchen,
Very,
a PVC pipe of some sort, and shove it in it and kick the tube.
Yeah, I was thinking about a way of just
Oh,
closing.
like it's shut
Like
some
dark,
sort.
it was black, not black, coral black, like it was, it was one we were panning across.
How long ago?
10 minutes.
Yeah, deeper. If you want to, if you can get within a centimeter or two of the surface of the. I know it's hard to tell.
Are you
Like
getting any
ranch.
measurement?
Yes, I saw that.
It's not really changing too much, so
It
it's
did,
around
so
10,000
I saw that
outside.
as well.
It could be a tree.
That's the background.
I think because it was more.
Yeah, but some corals could look like trees. No, there was definitely a tree branch.
Oh,
Yeah.
tree is tree.
Oh, no. I saw that earlier also, that big tree.
The only corals we see are like the royals and
It was like in a crevice.
Hmm.
Yeah, it's a really pretty blue.
Yeah, they
Let's get a stranger of that.
Pretty well.
Turquoise.
There's something alive on the right of the sponge on the outside.
Yeah, there's some shrimp in there. See, you just went over the edge,
On the
Right
right
here.
side.
right here.
Because somebody's crawling.
Well, there you went.
It's a seahorse.
Oh,
I
oh,
want to see you see one.
sorry. Oh, you're not listening. I accidentally tapped the mic.
That's okay.
Something right here.
Ah, there we go.
Ooh, yeah, that was our big issue.
This little thing down there. I don't know what that is. How are we doing on?
Nothing.
Nothing. Okay.
We do hopefully see. Really great placement with the lawn.
Was there something there?
What's that?
Scott, are they normally that blue on the interior, or is that, that's just an artifact of the camera?
I feel like that's just the lighting. But...
Okay.
I think you will be able to
Okay,
see.
well, we can pull the stow on, Mike.
We've sampled this, I think the same species, so we'll be able to see if it's blue.
Whoa.
Scott, are you know this down?
Yep.
Yeah.
Okay. All right, we're gonna stow the wand. I'm gonna see if we want to squirt some fluorescein,
Sure, why not?
squirt some dye.
Although if it's not producing ROS, it's probably not perfect.
It's not, but also this will also get us a little more experience in
Yeah.
squirting of the
Yeah, for sure. Oh, hello. Didn't know you were
You're gonna say something rather than just getting like,
Yeah, that was scary, because I usually get my back.
oh, that would have been something.
Hey, when we got that earlier sponge sample, we got some resoyans on it, right?
I think so. No.
It did.
On the backside?
Yeah. It was
Oh,
sided
you know
like,
what? I have
Oh,
pictures.
one of the
it
ones
was
we-
sided just like this one, but it was like a it was like a paddle.
No, she means a different
Yeah.
sample we pulled up. We already sampled a different dye.
No, the
Oh,
one
yeah,
we
the
did
one earlier today.
Yeah, it had zoanthids on the other side.
Okay,
Yeah.
Andrew's requesting maybe we can put some in a little
Oh,
tube that,
yeah.
no?
Yeah, I think we were already gonna do that.
Yeah, cool.
What does she think it is?
I think she agrees with theirs.
Parazoanthus parasiticus.
Oh, I was spelling with two O's. Oops.
The zoo
So did I. It's not.
It's not.
No,
Just
one.
one O.
Yeah.
Yeah, I thought it looked wrong.
So outside first or?
It's only gonna be outside.
Close to the bottom. as
Yeah, near the
possible.
bottom. Sort of. Yeah, exactly.
Yeah.
Sort of down here, over here somewhere.
Yeah,
And we'll get.
that's good.
There?
Yeah, and let's give it a little squeeze. Oop. Maybe a. Yeah.
Oh, yeah, this
Did we-
was the sponge we saw the other day.
Did we give it a little squeeze?
This is what it was doing to that sponge.
Squeeze? Yeah.
Remember like it had like tracks?
Yeah.
Squeeze.
Zoomed in on like a day or two ago.
Squeezing. That's it's working. The squeezing is working. Can we place the tip of the wand more upstream?
Yeah.
Sort of up on the sort of 2 o'clock side of the sponge here. See if that changes anything.
Yeah. And is the setting just on off?
No, it's squeezing. I mean, he squeezed for a little while, right? And then
It makes sense.
squeeze a little more. Yeah, squeeze a little more.
Yeah.
So the sponge pulls in from the outside. So ends are kind of putting themselves before the sponge and let the sponge pull water past them.
Yeah.
That's great.
Oh, is it pulling it in? Oh no, that's the.
I don't think
Oh,
it is.
You
It's
would
only-
you would see it coming out in here if it were pumping. You'd see it start to accumulate in this bowl.
Yeah, I don't think it's pumping.
Oh,
It's fully, the ram's
Yeah, when
fully
it
in.
wouldn't move, I thought
It's
it was
fully
like-
engaged.
Look, it's like your cat, it's following the laser dot.
Okay, kitty.
All right, I don't think we've got any pumping.
That's great.
So now unsqueeze fully.
With the laser,
How can you tell how
I
extended it
think we
is?
can turn it off and
Okay.
turn it
there's
back
a little
now.
chain on it, and then he'll watch it in your hand, and then he won't follow.
Okay.
the laser.
Okay,
That was
Look
awesome.
at it zoom in, and when you're done
That's
with
such
it,
a
it's just
beautiful
like
sponge.
Oh, yeah.
it is.
just great surface.
Hey, Scott,
Yes.
we want to zoom in on something to the left of the sponge for a second.
Is-
So like,
Can I
behind
see a
the
stick?
sponge to the left a little
Yeah.
bit.
Take a
Right
look.
here, Jeremy?
Yeah, please.
I can't, sorry.
So there's style patterns all over, but this looks different. Just curious of this. And then,
Ooh, oh, you saw that. I was like,
like down a little bit.
A little
to the
tiny
right.
thing?
Style patterns.
To the right?
This?
Yeah, there you go.
oh. Wow. Is it a little, is it a little tiny cup coral?
Little cup coral there, yeah.
Oh, guys, so cute.
We're fine.
How big is this rock, do you think?
This whole rock?
A few inches.
Yeah, like probably this big.
Okay, so, that,
Could you
what is
focus
the cup-
on the coral, the branching
Black.
coral?
What is a cup coral?
So this is Claritidium.
It's
Solitary.
Oh, they just exist as one coral.
Yeah.
Hmm.
Sometimes they can be like a few clusters.
I'm gonna have to get out
Did
of
that be
to
by
get
real
repositioned.
pathogen?
My temperature's rising here,
Oh, okay.
so I could get
Sorry.
closer or
By
whatever.
real pathogen, maybe?
What do you think, Jeremy?
Massive in the sink.
I don't know, is it, would it be okay to just grab that rock? Is it possible?
You could try.
That coral is really
No,
weird.
don't grab that rock. You think they can fit in a quiver? Only if they can fit in a quiver.
I don't know.
They're not gonna put that in the biobox, so we're gonna try to find experiments, yeah.
It's not gonna fit in a quiver.
Okay, I don't think it's gonna fit in the quiver.
Okay, alright.
Okay. Happy?
Yep.
We can proceed? Just,
Proceed. Are we good to start moving northeast, west?
Yeah.
Okay. So we came down to about a depth of 245.
Sorry, no, that's strict rules. We got in trouble. That's weird. It was weird. And we'll be with this. I'm gonna get in trouble. Okay, we put that on our ROV.
You guys are on mission here?
What?
You guys are on
Cool. Got specific corals to get.
Okay.
Alright, off bottom. I'm gonna start cooling down. So that motor temperature, that's the hydraulic motor? Yeah. You know, I mean, but the data, I mean, out there on sponge pumping rates, I think it's fairly sparse as well, right? Like, there was some early work done, and
That's why the dye is such a cool way of visualizing it. And there's a lot of Israel that's doing they did something, I'm tired of forgetting what it was, but they created some way of modeling like the size of the sponge, and then not simply the velocity, but like for that sponge itself, with its size, what that speed is like changing.
Okay.
Size, it's some sort of special super type measurement. But other than that, I haven't really read any papers on it.
And with these other kinds of with the sort of vase morphology, it's easy to imagine that that's a easier problem to model than this amorphous kind of blob that are how do you even approach that? I know some folks that have done flux chambers where they put the whole sponge, cover it up, and then try to say, well, this sponge changed the chemistry by this much over this amount of time.
Don't want to perform on camera.
Yeah.
Yes, Liz. If
Oh,
the
Liz is
sponge-
asking. Yeah. Sponges can be disturbed and stop pumping. That's right. And one of the things that we're interested in is getting water samples from inside and outside the sponge while they're pumping, and by comparing the contrast, the differences in the chemistry of those waters, we'll understand more completely what kinds of biogeochemistry or biogeochemical reactions are happening on the inside of the sponge. And then Kate, of course, here is looking at the microbial and eukaryotic communities on the inside there that would be responsible for some of those reactions. So trying to plug all that together requires a little bit of cooperation on the part of the sponge. And I think at these depths, the large, so-called emergent sponges that we see are mostly these glass sponges. And it could be that their pumping rates are just very small. But we know that Zestospongia muta, for example, was the charismatic giant barrel sponge. Those have been studied fairly well, and using some of this kind of these dye approaches and things.
So is the red the deeper?
We're starting to climb up. Okay. Red shallower.
Red shallow, so we're going deeper now.
No, we're going that way.
But we're the white line.
Yes.
Yeah.
Yeah, Falcor is the, and this is our heading here.
Is this where you want to go?
Yeah, not, I think moving up onto this, not necessarily specific. The stylastras, we actually spent a lot of time today using SOLARIS to measure superoxide on stylastrids, and did not find much production. So now we're coming up on 200 meters depth on the Desecheo Ridge. We should, soon here start seeing evidence of photosynthetic kinds of organisms and algae and crustose,
on the
coral and
bottom,
crustose
there's
algae. Yeah. Just kind of hoping as we move up a little bit shallower, Sam, that we'll see more candidates for you.
I started an eDNA pump
Okay.
a couple minutes ago. This is just, we are doing a new thing that if we're traversing,
Yeah.
we have a 0.2 micron filter on.
Okay.
Just to see what we can get. And this is actually really good timing because the community has just changed a little bit.
Yep.
See more CCA, more black barrel.
Yeah, we're coming up shallow. We're at 195.
Oh, yeah, it's really changing.
195 meters. This sample is SH13543.
SH1, wait, SH18.
We have a different set of numbers.
Okay, SH13.
13543.
543.
Sorry, that could be confusing.
That's okay. So we're starting an eDNA sampler. That's, yeah, I think those are stocked crinoids, yeah.
I love the one.
Stick it back. The curly Q, that's a black one.
I was gonna say that and try to impress you. Beat me to it.
Sorry.
What kind of black coral?
It's a Pathies.
Yeah.
I know Pathies is black coral.
Have we seen any production of ROS on Stylastrids?
What?
Have we seen any ROS on
We have not.
There's just a question.
Oh yeah, there's a question there. We have not found any production of ROS on the Stylastrids so far. Oh, there's some fishing line.
Oh yeah, watch out.
Watch out for the fishing line. Straight ahead of you.
Go vegan! Plug in.
There's a stocked crinoid.
Gotta cut up a sponge, man.
Not
We
like
did that a
too
couple
much,
times,
just
right?
like a
Yeah.
That's how we
flap
see the
or
RS
so.
spike.
We did see that.
So
Yeah.
far, it's been the biggest spike.
Moving
Also
forward.
just like kind
Okay.
of stand behind while they process and then like fill it up. I like fill up the tubes extra. Probably started this.
We are moving upslope onto what I was calling this kind of nose along the ridge. South-facing nose. Here's some stylopathies. Is that right? Stylopathies?
I don't know. I have to see it closer.
Alright, I'm gonna...
There's a few kinds of black corals that are bottle brush.
Oh, okay.
South Pathies is one of them.
Okay.
South Pathies is really floofy.
What's our average altitude that we've been flying at?
Oh,
3 meters.
yeah.
3 meters? Thank you.
Here's some corals. Over here on the left, Sam, these yellow ones.
Yeah, that's a kind of black coral.
Okay, you don't want to stop for those.
Well, if we see, that's the ROV pilots. Do you think there's about like a dozen there?
Yeah.
Do you,
Which one do you want?
All of these different yellow corals back here on the back side of this. Sam, do you want to just get a visual on some of those?
yeah, if we can sit down and measure 4 or 5 of them and sample 4 or 5 of them, then I think we should do it.
We can do that. Of these yellow black corals,
Yeah.
or the Stylopathes-esque looking ones.
Well, we already got 4 or 5 stylopathies from the previous dive, so
Okay.
we shouldn't go through those. But we're trying to find another coral that we can get 4 or 5 colonies of, both measuring with SOLARIS and getting a physical sample.
Does this seem like a good area for you to collect those for the experiments?
We're gonna avoid using black corals for the experiment
Oh,
at the moment.
okay.
It's gonna be harder to keep alive.
But you'd like to set up and sample
Here.
3 or 4 of these.
I'm sort of asking the pilots if they
Yes.
think they can sit in.
I can sit here.
There's some boulders in the way.
Go on the other side.
Okay.
This area.
Okay. So we're at 183 meters and setting up to sample some of these black
Doctors who
corals.
see white things are coral?
Which one? These?
Yeah.
That's perfect.
This?
Those are so crazy.
Yeah.
Yeah, they're really
Are they all, is it a monophyletic group?
No.
Some are octocorals and some are.
Really?
Yeah.
Pretty
Wow.
wide ranging.
So it's a convergent,
Yeah, a lot of
like, shape?
yeah, there's a lot of converging everything in corals.
That's crazy.
Or a lot of the things I'm looking like when I'm trying to look for the morphology is either not there or it's like just they're similar to other ones or it's like the differences are so minute between groups that they need to be done like microscopically or
yellow one, right?
genetically.
Yeah, I think so, yeah. Sam, the yellow ones, right?
Up to
Yeah,
like
the
the
yellow
family.
ones.
And I'm like, great, cool.
Yeah, I think there's some. Whoop.
This one?
Yeah.
Is that a sponge?
This right here?
Ooh,
Yeah.
Oh, neat. Yeah, that might be one of those. Yeah, this
Wait,
could be a.
Does it have one
Oh,
Is it 2 oscilla or
what happened?
two siphons? Like a
Um,
giant city. I doubt that, but.
Yeah, we might sneak a squeezer into that sponge, just for. But we're gonna collect sand. We're gonna collect these colonies here. You want measurements with Solaris though?
If you can get it,
No. Well,
Yeah,
we're going with corals first.
the outcrop first.
Are there enough?
I just don't think we're deep enough to find the target corals that we're after.
Yeah,
So I
you're just
think
calling that.
The hard corals?
This one's target corals that we're
Target
after.
corals.
So I think
Oh, wow. this is
Santiago's
great
goal is to
spot. Okay, perfect.
get like 4 or 5 colonies of a few different species.
Okay.
We were trying to find the same species he saw before to round out their numbers.
Yeah.
But we're just not seeing them at that. So I
Yeah.
think now that we see that there's at least 4 or 5 of these, we
There's
might
at least
as well-
more corals here, right? Yeah.
Yeah, we should
Okay.
Just a little bit.
do 4 or 5 of these and then another species.
Okay. So we can Solaris. Yeah.
The parameter we have a lot of these, I know,
Okay.
only found deeper than 200.
I'm gonna maneuver a little bit more to the right, so
Okay.
you wanna put the dye where you want.
Well, first mostly what we're gonna do is use Solaris, measure a few of these yellow corals, then collect them. Then we'll probably, we can also use Solaris to measure in this sponge.
Well, what if we try the dye next to the sponge first before we disturb it?
Yeah, we could try that before we poke.
Before we start waving
That's a
around.
good idea.
Kind of yellowish.
Let me just move a little bit closer.
I think, like Santiago was to a few species, we try to get like 4 or 5.
You think we should do that before we collect corals?
Sampled some of the past that I compared
I
last
kind of think that the sponge will stop pumping if we're waving around.
Round out those milkers.
I mean, it might not be
because I
pumping
just think
anyways,
those
but-
species are-
Yeah.
Our best shot at it being, at pumping is it being our,
This area was.
Right now.
our first prod of the area, you know?
Add this yellowish, green, black coral, there's like a dozen or 2 of them
Really
here.
cool. Okay.
So we can just bank that as a
Is that better for you, Mike?
gold breasts, and then see if we can find other species around here. This is not a model brush.
Wow, it's such a beautiful shot.
It might just be that it stinks like okay.
Am I good to start
Sorry.
collecting the background signal?
Yep. So she's gonna take a background signal with Solaris. Wait, so we're gonna, you wanna try the,
Stand by.
What do you think?
I got too close.
Those was the ones there that they're.
I think with the Solaris coral measurements, we'll be far enough away from it.
Okay.
That's the one that they got a reading on.
Oh, okay.
They got a reading on.
Which one?
So that, I think we should try the yellow feathery one on the left. 'Cause
Here?
Yeah, because we know that there's like a dozen or 2 around in this area.
Okay.
So Solaris first on this yellowy one.
Here, yeah. And then, then can we, yeah, there's a few others, I think, within reach.
Look good.
Oops.
That's got, I'm saying, yeah. It looks like it, but I think it's diced pretty bad, because it doesn't have. What comes up that over there?
Alright, we're good to move closer.
Okay.
This is event 8 for anyone who's,
This is what? Event?
event 8.
Pretty close there.
It's pretty close here already.
Yep.
You wanna go the other side of it, or?
No, I did that.
I think the current's kind of going right to left. So,
It's coming from forward.
is it?
Yeah, it's heading directly. into
Okay,
this.
then, yeah, you're in a good, I think in a good position. Yeah, there. Yeah, that's great.
Nothing so far.
Okay.
See everything traveling.
Mm-hmm. No, the last bunch we did not.
But it really looked the same as the other one that we got.
That you did. Yeah, I think it's probably the same.
That was part of why we.
Can we maybe poke it a little?
Sure. You want to give it a little bump? The opioid. The opioid is like, what are you doing? What are you doing, man? There you go. There's some bumping.
Started doing a sample and they log it and then I call out kitty.
They stopped it.
Yeah, it never
Oh,
started.
oh, oh.
There's a signal.
There's a signal. Oh, flying. What?
Good.
Yeah. Nicole's like, what? What? Yeah, that's a pretty clear signal. And that was, I think, after bumping or after moving to the other side.
Went back down.
I mean,
So it's
oh, hey.
probably initiated by the bumping.
Yeah, but I mean, SOLARIS is working.
Yes.
Yeah, yeah, yeah.
That's part of the,
Yeah, that's a good grammetry thing, yeah.
yeah.
Okay, cool.
So clearly can make it, just isn't
Turning
making
off
it
the
unless
pump.
we perturb it.
Okay, I think what we're gonna do is make a couple different measurements of some of the, yeah.
Okay, I just don't wanna leave it on.
Yep, Sam, you wanna point out the next colony? What about, is this one easy to reach? That one okay? That's a different one, isn't it?
Yeah, it's a different.
Never mind.
species.
That guy, is that guy reachable? Or these guys?
Dies.
Not really.
No, I could just reposition.
Can you zoom in on it just so we can confirm that it's like that other one?
These two?
Yeah, those two. Okay, yeah, I think that's
Same.
like, yeah, it's the same. Yeah, so you can measure those two. The one we just measured is going to be SH18635.
Alright, we're gonna,
Okay, let me take her back now.
alright, we're gonna do a background measurement here for a sec.
Shows it there.
You see what
Alright.
that number is too, Scott, and that we had superoxide production?
Did you already give it an SH number, Sam?
Yep.
Yeah, the one we just measured
Yep.
is SH18635.
635.
I don't think we've had it yet that we're just everything okay. So I think they were all like, the main one.
We got one of them, but
Oh, we did?
on the last, our last night.
Okay, a couple minutes ago, I started
Maybe I'm
using
missing
a
like
circle SH.
1, 3, 5, 4, 3.
I'm
Oh,
just
okay.
moving to, yeah, 1, 3, 5, 4, 4.
Okay, we can get closer whenever, and this is about 9.
Okay, Mike, you want, we can move in and get close now.
0.2 micron filter is gonna be the first one.
Is this gonna
0.2 micron filter. This is an eDNA. And this is SH13544.
Correct.
Starting now. Or it
Maybe
already-
5 minutes ago.
Okay.
Got a lot of data to log first. This is altitude at 0 meters, 184 step.
We are sitting on the bottom at 184.7 meters. All right, the wand is
Super tight.
still a little far, aren't we?
Great photo of SOLARIS.
Wait, Dr. Dennis Crespo works at the Smithsonian? Can we get a look closer?
Yeah, I think we're still kind of.
Solaris sample control.
That's stylopathies right here.
What's that
Right
little
there.
white
It's like
thing?
really, really finely branched.
What's the white thing in the?
This, that's a worm.
A worm?
Filigrina.
No way.
It's got a little fun there.
Oh, cool, like a Christmas tree worm almost, but
A little bit, yeah.
that's really That's
Stay
so
on
cool.
that.
I totally wouldn't expect it. I thought that was like the little basket.
I don't know if it's.
Okay, now signal, can we bump it again?
Yeah. Won't pick it up.
Are we saying this is also dicey?
Yeah.
I wonder if there's a diurnal cycle with ROS. Like
Sam, does this
That's
one?
it. He's touching it.
Okay. This
Maybe you've seen that.
SH 18636.
Yes.
Yeah, but this footage is trickier.
He's a millimeter away.
All right, gave it a little bump.
It's such a small little piece. Maybe.
Sam, can you repeat that SH number again? 186.
SH18.
There it goes.
36.
36.
Well,
Just a little
tiny
bit.
blip. Looks like a blip though. Tiny peak.
It's just such a tiny.
Very tiny though, yeah.
Why don't you put it in the pocket?
65 and
bigger
66.
coral.
Okay,
You want to try and put it in the pocket?
down in the pocket there,
Yeah.
near closer to the base.
Yeah.
Sure.
Surrounded by everything else. There you go. He's in there somewhere.
Yeah.
Okay, turning off the pumps.
And you think?
Okay. All right. Okay. Well, she turned off pumps. Okay.
We're trying to save the region.
Yeah. Okay. So let's. So if can you reach the third one that's down below?
Jeremy knows him.
Which one? Oh, this one.
Yeah. Right here. Oops. Sorry. Right there. Jeez.
Is this a third colony right here?
Yes.
Okay, that would be 18637.
SH 18637.
Yep. That's H18637.
Okay, we're seeing a little bit of a signal.
Yeah.
It's like 1500, so it's not like 2000. It was like a little pulse again.
Okay.
Which one had the signal? This third one?
Oops.
Yeah.
Got my notebook.
It was like a small pulse.
A little pulse. Shall we bump it?
Yeah, I think so. But we can't sing. I was about to start singing. I'm bumping.
There's this guy that wrote a song. His name's Ghostwriter, and he used AI to make Drake sing it. And he
Like
posted
there
it
was
on
a
I just
decrease
saw
there.
that. It's
Yeah,
so
there's so
good.
little.
Was it?
I'm trying to
Yeah,
sing.
it's like the
Just
best
too
Drake
small,
song.
but
It's really good.
So we could DJ our own music generated by
If we see a bigger one again, that would be good to get, but for now.
They're also making like Kanye and like other people. Like,
I would say
they're
there's an
making videos of them
increase.
singing other songs.
Yeah.
Small.
But it's just
Weird.
small. Yeah, because it's so tiny.
Okay.
And it's, I'm getting diluted out by the background seawater, I'm sure.
It is
Yeah.
a black corals. Little cruise.
But look at that cool
I just
sponge.
noticed it, there's just little biomass, there's little tissue.
Yeah.
Okay. All right. Do we want one more? You said 4.
4 is our target sample size.
Is there a 4th?
It would be nice to find a bigger one.
Yeah, maybe we can sample these 3
Okay.
and see if there's a bigger colony nearby.
Okay,
Like
bless
the
you.
bigger one, kind of like the first one or bigger.
All right.
Are we gonna search?
So-
or are
There's
we
another
gonna
one
turn
down
else right
below.
here?
Nope. We're gonna. You can turn the pumps off.
There's another
Are
one
you
down
gonna
below.
do that sponge while you're out, or no?
We are, but I think first we're gonna collect. There is a colony down here. Is this big enough for you guys? Right there that he's looking at. Still
It'd be
small.
nice to get a big one like the first one.
Okay.
That's bigger than the other 2.
It is, but yeah.
Look at the squid.
Where? Oh, I see.
Well, we have the SOLARIS
Solaris or not, though, maybe we should measure the ROS of the sponge.
Yeah, when you have it out,
Okay.
we'll just do that.
Do you think you can slip that in there?
Do it outside measurement first.
So we'll do it. Oop, oop. Sorry.
Is it not working again?
No, the pump's turning. No, we just bumped the rock a little bit. It was. I think it's fine.
Do you want to look at the
Background. Yeah. So we.
Doing the background.
Okay. So we're gonna. All right.
maybe
And
a little close on that side?
Do we know what kind of sponge
Yeah,
this
the right
is?
angle.
no.
Yeah.
Brown.
This. You know what this looks like is the ones we were the dirty marshmallow we were calling it.
Dirty marshmallow.
Okay, just
Yeah.
store it on.
This one doesn't look like the dirty
I
marshmallow.
apologize for the terrible names.
I'm going to have to
This looks more
reposition.
similar to the peach stuff.
Not the toasted marshmallow, the burnt marshmallow.
Yeah, maybe it's a bit.
No, I'm serious. They were the ones that were like the big round boulders.
Yeah.
Oh,
Just
really?
Yeah, that was the dirty marshmallow.
turn the angle of it and open up the angle a little bit.
Scott, I'm going to have to turn a little bit.
Okay. We're going to reposition the ROV here a little bit. Get a better angle on the orifice, the osculum of the sponge.
Okay, we can measure now.
We need to reposition the ROV here for a sec. Sorry.
You turn off the pumps maybe.
I'm used to it. I have to ask, and I need to be told no. It's just because. What is the stick on that piece? Was that like 10 minutes ago?
I'm just going to carry us. Frame sitting on the rock underneath.
Like just the circular mounts?
That does look just like.
Is this, yeah. Are you going to try it from this vantage
Maybe
point?
we're a bit too high actually, so we'll come down low with the arm, do it.
Like
Maybe
that.
get that bumper closer to where you had it, but just like
Yeah,
on this
that's
heading.
what I was talking.
You know what I mean?
Amazing.
So that at least we're
Wow.
got
So
a shoulder that we can use.
Okay, but the professor seemed so nice. It was, I wrote it down.
There you go.
Yeah.
Yes.
Oh, and this is.
The thing we have
Definitely not. This is not.
Well, can you put my
These feel that like colonies are too dense.
It's like the one you got earlier. Right?
Yeah. And this is like just a bad picture. I mean, it's a big dead ringer.
we discovered in 2020?
Sure.
This might be a
This could be the first record, well, the second record.
Was a little spoon board. It was
Can't stay here very long.
Yeah. They found
What you
it.
gotta do? No, I just can't stay here very long because I'm applying a lot of trust.
Well,
Yes.
Are we measuring?
we need Hollywood.
We're going to try. Yeah. Yeah, go ahead and turn on.
No, it was an article on what is how Dr. England and one of the students on board.
All right, so thus far on this dive, we've only measured using Solaris on corals and glass sponges. This is the first sponge we've looked at here with Solaris that has a nice defined osculum or opening, effluent opening here. This is where the water is pumped out of. So we're going to try to sneak the wand inside. What's on the tip of the wand there? Huh.
Can anybody give a best guess for the sponge? I can sort of break it.
Oh,
Yeah, I don't know, Chris has the film up.
oh yeah, I think our...
Would you tell us? We have a few minutes.
What about ideas?
I'll make room to this one.
He's gonna put it in? Okay. Yeah, go ahead.
So much paint.
What's our depth?
184.
184.
September.
There you go. Halfway to the depth range.
Nice.
He's at the mouth now.
Can you sample this?
Okay. He's in there. Yeah, we're waiting on the instrument here.
Oh, it's
Like, are you gonna be able to collect these, you know? Okay. I was like,
Probably about 3 minutes. Okay.
not really. I guess not.
Nothing.
Yeah.
No. I don't think we've got anything. Wait, yeah.
Phoebe doesn't like being
Oh.
roped. Just like a little bit.
This is
You know.
going up.
Alright, it's going up now.
Yeah.
Oh, let's get the dye on it.
Well, we don't have, we need to come up, the RV is getting too hot right now.
Oh, it is?
So we're gonna, oh, it's climbing.
Yeah, it's kind of like all over the place. It's
But those are settled
Is it
images?
covering our 1300 I have a baby pulse.
Baby pulse, yeah, it's coming back down.
yeah, it came back down. I would say that there was definitely a signal, but not huge.
Right.
Yeah.
Okay.
Okay, I think we're good to stop.
Okay, I think we can stop.
Is there something up at the tip there?
Yeah, I was gonna say, so
Just look
we,
at the tip.
we will, it dragged on the bottom,
Oh,
and I think that our little filter,
oh, it got bumped.
I think it got,
Just a little bit.
no, I think it got sheared off.
Oh yeah, we changed this to.
This will be collected.
That's so cute. That must be terrible.
But this is
Yeah. It's not
Listen.
bottom.
no, it's like that's more.
Am I doing anything?
It's like more bramble.
Yeah, stand by, go ahead.
Is that one right next to the SOLARIS or not? But I look at it.
Yeah.
Oh, it's fuzzy, you can't see the formulation anymore. Even
Yeah.
over here. This is the white SOLARIS. Yeah, yeah, it's right,
Oop.
touching on the right.
Sorry.
No, that's right, other's right.
I'll wait till we get back on this.
Yeah, so I think we sheared the tip of the filter off.
Oh.
Man, the zoom on this.
Do you have extra tips?
Maybe.
Yeah, I'm pretty sure we have extra tips, but now is the question of do we want to keep collecting and then we have the same issue happen again?
Good question.
Okay. I need to get out of this position.
Okay, you can, yeah, let's take the RV up. Yeah,
So
sorry.
you happy with that?
Yeah. Cool.
Where is the temperature on this one?
60.85 up there, the motor temperature. And then see the alarms at the top.
Above to the rest of the diet.
Is it possible to put like a marker at this location?
Oh, you don't want to run it to always running and creating that one. So we just collect these, when it cools down, these 3 colonies that we measure. And then, I guess, unless you don't want to bring it up right away, you can collect some.
Do you think we have any chance of finding any of the?
Not the tightest mix, but we've gotten even shallower than we were
Right.
previously. We're at 180 now, we're like 250 and below. We're kind of like in like the middle between the 300 depth zone and our 50 depth zone.
Right.
And maybe since there are a bunch of pools right here,
If we can come back.
put some
Yep, yep.
The stickle batteries would be a good one to collect a few of.
We're gonna collect the 3 that we measured.
Great.
Then we don't want to run SOLARIS anymore, because junk's gonna get in, go on there, and then take all apart, and we don't want to do the full rest, so they don't want to do it. But, she said,
But it's okay, we don't need to collect it now.
what I was saying, if there's not a rust,
And
there's that ROV, we have to go 3 testing batteries, we can collect them in there, there's a lot of black holes, right?
Okay.
So it's, you could probably, should just take it out.
That's, we should get one in the building. And then there's another one that's like brown, but another that's like, fucking tip. It's different.
Pretty sure we don't. But then we were talking about that. He also got Wilco written inside. He said a lot of that stuff was in the Desecheo papers.
Oh, okay.
You do?
Yeah.
Okay. Then that's
Was there a little shoot out?
well, we got time. We're gonna get it right now.
No, I know, but I just want to know that,
You know what you're talking about? What we're looking at?
Yeah.
Okay. Okay. I said you
I
go in
wasn't
the show
really
tomorrow.
Or the one little box. Depends on which disco spares
Yeah.
I brought. Brought a bunch of the disco spare.
Scott?
Yep.
You wanna keep heading up? What do you want to do?
Let's see, we need to collect those green, we're right with the green ones that we were sampling with SOLARIS. We're gonna collect those and put them in coral quivers. So we kinda need to sit back down where we were.
Now I'm not sure.
So I think that the amount of dye is something extremely small, it's just a strong dye.
Sorry about the net leaking.
Yeah, I think it's very pretty.
Super small, I just think that it's such strong dye.
Have you seen the video of Colleen scuba diving with
Yeah.
that?
So awesome.
Yeah.
That waypoint was like right in the middle with all of these.
All right.
We're
gonna
kind of sailed off.
Who took that?
It was a well-intentioned one, and I did not, I didn't. I didn't bring it out until.
It looked like I'm thinking hard.
This is the first one for
She showed me my picture and I thought I had to resurrect it.
Who's asking if you
You can put that in your PhD. Hadn't worked with this guy.
Whole collage.
Yeah. Wow. Look at all these. Are these all stylopathies?
The bottle brush ones are probably, not this, these fans are not.
Not the fans, but all the bottle brush ones.
Yeah.
The abundance of
Amazing, huh?
bottle brushes.
Each one has at least one worm. So
Yeah.
you can double the diversity.
I'm gonna miss World Ocean Day here. April
miss it, you're
27th.
gonna be on the ocean.
Yes, this is the best place to be.
No, I'm not.
Oh, you're gonna go there?
Yeah.
No, you're not. On April 22nd?
She getting off at the.
The 27th.
27th. Oh.
Oh, they look long. Well, we should do something fun for that. Yeah. Yeah.
Did you guys find a particular green folder in the lab? I know, but just want to get it started. Make the rounds, everybody shifts and all that stuff. It's like where we were processing all the chemistry. Green folder, card, chickens on it.
That's a sponge there, see it?
How many do you guys have?
three.
No, I just saw
Yeah.
it get too
Why
complicated. Why did the ROV on one of the Pro websites I was looking
Just
at
holding
said
it, holding it down.
biodiversity,
Oh,
then
the storm behind us.
Are you guys trying to find the, are you guys still cooling down or trying to find the wrong beer? We flew over it.
Yeah, I think we went past it. Yeah.
And I think right behind us, if you like turn 180.
That looks more.
Oh, sorry.
Oh, sorry.
There it is.
This is
the
it.
rock.
This is it here. Yeah.
That's our.
Yeah.
That was the big one that we sampled, and yeah, here's the two little ones that we sampled. There's the sponge. Hi.
Yeah. We got some for the crown ends, actually style asked you. We got 3, measured 3, but one that we measured we didn't get. And the one we got we didn't measure it, just fell in last week. This one's we couldn't get. Little sponge. Remember it was for Encore. We found these
Not till
plastic
noon.
patties here. There's like a dozen or
So it'll go past
You
midnight.
can sit here and
Yeah.
measure for and add.
Huh? Yeah, they'll probably be up. I would guess
Just taking a shower.
8 a.m. or something.
8 a.m.
I'm just guessing. Or 4 or 5. I don't know. Without Solaris being, if it's out of commission, they might want to pull early.
So the filter gets scraped off the nozzle so that the client didn't want to run it anymore. So there's a risk of jump getting in and flying in.
No, I'd like to do a squeezer in that sponge too. If we after we grab the.
Yeah.
Make a squeezer.
Okay, I just stopped. Sample
EDNA?
pump A at 75 and 3/4 liters.
So this is EDNA sample. This is 544.
Correct. And it ran for about 30 minutes.
All right, so we've got the EDNA sampler. It's turned off. That was sample, yes, SH13544 for the EDNA. Ran for how long? Ran for how long?
About 30 minutes.
Okay. Sampled EDNA down here for about 30 minutes with an EDNA sampler.
I'm just gonna shift it right a bit.
And I just started pump 9, which is a 1.2 micron filter. This is.
Oh, so this is going to be.
Probably equivalent.
SH13545.
Correct.
It's not that big like this. It's like those 3.
Pump 9.
They were like crowning, but yeah.
So some questions in the chat here are, let's see, who designed Solaris? Yeah, that's a great question. So I was part of that team. Colleen and I started putting our heads together about putting some of her benchtop instrumentation for measuring superoxide in the lab underwater. I've got a background in being crazy or stupid enough to put lab-based instruments underwater, and so we put our heads together and joined forces with an engineer at Hui named Jason Caput. So yeah, so that project was myself, I'm Scott Wonkel, and Colleen Hansel and Jason Caput, and we got the first bit of funding to start the development of that project for the deep sea sampler from NSF. And Yeah, that started, gosh, 3 or 4 years ago. And we had our first deployments of the Solaris on the Atlantis with the HOV Alvin off of the west coast of North America, off California in 2019, and it worked fabulously. And yeah, a shallow water version of Solaris known as Disco was funded by the Schmidt Marine Technology Partners. Same team. So Colleen's group is really interested in understanding how these reactive oxygen species behave in these environments, and she's been really pushing these projects forward with her group.
What are you guys going for first?
The furthest away we'll go for. Yes.
The little one there? That little one?
Okay, that one is
That one should...
18636.
Yeah, that should be 636.
They've got worms. These worms excrete something to create too.
Okay. You have a quiver in mind?
3.
Okay. Already open? You're way ahead of me.
Juliana, I'm restricted from putting things on here again. Same. Yeah. Oh, yeah. Seriously?
It's a small piece or...
Are we taking the whole thing or the small piece? Or how do...
If you can take, sorry, just one of those branches at the bottom.
If you can get one branch at the bottom.
Wait. If you think you can fit in a quiver the whole thing, then we should go ahead and take the whole thing so we have enough.
You might as well
for all our purposes.
Okay, let's grab the whole thing. Yeah.
But don't grab it at the middle. Grab it at the
At the
bottom.
base?
If you can.
It's kind of neat, I went to a lot there, but
I know it's tough.
you get it the best.
This is
But
what
assuming you're not sit for a little bit, yeah.
sometimes you can kind of scrape along the base of the colony and it'll just pop off.
When did the Solaris was busted? Just noticed it. Or we measured these 3 guys, we measured
Yeah.
a sponge next to it.
It
We're about
dried
the
on a
same.
rock, sheared the tip, the filter tip off, which Colleen went to the lab to check and see if we've got spares of those in the Disco spares. I talked to William earlier today, and it didn't sound like there were any of those in the Solaris spares. He thought they were in the Disco spares, which she did bring, so hopefully there's some replacement for that. Otherwise we'll get J-Rod to print something, 3D print something.
Right.
Yeah.
But this isn't this different patches. See the interior visuals.
Yeah, so even just in the time that I've been working on ships and doing deep sea research and science since early 2000s, mid 2000s, the advent of 3D printing has really kind of revolutionized the kinds of pivots and adaptations and MacGyvering and things that can happen. There's already a ton of that kind of attitude and engineering adaptation to problems that are presented by sampling in the deep sea and trying to do work in the deep sea. And the RV teams that I've worked with are all really great at machining and coming up with really novel and clever ideas and how to fix things and how to adapt sampling challenges. But the idea of being able to 3D print is really an interesting. You can actually, start to build things that you couldn't do before. Anyway, it's been kind of neat to watch that happen. Oh.
That's okay.
There you go. Yeah.
Yeah, good. That's good.
So this salmon is
This one may like have some lip.
636.
636.
This is Coral Quiver 3.
Jeremy, which group is Desecheo a part of? Model Crush,
A fan of that targets.
Fan Cabbies?
Yeah.
Just want to turn it.
Wait, a fan cabinet in?
Yeah.
Yeah, whatever you call it, it's fine.
So about fused branches? All the way up.
Well,
Or not fused?
Sorry, say it again.
Is it not fused branches?
Not fused, not branching.
Thank you. And this is quivers.
Three.
Thank you.
Nice. Oh.
Shaky, shaky.
And for this one, you guys saw, remember that first photos we did?
Oh no.
We saw,
Oh no.
Yeah, they're floating.
Oh. Oh. It's there in the. Mm. Oh. How is that possible?
this is a new collection.
So we're using the coral cutters here on the ROV's starboard manipulator. So there's some jaws. There's some blades there on the jaws, along with some red rubber padding. So the idea is that the rubber pads there grab the coral while underneath the blades there cut it at the base.
And they named it after a guy who's he's been working there, I think, it said like 1979.
I guess.
Where'd you do your undergrad?
Yeah.
Would you call this one? Oh, sorry.
Okay.
Did you already click the collect?
Yeah, twice now.
Okay, let's change the later.
What do you want to call it?
It's probably more like asteropathes. Same family.
Anyway.
It's a good one to collect on editor picture book.
There you go. We're still here in the waters of Puerto Rico and along the Desecheo Ridge. Been working here for several days. Just an incredible abundance and diversity of corals here in the mesophotic and aphotic zone. Working with Santiago Herrera from Lehigh University. We've got lots of several grad students from the University of Puerto Rico at Mayagüez, and some students and scientists from Woods Hole Oceanographic Institution. Mostly folks at SOI from the Marine Chemistry and Geochemistry Department, grad students and PIs. Colleen Hansel is a PI, and myself. And we have Ann Tarrant, also from SOI, the biology department. And several students again from the Marine Chemistry and Geochemistry department there at SOI. Interested in lots of different aspects of the work going on this cruise. Yeah, it seems to be sticking to the blades, huh? Yeah, it's tricky here. Seems like the yeah, the depth perception on an ROV using the flat screen cameras here, it's definitely a challenge, and so we have multiple cameras that we use to gain some situational awareness. There you go. Well done. Well done.
I don't think we should collect many of these until I see what it is.
I thought we wanted to. The other two, yeah. That was the.
I mean, they're the same. So.
What?
They're the same, so I guess that's the point.
Right, I think that was the point, yeah.
Jeremy, what you just suggested one of those like it.
The same ones like that. That's the point, right? They're the same.
Also, that's we started there. We only went so that's the wing of it might be a little bit.
Sure. Oh yeah, we have a fluorescein dye that's in a bag, and I see someone online asking about the water. Yeah, exactly. So we have this dye online to that we were using to. It's a fluorescent dye that we use to pump near the sponges to see if they are indeed circulating water and pumping, and we've used that on a couple dives, or a couple of sponges already. But yeah, this is a common approach that's used in shallow water sponges, and that dye is very easy to see circulate through the sponges when they're pumping. And we have we've been engineering a way of kind of an easy mechanical way to deploy it. We designed something that's kind of there's a sort of a bladder of the dye on the back, and then a hydraulic ram that kind of gently squeezes these two pieces of plastic together. Anyway, out the tip there, that's the tip of this bladder. The tubing that comes out from that bladder is attached to this end of the slurp there, and that's what you see diffusing out the tip of the wand there. So we don't have a way of closing that tubing easily right now, so we may be putting that on the drawing board before the next dive. just as a way to eliminate that. Are we putting that away?
We're going to use the suction, the plastic, the suction to pop the coral off the substrate.
Oh, okay. Oops, sorry.
Just gonna float away. The current, like, you won't be able to grab it, it's not gonna fall flat.
There it goes.
Oh, you don't
We're not slurping it.
You don't need to suction it
I'm
off.
just using the tip of the slurp to scrape the off at the base.
It's gonna fly off.
I think the current's gonna take that away immediately.
Yep.
Scrape it off.
I mean, you can turn the suction on and just, I don't know. I think
There's already a wad of sargassum and a coral clogged in the hose.
Yeah, this might be a better angle for it.
Okay.
Otherwise, you could have cut it.
Alright.
Yeah, yeah. We saw corn algae down. How deep was it? Do you remember it was like 270? It was
You see they're pumping, but
And we were like, nah.
none of them seemed
Crazy. It's what I said.
Couldn't-
I don't even remember.
couldn't see the poison go in and out, but I don't know if this is the current.
Another one that's behind the rock, but the rock was sideways.
Thanks for all the thanks for all your interest there in the chat, everyone. We always enjoy hearing from you guys and what you're interested in and what you guys find fascinating here. Of course, we're all here because we find it all very fascinating.
2.5 meters.
So, wait a minute, we gotta put this up, we gotta open up the door.
Oh,
Yep.
What's like the dial vibration, they just go from the shallows.
So like 2, yeah. So that's like 2,000. See, there's like 2 big. Down at the bottom. I'm trying to yeah, part of a fish went into that quiver. Yeah.
Part of the fish.
Yeah, there's a sliced fish on the quiver box.
How'd that happen?
I don't know. Sliced fish. What quiver was that?
3. The one that we just had open,
Yeah, that's 3.
or the one that we just opened for this.
Just open for this.
I don't know which quiver that was. I think it was 2.
Nicole, this is number 63.
Give
Seven,
me a
yeah.
second.
Oh,
Oh, and what's 7 things going at once? Okay.
yeah.
Nicole.
Okay, SH18637.
Really?
Yep.
Yeah, what we can do is
Alright.
pull it
I think those are later patches and that one on top of that is patches. I think
When you said
it's
it's really out.
patches.
Do you think the one in the top left is that stick patties?
But not all the way. It's almost like a full analysis access. It's like they make a full, these guys here,
Yeah.
it's like almost like still but they look a little different than the diaphragm.
There's only one species of later patties.
No, and it's got 6 rows.
Oh, this is
Part of
gonna
it
be a
is-
new one, or the same species.
So it's
Part of it was launched, you know, we'll be able to see right away. That will show how long.
This guy's stick pack is genetically sister to
And it's
So it's
quiver 2.
Big
Quiver 2 is going into.
Yep.
Yeah.
Or see where it's at.
Yeah, we know.
So Mike's getting the coral cutters positioned there near the base of this black coral.
That's the definition of genus.
He's gonna.
Oh, yeah, yeah.
Wait, so how many number of rows do the guys have?
The strength of the jaws are incredibly strong, but the ROV itself is very buoyant and it's easy for the force of pushing off of the rock or the base to move the ROV. So there's a challenge here with how to collect things like this that are.
I think it was an accident. Yeah, there is a fish that's in there. I'll show you when we get back to the
Yeah.
It wasn't part of the
Yeah, that's like a nice, really nice one.
little tiny one.
Nice.
That's the whole thing.
Masterfully done.
That's a what's the curve?
Oh, that's a nice picture. Associates works. I guess probably can't be better.
Can we take a screenshot?
Yeah, we'll just check. Drop down to.
Sam, can you take a screenshot at the moment? Here, maybe I can do this.
No, the top's in the center. So we can see like the rows of branches. So there might be some rows on the sides and the front.
Are those the worms you were talking about?
These are different. So these worms excrete something to make a tube.
Oh, okay.
Rather than modifying the corallum.
Oh, okay.
That's it.
So you can see the worms that grow up through the middle of the sort of central stalk.
Do they have the shot?
I think they do, yeah. You guys got the screenshot?
Yeah, I
Yeah,
got
sorry,
quite a
thank you.
few. That was really good.
All right. Let's stow that one, and then we'll sample this last one, and then hopefully have time to sneak a squeezer into that sponge. And, and then it's my bedtime.
Oh.
I mean, I'd be part of it. Certainly, I might do anti-cut because you don't pop channel. She got some Atlantic with mostly
Even the world, man.
she got something that might be hybrid. She includes all the anti-patties in their data set. You want to get some two disparate clusters on the first principal component analysis with genetic data. And there's some things that fall in between. So for cut and
There's one big species that's never been.
are they sort of bimodal? Bimodal? Species complexes?
I mean, it'd be worth
I know, I'm just longing to
Die.
do this at once and one
Fluorescing,
requires a
this
start
person
stop.
sponges to see, check.
No, sorry. DI, big guys.
I wish it was collecting it. Stop it. Stop.
Two rows of something on it. I haven't gotten there yet.
I'll say what that is.
Because Patney's is Greek and Bi, which one's Greek, Bi or
It's
Di?
certainly, yeah.
I think Di is Greek.
Let me say what's.
A lot of things are Greek. Patney's is Greek. So
Yeah.
in our word, it's Latin Greek.
A lot of black holes are made. I use Greek, which
Maybe a person likes Greek.
But do they use a Latin prefix, the Greek suffix with blank Patney's?
I mean, everything was Patney's. So some of them, some of them
You're
are
gonna
black.
like to fry, but you should do it. I don't know.
What is
Tim, I could just ask her to go to bed.
Okay.
If you do the squeezer, try to get a bit of it.
one.
You could
Oh,
never
of this.
of the tissue.
You go Greek for two is die.
I mean, you want
Tricky spot.
that osculum, yeah, it might be hard to.
Yeah, sorry.
I don't know. It's like we can rip the whole thing open. But
Yeah, maybe it's just a cool squeezer sponge.
I know.
Well, there's spines that form rows and columns.
We'll have more dives,
Called that.
more sponges.
Where like they form a shape.
Climate change.
Oh, right.
Sun will win.
In the Caribbean especially. Yeah.
So we're gonna leave behind
Sweet.
All right.
colonies for
Well, okay.
to explain all the excellent, yeah. So it'd be good to decide which ones we, give to them. Yeah, very cool.
So, Sandy, do you guys see SOLARIS? Sweet.
But they were negative.
No, one gave a little bit.
One gave us,
Four being toast.
oh, that's why she came together.
Is that all? Or we're gonna do one more?
You got the one more. I don't think we want the whole thing. I think that one's bigger, right? So we can just take a branch.
Yeah, we'll get the one that runs from.
Bring the trip.
So you could, yeah, kind of talk about how.
Are you gonna get that one?
Hey, we're good. We're been wiggling around a lot. We got into the water around 4.
Yeah, it's all like around 5. That was when Colleen's messing with me.
Okay, around 5. Yeah. Worked on Solaris all day long.
Yeah, that sounds.
We got it cleaned up. I mean, it seems to be working. There's not a lot of signals that we're measuring.
Can't use that quiver, so we're gonna do.
Can't use number 1. You can use number 4. Or
Yeah.
okay. I'm gonna try to sneak a squeezer into that sponge right there. After this.
One more than we
I
measured.
think that one in the.
Okay.
Unless you rearrange the order.
Uh,
One of them has that, adapter we made.
Oh yeah, which one was that? You put that in there. I don't know which one that was.
I guess it would be number 6.
Okay. I might try one of the others. Which ones do you think have the? I guess it's the 4 of the other 5 have the regular tubing on it.
Yeah.
I feel like this is a nice, pretty big hole. I don't know. We didn't see anything with Solaris. We don't think it's pumping. The fluorescein dye. You can't really see it, but we opened it. You can the green tube.
That's what the fluorescein is. Yeah.
Yeah, and it's sort of just leaking out. But we pumped it around a couple glass sponges and they weren't pumping. We didn't pump it around this one, but
You want to take a gamble?
I'm just gonna we'll pull the water in and see if the chemistry is different. This one, it'll be easy to like sneak the big tube in there, fire it, and know that okay, we got water from the inside. Whether it looks just like outside seawater or not, we don't know.
That's the question.
Yeah. You've got some ammonium sample. I mean, like that, like we might see ammonium in that, right? And eventually when you get enough that you want to measure the OPA, it'll be interesting to see if any of the squeezer samples have any ammonium in them.
I'll probably do a run either today, like this shift or next shift.
Okay. No big deal.
We'll get up there.
Snap right underneath.
Yeah, we've spent most of the dive down in below 200 meters, and it's just been like just all sponges, not the emergent kind of sponges.
All sponges all the time.
It was like, I don't know if I have any pictures, but it was just like sponge.
Well, if I can relieve you here, I can, but I don't think I can.
No, I think Santiago is about to take over.
I do it when we do the Solaris, and then I just put it in collector and just keep it off.
I guess
It's a pain to like.
yeah, it's like looks like that. That's what it's just like all one species of sponge, just but they're not like
if they're gonna
There's
do
nothing. There's not like
free
an
Solaris
upskill.
collection,
Yeah.
just do it during the free Solaris and leave it off for
Okay.
They're literally
I
just
should maybe
covering
get
the
one
bottom.
during Solaris.
Yeah, yeah, that's fine.
Yeah, that was one of the
It's
check
at the
valves
same
in
depth.
Solaris.
That looked like it's gunked up.
Yeah.
Do you know Professor James
Try and get it.
Moore?
Clean all those
Yeah,
out.
what I
there,
assumed,
yeah.
but it was gunked up.
Yeah.
He works with Fox Show.
Yeah,
Plug.
if they're gonna do, so it's on now.
Anyway.
I like
Yeah,
Fox
if they're
Show.
gonna do
Yep.
a squeezer,
Yeah, and they
get
have
the last one for this one.
That was the flow cell. We found some biological gunk in the flow cell.
I mean, between the ones that seem like a protein eaters.
Well, okay, for jellyfish.
Anyway,
Between, I waited.
so there
for
will be
everything.
hopefully at least one squeezer
Yeah, the larger
process
pore size is not too
comes off.
We might be, well, if
Potentially
nothing
more.
else, should stop with all these.
I'm not sure what our plan is. The Solaris, the wand. What was that?
It's my text was like a long.
Still the same rate. It's kind of
The Solaris wand, the filter tip on that got scraped off. So I don't think we're planning on using Solaris anymore. Oop. Yeah. Yeah.
Or was that a, oh, that was a ship.
All right, we are having a shift change here.
Oh, this is two. This is nice, sticking back.
Yeah. Although here it looks like there's a third row.
Nice, sticking back.
Well, it says that some of them, there's some branches that can occur.
Or what?
I'm
You want us to
reading
try
deeper.
the, try to do what?
The what? The floor seam on sponge?
Sometimes with a few,
You
it's
can't.
kind of
I think.
infrequently on the interior side of access.
I would like a squeezer if we could stick one of the squeezers with
I think
tubes
it's
on it.
Fine, let me submit it, let me submit it.
This is just such a nice little oscillator.
Which one?
The sponge.
But yeah,
What's the SH number for
No,
this
there's a
one?
big, it's hard to see right now. Yeah, you can see it here.
635.
635.
I don't want to get in his way.
Are we still calling it distichopathies?
Oh, that one.
Yeah. I mean, we measured it with Solaris and didn't see anything, but
It's like simple, not something.
Well, I don't know. Colleen and,
we could try to get a squeezer fired in there, that would be great, but
yeah,
I
I want that.
don't know how much time we have left in the dive without Solaris.
Well, trying.
You're gonna go deep?
Yeah.
Try to find, okay.
So we can try to find out sponges now.
Also, yeah. Yeah, that'd be
You
great.
guys want to put this in a bio box?
If, yeah, sorry, Biobox? Yeah. Guys?
Or, it won't fit.
Won't fit in quiver?
Question.
You got anything else in the bottom?
I mean, we could try to.
The apartments are full of style.
637.
For the testicle
No,
Is
35.
that a bonus? Is there supposed to do? Try to
Bonus
put in
sponge.
It would be best to put in the quiver. Yeah, so it'd be best to, we can try to fit in the quiver.
Okay, let me point out one more.
Yeah, right into that, yeah. Oh, yeah.
Yeah, I mean, that's the only one on here, but I don't know what
Yeah, we
the-
started off at 300 meters, you know, just, these were trying to come up, we started, we already got below down.
There's a little sponge zone before the
Thinking about
CCA.
dropping.
Right, yeah,
There's no corals there really.
right,
Down.
and it was not, yeah, it was,
Yeah, that's where we ended last night, but I wasn't sure how.
The previous
At
one was
300?
37.
Yeah,
Okay.
big 19.
yeah.
Wasn't sure how.
The first one we collected was 66.
No, I thought maybe
The second one we
added.
collected, 637.
Yeah.
Final one.
But
And we did
it's
see
only
the one big corral, corral, yeah, it was not, it was a big one.
If it sinks hot, if it sinks faster.
Let me, that note.
Okay, so I want to show you
Thanks.
how to make it. So this is that same Excel sheet we've been filling out. But then I made this tab, which will fill out say, two for every sample. When I do microscopy, I'll take a piece inside the side.
Copy it to the Jason for labels tab.
Pinwheels, I'll just
Once you
use
get
them.
that, make sure it fills out.
Yeah, it's
That looks right. For some reason,
I think the picture confused the desk we right.
Does it say it's been collected anywhere else?
No, I think the only thing is the
Yeah, it fills out
birthday.
two for,
Yeah, it has a, yeah.
and it's like
Because
10
it was only
samples so far.
a rent starting with 25.
Yeah, yeah. Good. So I can,
This one thing.
Remove things from this.
Yeah, I like this one now.
you didn't like it before the months.
Well, no.
No, deleting seemed to remove.
Yeah.
It's cool. Find exactly what it said. But I saved this format and then file that I hit the folder, so if you do delete it, screw it up, but I can find it.
What the?
It much more closely matches with this than the other.
Take over.
So are you serious?
Wait for the sample to come out?
Is processing? File it as you. File box.
Oh, yeah.
That's not everything.
Yes, come home, cry.
Where'd it go?
Export.
You stupid.
Hope you read it.
Oh, nice. I'll put it
So we've got
Alright.
another one that crumbles.
You have the clipboard here, Nicole?
Yeah.
Do you think it's all in the bio box?
You
can take your computer, I'm just gonna use yours as a bio box.
Same species. It's gonna look like panic.
You're getting out this one that we can match
Position this side.
situation.
So the first one we put 16.
Do we only have any?
I have not asked yet.
The second one we put a smaller piece that just.
No, I did not. He
When does he get?
was on the very, very, very beginning of the But it's very much everything, you know.
I'm gonna go see what the meters are. Sorry.
But, you know, I think
Sorry.
You guys don't have to worry about it. I'll do the samples. Once I get to try to.
Yeah.
Oh, yeah.
The basic plan was.
What quiver is this?
Make some fixes.
That was
There's so much science in that picture.
Looks like, yeah, fine.
We're gonna call it next to it.
Yeah, that's us leaving dried out. Look how shiny they look.
We're gonna lose that associate.
Thanks for, you guys working so hard today.
No issue. Should used to be red.
Stay attached.
Remember it was red. It goes down pretty far. I saw a picture.
Oh, look at that.
More recently.
Nice job.
This is
See Sophie or something?
I still have how much
I'm sure the people in the comments are happy that it escaped.
But there are also other little pieces. So like there's a big piece, you have to keep that whole one as the
Hasn't escaped yet.
a smaller piece, you keep that as the second one.
It's tough to
Pieces. Don't
Oh, nice move. He wants to be a part of science.
mess with those.
I don't think so, yeah.
Put them in like that. Second year, so you don't.
No. I wanted to talk to her.
Okay, who's in charge?
I am.
There you are. What's next after this? We already got 3 samples out of here.
Yeah, we want to drop down deep again. We got new waypoints. I don't know if you can enter them.
Also to cool off everything.
We pretty much started where we are, but deep.
Yeah. Rather than go
Waypoint
shallow.
21, 22, 23.
The 21st.
It was not 21st. I don't know. SH.
it's not a lot, it's not right there.
show up, and then I refreshed the page and it showed up.
Yeah, sometimes it has to refresh. I don't know why.
Thanks, Jess.
Oh, that's cool.
Yeah, she did my
I'll be back. I'm just taking yeah.
Anyways, yeah, we can, look it up once the session starts.
So do you think we can try to squeeze her into that sponge?
Sure.
Oh, we're just sitting here. Are we overheating? Yeah.
Oh. Are we?
Another minute or two. It's 59 right now. You want to try to squeeze it?
Let's try to squeeze her on that one.
Right into the same hole that we had SOLARIS.
Like Jira to this thing.
Okay. Jira's doing this thing. So, yeah, actually, let me see that piece of paper.
This?
Yeah. So, the squeezers are, well, they're not numbered exactly in this order, but you can see the numbers on them, I think, when they hold them up. This one in the front right, that says number six, has a 3D printed nozzle on it.
Oh,
We
fancy.
don't have, yeah, we don't, I would say we don't necessarily need to use that one. These other four here have a piece of Tygon tubing that sticks
Okay.
off of them. Those are probably gonna be easier to stick in and fire.
Okay.
Give it a shot. Don't waste a whole bunch of time.
Okay.
I know.
What's the purpose of the six you're
The
reserving
nozzle,
now for?
Kate was just testing it to see if we wanted to see if it would help them get into a sponge better with a 90-degree angle. What's that?
What?
He was asking what those waypoints are. Those are
Sorry.
your new waypoints.
Those are new waypoints where we want to go.
Oh, you got 20.
So we want to drop down deeper.
Okay. Anyway, use your judgment.
Okay.
Hopefully get a sample.
I
think I saw that.
All right.
So you haven't thinking any squeezers? Okay.
No, there's a lot of surveying, looking for stuff. And there was this zone where there was just not that kind of thing.
The white little sea.
Yeah.
Something like this. Did he show you this? Okay. So in here,
Yeah, we're gonna drop a bit deeper than that.
pictures for every sample within the SAT.
And you're looking for those
Yeah, those gave a pretty good signal yesterday on the previous diet.
But we don't have SOLARIS.
Colleen was saying that
Oh,
thanks, we can use it.
be careful.
Yeah.
Okay.
Well, that's what she said.
Let me
Double check with her.
We should double check, we should just communicate.
Oh, yeah. Yes, that's why we just chatted, but.
Yeah, it just took us a long time to figure out all the things that were stuck in the manifold. So I'm a little bit whatever she says. We haven't really seen much into it.
Oh, the sponges?
Well, superoxide. We haven't seen much.
I see, yeah.
It's all the ones we collected. And then open this.
Just for audio record, in this step zone, we've sampled 3 EDNA filters. These sample numbers are SH13544, SH13545, and SH13546. Pore size is 0.2, 1.2, and 5 microns.
Thank you, Luke. I think the idea is we can use SOLARIS for the Maricias. No?
Yeah.
Yeah.
Same. That's all right.
All right. And so we just aren't kind of don't want to put it into, sponges and places and poke it in a lot of places where we think we might get, we're just sort of trying to, not to be careful, like don't get too close. We can get close, but we don't want to run the risk of entraining a bunch of particulate. It's not like there's a lot of control over that, but.
I see.
Yeah, it's dispensable.
What?
Isn't it set in the water itself?
Well, I don't think it's.
Oh, no.
I mean, it doesn't. It looks like a.
Well, those are little creatures.
Huh?
Those are like zooplankton.
Yeah, those are big.
Santiago, does this look right to you?
Yeah.
You would go
Right.
every 20 and 21 and back over here.
Anyway.
Is it like worth it to
Yeah.
clean it?
That's right.
Okay.
Have you take that man pulled out and clean that?
Do you know what these
That's what crazy.
Yeah.
And yeah, I think we're probably gonna want to do it anyway.
Because it's already sterile.
We've already had
It's a
it.
cat thing.
With the
Should not already all be
I don't think we have spare tips.
That's
So we're gonna come up with
And I'll probably get this company for the real quick and then I'll be back.
You might have to hear some smaller piece or something.
I think we can do this quick.
Or come up with some other strategy. Might even be an entire new front end. Come off the one or something.
Picture.
Dream about it.
Apparently the sun has like a 3D printer nozzle with a 90-degree intercept or something. This one's have a tubing, so maybe one of those. Although, I mean, that was the easiest. And
You watch it.
And here we have. Yeah, that's for the sponge.
So it's the one for the tube's gonna be better if we're getting people
Yeah.
in the.
Okay. Alright, we're gonna go like
Six has apparently an attachment that makes it like 90-degree angle.
Yeah.
I don't know about that one.
If you're right too.
5. Pretty much all of them except for this one have a nozzle. Did you get what number is that? 3. Would you skim all through it all the way back now? Or 4, whichever is easiest.
I can relate it to we're just getting them.
Yellow.
We use 2 PSI to fill out whatever the
Oh.
it should be like
Oh.
this one is 3 years.
Oh, okay. That's whichever you want.
This one, right? Okay, that one, that one.
Oh, yeah. Oh, it's like that. Yeah, I mean, whichever. Either one.
So, Nicole, one, this was
That's the guy. That's their numbers. Okay.
what is this number? Right, for both cameras. That's 16. So you only measured 2 of the corals you collected?
That's what I have. This is fine.
Okay.
And I think you guys, I just made my notes.
Okay.
So we didn't measure all of these different.
Maybe they were.
3 separate difference. Maybe really fast between.
Maybe it's just, maybe 2 are lumped into one.
More light on there, is it just
Okay. So I have you down for 8 and 9.
I only have it set for the high PI. Yes.
Okay, so that must include 38 then too.
You wanted to squeeze close to the hole, right?
Yeah, stick the nozzle in the hole
So then,
as much as
so
possible.
the next number is 18638. Okay.
Was that snap?
Oh, yeah.
Why? Tired.
That's a filthy
Have you been saving the frame grabs?
Yeah, for the samples. Yeah, I got them all.
Oh,
Even the coral we didn't bank, and the one that we got extra, the one that we this opportunity, the one that jumped in the back of the biolox.
Nice.
When J Rod tried to catch the. Look at that thing. Look at squid, the way it's moving.
Oh, it's an octopus.
It's an octopus.
A little bit.
I'm saying, are you saving directly to the server?
I'll put it in a folder. I did have permissions. Janelle got me permissions, and I've been all the ones that I did are now on the.
No, I know. I was just curious
server.
how you were saving them because I had to save them on my desktop first.
Alright.
Oh, is that what you're saying about second Oscar?
Yeah, is there any, like, what if? But as soon as
That's pretty big for a city. It's so much.
I've seen it like big.
What? So much.
See this bramble colony?
Yeah. Bramble, that's planar.
What's the red dimensional?
He's inside some tables.
It's easier to see on that screen.
Well-
Never mind. Maybe this one. Is it?
Okay, he's inside. So squeeze.
Yes, please.
Squeeze.
Whoa.
That's right. Speed it up.
Sponge spit it out.
I got a little too excited with that.
Yeah.
Yeah.
Squeeze out.
Punch me back.
Squeeze though.
It squeeze.
It did?
Yeah. Got your sample.
All right.
Okay, what was the squeezer number?
3. Squeezer number 3 in a sponge.
Wait, let me get a.
Okay, are you happy to do that, or you should have a good view? Or I could porch out.
Might be the queen snacker. Not sure.
Yes, yes. Your hose is on
I'm sorry.
Currents are to the southwest at the surface.
Where we are. Yeah, we're at 184 meters, and the current are, yeah, coming from the northeast. But they don't seem very strong at this depth.
Half
Just
a knot.
consistent. Not much less would be the blue one.
Oh, sorry. Yeah.
So they are like 0.1 knots. It's consistent with what we see right now. Doesn't seem to be.
Yeah, it doesn't seem like there's any prevailing
Yeah, not a lot of current.
strong current.
Check the tides. Yeah, so the last high tide was at 9:30 p.m. local. So the next low tide is gonna be at 4 a.m. local. So we're in the slack period.
So it should pick up pretty soon though as it drops.
I'll say probably in a couple hours.
Does it have a let's show the current. And look at windy.
So the squeezer fire going away, right?
Yeah. What are we going next after this one since it sets us down?
20. It's down there. It's deeper, so we had to.
You want to head down there as fast as we can, or you want to
Yeah.
down long way and down?
No, as fast as you can. You can do like a quick off bottom movement with the ship. So after this, we stow away this squeezer, we will shift gears and drop deeper to where we're seeing our target corals, a couple yellow Parmericias, and some other corals we expected to find around 300, but we encountered this zone dominated by sponges and climb all the way up here to mesophotic. So we're momentarily gonna be lifting off bottom and dropping down, so we'll probably see just black water for a few minutes while we move to a deeper area on this ridge.
It's better if you just move it up first and see where that thing actually goes. Actually, it's gonna turn. There you go.
Yeah, you could then, that's what you save it, that puts probably make a filter, like, what would make a filter, like, purchase and make them, that data, updated data.
Right now, I don't.
Yes, I just same here.
So I would just.
I was thinking it'd probably be
Yeah, it should be alright.
okay there, right? It's not gonna go anywhere.
I'll just do it.
When we pick the next one, the middle one.
It might fall.
Yeah, but
Yeah.
at that time, anyway, we could put this in the front quiver and then we'll fix this one on the next piece.
I see, yeah.
Juliana, there's a sample of I need to share some.
Oh, you got it.
Beautiful.
Nice.
Just all parts with no more.
All right, so the
Shoulder left.
squeezer number 3 is secured. And now we can do our move.
Once he's secured. I'm gonna pour in. We'll do that.
About what that would reach something?
290.
Okay, we are gonna move the ship.
To that waypoint 20.
Wait, what's one? Okay.
It's 370 yards, 380 yards, not meters. So. Bridge RV. Yes, you see the new waypoint? It's about 380 meters south, southwest.
I see that, yeah. Fantastic. What speed would you like to get at?
0.3.
0.3? Okay.
Roger. We'll just follow you.
Great, thank you.
God.
Good. We just did a squeezer and we're gonna move to that waypoint 20.
Okay.
That's gonna be like about 390 meters.
Oh, okay.
So we're gonna go back down to look for the primaries.
I looked at the tanks, Nella filled them up, so.
Yeah, I checked them out earlier.
Same problem?
Temperature looks good.
You need to log out on that. Okay.
And we have 4 bio boxes open. So that should be good number.
Yeah, 4 sounds manageable.
I'm sorry?
4 sounds manageable.
Yeah.
You collecting these for the aquarium?
Yeah.
Nice.
Yeah, that's the goal.
Aquarium, try to.
Oh, thank you.
Yep.
I'm gonna go get a coffee. Anybody want it?
I'm good, thanks. I just had a,
Jeremy,
just had a red eye.
Okay. You what?
I had a red eye.
What's a red eye?
Coffee with espresso in it.
Nice.
I know Juliana.
Mike.
Mike. I'm Anne.
Yeah. Nice to meet you, Ann.
I was just thinking of you as like the nice guy who brought Snickers the one day. Mike's the one who has the cool accent.
It's not often called a cool accent.
Really?
No.
I think to Americans it is.
No, thank you. I just think it's better. Just like
So it's stuff that, I mean, we talked to them about what they collect. Santiago and I talked to them about what they collect. So then I talked about what they the other stuff that is.
well, I'll do it, yeah, I'll just do it. It's just, just, I guess it's just in general, it's just good to have. Even if we're just like, okay. That makes me, I should have just. It's okay.
What is your discipline, Ann? What is your discipline, Ann? You got some time to get to know each other. What is your discipline?
Oh, sorry, I'm a biologist. I mostly do molecular biology and animal physiology, so I tend to look at like how the animals respond to different stresses.
Who?
Yeah, so I do some work with sea anemones and some work with plankton, the copepods. I used to work on corals a long time ago, so I'm definitely back doing this. Yeah. I was, I worked with Santiago when he was a student.
Oh, really?
Yeah. Back in the day.
Back in the day. That was a long time ago.
Yeah. It's true. He was a good student.
I used to go to Huli a lot.
Yeah.
Oh, that's with Harbor Branch.
Oh, yeah.
The good old days.
It's not a bad place.
No, that's we have a great time over there.
Yeah, I've been there 21 years now.
21 years.
Yep.
How when there when you were there? No, just kidding. Remember a few weeks I'm sorry to say.
Where does the ship go after Puerto?
Okay. Costa Rica.
Something runs like every 10 minutes out of the website. I'm just trying to figure out what that is. Sure, that's it. access, you probably need to walk back in.
Yeah, I almost went for a movie a couple times. I just couldn't swing the time. You know, Phil Santos, Alvin,
No, I don't think so.
he's not with them anymore. Have you sailed, I'm sure you have, sailed with Alvin?
No, I've never done deep sea work really. I was on the Pisces once or twice, so otherwise I haven't done any.
With, what's his name? Who's the head guy in Hawaii, in the Earl, the Pisces group?
Well, I don't know who it is now. Did you know Chris Taylor?
Should I review it?
He was at Hui for a long time.
Sounds
Doing
familiar.
ROVs.
I didn't know much of the ROV guys. I knew most of the sub guys because I used to use subtitle.
Did you know Phil Ford?
It's been a while since I've been
Yeah,
there.
see, I only know a few of them.
Yeah, I'm still like trying to figure out what's on time, exact dimensions. So just keep on letting me know. I'll keep on letting you know.
Desecheo. So, I'm renaming the genus name. It's obviously varies what we've been calling things. Should I just do the genus for everything? No SP, or those species that we've added?
No, I think if we have species added,
Okay.
speed. If it's not identified species, just the genus is fine.
And underscore species name. Right.
Sure, that'll be great.
Okay.
Yeah, if we have somebody who knows the species.
Right. So I'm not gonna figure that out.
We're gonna take advantage of it.
The other thing is, should I update names now or no? Because that database would be different. Should I like just go on the Excel sheet that we've been adding to, and then later, when we make a change, update everything?
However you prefer, I think.
Well, just if I change the name on the pictures now, you may have trouble matching.
Oh, I see. Yeah.
It'll
Keep it to
different in
what.
the Excel.
I mean, as long as they have the SH number, it'll be fine. Because it just would be good to know which one is the most up to date.
So maybe I'll just change the Excel also.
Yeah. Thank you.
We'll go up to 20. It's 390 meters. This wants to get there. Ouch.
Okay. So you got it. I got time to.
Yeah.
One more technique.
So we're almost halfway there.
Yep.
It should be only a few more minutes until we get to our target waypoint.
So when we get there, are we just continuing general collections or getting stuff specifically for experiments?
Both.
Yeah.
Yeah, so we have a couple more of those, a few more of the primaries, yeah, with experiments. It'd be good to get one more of the purple primary seed that we saw yesterday and see what else is there. I expect to see a bright purple, kind of like primary seed, same family. Similar color to the Mauricia that we see in the Shala, but it would be different species. See what else we see.
Yes. They were calling this was eyelast.
Yeah. Yes, it's not show.
So it's not around.
Yeah, they collected two samples.
How do you tell those apart?
Well, they probably be pretty good so you can see the qualities.
They grow so similar.
They do.
I'm glad I don't see them in my media.
Why is it blue now? But why is it that blue now? It's pink, right? Or is that a different color?
I was just saying, don't you have some national waypoint? I don't like it.
Oh, what?
You could put any color. It's just easier on, I guess, visual.
Yeah.
Yeah, sometimes you forget.
What's the little puffer fish? See that Rico? That flower out nice. Can't the gas burgers.
Looking at the sample image?
Yeah.
Yeah, I was gonna do that too.
I think it becomes pink when it mixes with the red, right? It's within the inside the circle. Then it'll turn pink.
That's the one we were sampling, right? Distichopathes?
Yeah. And we sampled some.
That circle is a circle of death.
I think these are actually a later package.
You don't want to be that close.
Yeah. This is a good, that we're not really sure, but I think these are a later package. And then this is Distichopathes. You can tell the difference there. There's an Oleda package right there. See? Distichopathes just has two rows of pools.
So it's still dying?
Yeah. This was recently, this was described in 2020 in the Flower Garden Banks.
Oh, really?
It could be the first record for Puerto Rico. Yeah, I mean, it could be the first record anywhere besides where it was like described.
Yeah, I mean in the paper for sure. It was only from the Gulf of Mexico.
Yeah, it's from Elder's Bank at like 170 meters.
That's cool. How we collected it?
No, I don't think so.
Yeah.
I think Emma Hickerson.
Although that's one of the samples.
Her, it's named after her.
It's
Oh, really?
Yeah, it's Distichopathes.
where like I could
Oh, that's super cool.
from the same.
Yeah.
For that sample you can come on.
There you go. Alright, so for that sample just.
I don't think I've actually seen it now that I have seen one up close. I think I only see Oledopathes.
Because you
Yeah.
probably want the, it looked like they got almost the base.
They got the
So you
baseline.
probably want that, right?
It's cool. The base is good to have.
Yeah.
But you can like cut a whole branch
Yeah,
off
yeah.
and chop it up. You can even keep one alive. And I don't think no, but you can.
I'm not sure if we should keep everything alive, but we can cut.
I will. You did it. This
Yeah.
is the first time.
Yeah.
I don't think I can open anything else. you open the
succeed
files
this
from
time, but we're going to next.
You
These guys have
download
done
it,
it before,
or-
we just,
I'm on the
yeah,
server.
they do well, so we have a few ideas about what we're
Do you want me to
so we're gonna try that.
try and download it to my
We
computer?
take to temperature, we check.
Man, I'm
There's
just.
like a cooling
Oh,
coil in the tank that's a
the-
limit. We have it
I was like, I can't open anything.
Oh yeah, I can open it. I'm just reading
Yeah,
it.
so those are two of the things that we're doing.
So I figured out who's there. They're not on the back, sort of thing.
Oh, we don't know, like, oh, like.
Yeah, this is super useful.
Yeah,
Right.
well, the thing is, like, when we have them in the tank, because the
What's
polyps
interesting
are
is
really small,
on
it's hard
both
to see here what's going on. So,
dives that we've kind of
you know, we're kind of looking
traversed,
at it.
that's,
It doesn't really look good.
seems
So
like
we're
there's
kind of
this
like
area, just, I guess that there's this depth band below where we start to see CCA that's dominated by sponges with very few corals.
Yeah.
Well, so now it's 14.
The zonation.
Because we're getting corals from shallow waters. So
I,
actually,
they're so dense that makes me think that they actually outcompete
Yeah.
the corals. Unless there's some environmental
Well, it
parameter.
could be both. They might be outcompeting the corals because of that environmental gradient, whatever it is.
list for us. So, yeah, it was crazy.
I don't know how to-
It's right on the edge of where there would be any life penetration.
I don't have the full. Didn't you take a picture of the CT, the deep one, the other day?
No, I didn't take a picture of the profile,
No.
but we have the data. We can plot it.
I thought somebody did.
Oh, maybe Scott did.
But like share it with the group, or
I didn't.
or
If they did, I missed it.
you got the profile that I sent
Yes, I did.
yesterday, right?
Yeah, I got that information. Put it in the wrong.
Oh, it's almost International Earth Day, apparently. I didn't know that.
Earth Days today?
Tomorrow.
Tomorrow. What day?
22nd, 32nd.
Oh.
Yeah, I got this story about the now. Got an alert on my watch about Earth Day.
Oh, nice.
Earth Day, yeah. So I posted it, and then my watch was just dead ahead preparing. Gotta get ready.
Earth Day Eve.
Earth Day Eve. Earth Day here.
I think we should get cake for that.
Cake, yeah.
Wait, we're saying Sam's birthday. Isn't it soon?
Maybe for Earth Day, then we could all eat vegan food.
Sam's birthday?
It's the 26th.
Colleen's birthday is
Is on Earth Day.
Sunday. Or Earth Day? What day is today?
Friday.
Today's the day before Earth Day
Well,
now.
it just turned Friday.
Yeah, so her
Also
birthday's
birthday's
like tomorrow, like the
Saturday.
real tomorrow.
Yeah.
Real tomorrow.
And there will be cake.
Yeah, it's true.
Yeah, more birthdays, more cake. That chocolate cake, the first one was like the best chocolate cake I've ever had.
It was pretty good.
Woo! That's like that smoke signal from the rescue West Coast.
Oh, like the flares?
Yeah.
Yeah.
Official wireless down here.
The little rescue ROV comes up. Sebastian, you need help?
Now, Hawaii ROV actually needed help.
Recently?
I was there for that.
Oh yeah.
Did you get tangled or like just stuck in the
Yeah.
car?
I only saw when the feather of Hercules got snapped and then Jason went to rescue it. That was cool. They did it, the broadcast live, the rescue, that was really cool.
Blue kind of was rescued by the petrol. Too deep for Sebastian.
Oh really? What deep? How deep was it?
4,800 meters.
Oh,
What's the depth rating of Sebastian?
4,500.
4,500. Okay.
I think within the ROV team, probably everybody has rescued an ROV.
Oh, really? I have seen ROVs go down and never come back.
Oh yeah.
Were you there
Goodness,
when
I don't have that.
Dario got lost?
Huh?
Were you there when Dario got lost?
Yeah.
Company I used to work for, one of the ROVs got severed and floated away and then turned up on a beach.
On a beach?
Miles and miles away. Yeah.
Oh.
That's an easy
Floated
recovery.
all the
I
way.
guess.
Like lost for months.
Did just a random person find it, or you guys?
Yeah, there's a
Somebody's-
number on the side of it. If I recall.
What a pretty exciting fact.
That's not good. That's a cheap recovery, I guess, compared to going down again.
Right. Just wait for it. Oh wow. Good thing it was buoyant.
It's not just.
The sampler doing okay?
It's up.
Oh, let me check. Seems to be.
Keep talking. Keep talking.
I can't tell why I'm not changing. I think. Yeah,
Good.
390. Okay. First dive was 800. I don't know exactly when I couldn't talk to it, so we'll have to see.
I think it was good to have the continuous series.
Yeah. Yeah, I hadn't started on that dive just because I didn't want to fill the file up with zeros, but I see your point.
Well, you just easily delete them later.
Yeah, I know. Oh, I didn't know how big the files would be, then I realized they're very small.
Yeah, it's just text file.
So I'll put it.
Ever seen ROV rescue man stuff?
No.
Oh, that sounds more scary.
They're trying to recruit me for that job for the man's out. No way. I know better.
It's not gonna happen.
So really nice sea stuff. They're perfect.
Sure enough, they got in trouble. They put a torque tube on a man, so and they ran out of power. The torque tube wouldn't open.
So we're stuck on.
Yeah, I had an emergency call. Man, sub stuck with a torque tool.
Yikes. Well, I see one of the yellow Mauritius there.
Oh, yeah.
The turtle, you said?
What do you need a man so to be taken away?
They were trying to see if the man can do
Yeah,
all
every
good work.
time we go through
Which,
this, we try
I
this
said,
thing.
first rule of man sub operation, never go fly underneath.
I'll let you know.
That's why he's taking it.
And he
Or
flow into
hard to, it's hard to see what is actually running in the background to
valve
change conditions.
of your knee, and ran out of your knee, they can figure it out.
Perfect.
There's a Cairo FT all the way down here.
What temperature did we set the room?
14.
14.
What temperature is?
15.
I think that's okay.
Yeah.
Wasn't it 14 yesterday? Here? Might change a little bit.
Was it?
I
Can't
think it was
remember.
a little colder, yeah. It's gotten a little warm. Maybe with
Yeah,
the
I'm sure there's
tides.
some fluctuation with the tides.
Yeah.
so.
Oh, yeah.
We can, I mean, if we want, I guess when we start collections, I could ask Dan to, or whoever's awake or standing there, I could ask him to change the room. I don't think we have to change the water.
Yeah, right.
Then it'll graduate.
July I might be able to ask one of the whoever engineer is
Oh, yeah.
on watch now.
Yeah. Is the temperature up on one of those screens or is it just on your?
I see it on the sea
Okay,
log.
yeah, yeah, okay.
It's in one of those tabs, but they need
Yeah.
navigation.
There's like so much to look at, just making sure.
Yeah, there's a number somewhere.
It's what? Oh, yeah.
Alright, so we're at the seafloor. close to waypoint 20.
Oh, it's break. Go slow. You take off. Or out.
So we're looking for
Just check the waypoint 20.
Getting deep. Come on.
Yeah, it looks like that 392.
So we're looking for this coral, the yellowish one. Yellow. Yellow fan. Is that a line or is that a crack? Really?
Take off. He's gonna be back. That's all I need to say. I got really hard too.
Wow, this is strange fracture lines there. There's one of those yellows. Uh-huh.
We're starting to see more of those whips we saw on the first dive.
The Elisella?
Yeah, the one we're not, that looks like a bamboo, but it's kind of,
Yeah.
might be worth looking at too.
What's all that?
That looks
Like a brisingid or a crinoid?
yeah, it's hard to tell.
It looks too feathery to be a brisingid. Yeah.
Just have like a thick
You
Yeah.
want to get the yellow first or the what?
Yeah, the yellow. Doesn't seem to be that much current here, or a little bit, huh? So that's coming from the northwest.
So it could just be wrapped around the rocks.
Right, yeah. Because the near surface current is coming from the northeast, but so general direction north-ish.
This coral seems to have hermit crabs every time you see it.
So that's actually good because we can go with SOLARIS. Current we're downstream from the current.
Yeah. There's also some corals in the crevice. I don't know if you want to test for ROS in the crevice.
Yeah, that's a good idea.
Just be careful with the tip.
This looks good for the species we.
Did you take a finger up?
Okay, I'm collecting the background.
We're gonna sample this one, right?
Beauty. Yeah.
Okay.
Oh, you know what's? See that coral behind it, that yellow and white?
Yeah.
What do you think it is?
We don't really know.
We've collected it a lot. I don't think we've ever had someone give us a name for it.
I didn't quite hear what you said, Jeremy. Did you say we need to fix them?
No, just IP.
Yeah, yeah. Right down. Okay. Yeah, yeah.
So there's 2, 3, I guess 2 hermit crabs on it. Did we, for the other primary that we collected, did we get the hermit crabs?
We got hermit crabs from this one. Maybe not.
Did they come up?
The snake came.
That's now.
The home came.
Oh,
The
just
home?
the shell. Just the
Yeah,
shell.
the shell.
I lost
Not the crab?
I did see something on diving.
What?
Abandoned ship.
I don't know.
Are we ready to sample?
You see the crab?
I don't remember.
I'm sorry?
Are we ready to sample the coral?
Yeah, I think we're getting
Okay, y'all ready to sample this?
some image. Are you ready with the Solaris?
Ready.
Okay, we can carefully approach the coral with Solaris.
11?
So that, I don't know if you heard, were you here when that happened? The tip of the one of Solaris got shaved off?
Today?
Yeah, so you know how it had that little cap with pores? That part got shaved off, so it doesn't have that protective tip that prevents big particles going in. So we want to be extra careful
Okay.
not to get anything but seawater in there.
The yellow one, it's gonna be on our side.
Do we know what this coral's called?
Paramurisia.
E-R-A-M-R-A.
Coming in hot, man. Maybe I'll put that thing in a high thermal bit. That was the last one.
Yeah. This one will melt.
Is that better? Still got a little bit of go and sit. Just be careful that current.
Nothing yet from Solaris. We'll still have some distance to go.
Olives are open. For sure. Okay.
Looks like you're very close. Yeah. Hold it. No signal? Just, yeah, hold it for a little bit. Nothing, nothing.
No signal? No, no signal.
Maybe try.
It's going up.
Maybe
Sometimes they have left moving to the other side of the coral earlier. I don't know.
on the others, like behind.
Yeah, like behind it.
Yeah, we can try that.
Nice depending where the current is, I guess.
Huh? I'm sorry?
The polyps are really nice next to
Mm-hmm. See if that works. Yeah, right there. Yeah, just hold that. See what happens. Wait a minute. Oh. Oh. Oh.
Maybe a tiny blip.
Yeah.
Okay, yeah, no, I don't think.
No signal. Okay. Should we try another part of the colony, or you think?
Are there other species there?
Wow, that's diving down.
Yeah, we can try something else.
Should go in the hole.
Yeah, I guess let's go in the hole.
In the crevice, there's.
Where do you want to go?
I don't know what you're talking about.
So underneath in the overhang, there's
Yeah.
some corals inside there.
I think it's very risky for the probe. I don't want to risk the probe. Do you think we should try another part of this colony? Or you think
Yeah,
we're...
try push from the basement.
Yeah, maybe let's try
Yeah, try that.
kind of this part.
Zoom out, but. Doing the same thing?
Yes.
Yeah.
I'm dead. Sorry.
That's like what we had in our proposal to try different parts of the colony, because we don't know if like the tip's pretty small or.
This is event. What event number is this? 12. 12?
First one was 11.
First, which was third one?
that part of the colony.
Oh, these are different events? Okay. So these primary series events 11 and 12.
Keep it event 11.
11? Okay. This primary is event, Solaris event 11.
Pops are partially closed.
Anything there?
Nothing?
Another reason, like, it's not so much this colony, but like, there could be a coral where like part of it was recently grazed or something. There could be like wound healing or something.
Yeah, nothing here.
Okay. I guess that's no signal coral. I think we can
Okay, turning off the pumps.
turn off the pump of Solaris and we can put the one away, stow it away, and then get ready to take a sample of this coral. We want it to go in a bio box. Which bio box are available? Or rather, which ones are full?
So only biobox 1A and B have something in them.
Okay, so 2 and 3 are
2 and 3 are
available. Thank you. So the depth is 292 meters. And the code for next sample is SH
18638.
18638. Right.
Is the temperature still about 50?
Yeah, temperature is 15 degrees.
We call that Pararisia.
That's right. This is Paramarisia. Looks like it's small enough that, yeah. Interesting, because we measured this species yesterday, and we made pretty strong signal.
Do you think it's just to do with the current maybe? I feel like that's been playing a big role.
Possibly.
Another thing people were guessing, you guys remember this, yesterday they said that we started seeing measurements once the sun came up.
That's what we were kind of thinking too, but I feel like we haven't been around in the day enough to know. But
Yeah. We might get a chance, we'll see.
yeah, that would be nice.
Yeah, that's true. We'll see the transition.
Bring that rail in too to get you closer.
Sunrise should be within the hour, right?
No, sorry, it's only 1 o'clock.
Oh, I was looking at UTC. Yeah, so now we have a.
I mean, somewhere.
Somewhere, yes. In the UK.
When you said that, I'm like, I have no idea what time it is.
And I was just looking at the UTC clock.
Just nice and easy then.
Feels like it should be coming out for us, right?
Can you get there? I don't think you can. That's pretty far.
Try repositioning.
Yeah, I'll just reposition.
That's an arm of a crinoid. Oh, you see it? It's swimming. You see it on the bottom camera?
Oh yeah.
Oh, that's cool.
Sure. Those are awesome. Whoa.
So cool.
Now it's just gliding. Whoa. No. Might see it later.
It's kind of amazing that they decide to do that when they can't really see. Like, they're just like, oh, I'll just like swim somewhere. Maybe
Just
live.
trust that I'll end up
Land
in a good
somewhere.
place, right?
Yeah.
Because like once it starts sticking up, like how do they control?
That's what I mean. I mean, I'm sure they could probably get other place sense.
Oh, yeah.
I don't know if they can control buoyancy.
But
or
definitely
not.
tell me, like, don't try.
Oh, no.
Grenade, that's the grenade, right?
Grenade.
I call it that.
Yeah, I see that. When they close, yeah.
Did you guys see that on the left shift? Yeah, I was here. Yeah. I saw that on YouTube. When I was like that.
That's a little bit better, huh?
Yeah. Starting to get it.
Where are the hermit crabs? Oh, they're still there. There's one, I think.
Let's see a couple of shrimp. Yeah, I don't know if the hermits are still there.
They might have left. It's interesting. Oh yeah, there's one on the...
On the rock, yeah. They're like,
They left when the polyps closed.
Yeah, I guess see what you're doing.
Maybe.
Bye bye.
I bet they like it when the polyps are open. They steal food.
No, I'm backing off. Back in there. Just tuck your own back a little bit more. That's good. Sorry, I know I'm making you also sick with that.
That's a good screenshot.
Yeah.
Maybe Steve Cans. It's really weird.
It's kind of a half inch from the base.
Wow. Look instead, you're turning close. A little bit, I'm pushing back. Like this time.
Got it.
Sucker your arm up, just in that. For the aquarium.
And everything.
You said buy box 2 or 3, right?
Yes.
There are.
Yeah.
There were people asking.
Let's just go ahead and go to the furthest one then, and then buy box 2A.
2A. Sounds good.
Buy box 2 coming up.
Maybe it'll close or not. Give it a try.
Now it's fine.
Perfect. All right. Great sample. In biobox 2A, that's Paramaricia, SH18638.
They want to take a look at the whip sound yet? To the right.
A nice Stella whip. Oh yeah, there was a whip on the
Yeah.
right.
Pretty big one.
The one that had the crinoid that
Like
flew.
real good zoomed in or just a quick look?
Yeah, we can get a good zoom.
We haven't tried SOLARIS. I don't know if you want to.
Let's see how feasible that will be.
Polyps look like they close.
Yeah, the polyps look close, huh?
Yeah, they're close to.
So if it's closed, you don't want to mess with it?
Yeah, I think not. We've seen that the corals with open polyps are most likely to produce any signal.
No.
I think you got scared.
Crinoid.
Crinoid got scared too.
Might have been all touching.
All right, that's great. Thanks.
Alright, you want us to, let us know a bit more? Okay.
Keep looking around for those yellows and see what else we see.
You want more of those yellows?
Yes. I think we want to get the 4 spots to the bio box for those. You know if we got an idea on those funnel base sponges? No?
It's up to Alan.
I'm sorry?
Does Alan?
We sent that to Alan and he replied, but I think he replied to another sponge.
You might have been referring to both.
To both?
Ferrede. I don't know.
It is a glass sponge, but
It might be a family of different, a lot of different morphologies.
maybe, yeah.
More whips over there.
Oh yeah.
Can you click it in just a little bit more? I don't want that. It's got the tip sticking straight out.
Yeah, there's a couple of Elisella whips here.
Those whips look black coralish.
You think so?
Yeah, just bring the tip down, just right above the, yeah, no, up a little more. Yeah, I want to bring it above the bumper so the bumper protects it. Come up, come up.
Just outside it.
Here, I'll bring that bumper out. Bring it up a little more.
Inside or
There,
outside?
there, you got it, just like that. Sorry. Alright, so just keep going towards 20 or have a look around this area?
Yeah, just look around this area. There's nothing special about waypoint 20. Just a reference point for this area.
Something on the. Something what? Something on the
What's a crinoid right here?
Oh, this might be one of those purple fans. Can we take a closer look at this one? Right next to that yellow sponge. Oh, interesting. I'm not sure that's the same one.
I think it is.
You think it is?
Yeah. I'm pretty sure it is. You want to see a picture?
Yeah, I have a picture here. Oh, I think you're right because we're just looking at it from the end that has the polyps.
Yeah.
Yesterday we were looking at it from the end that had no polyps.
Oh, there's a pycnogonid.
Big creepy spider.
Oh, a big nogoni there, yeah.
And there's an apocopher too.
Yes, there's an aplocophora and a couple of snails. Yeah, this is one of the ones we want to sample.
You want to sample or?
Yeah, yes.
So set up for samples?
Yes, please.
You see, it's like a sea spider. Yeah, it's so cool.
There might be a thesia above there. Since you have a good formation.
Set up for SOLARIS, but we don't have to know before.
Thesia.
Yeah, that might be the real one. It looks gorgonidae to me. Just gives off those vibes.
Gives gorgonidae vibes.
Yes.
Can we try measuring there, just because it looks pretty protected? Just wondering.
Yeah, sure.
It's a glass sponge.
We can also try the fluorescein. Where
I'm trying to come down.
do you think would be the best position if we were to use the fluorescein for this one? To see if it comes out of the vascular?
I don't know what the glass
They suck up, or they filter.
I don't know how the glass, I don't know how that thing is.
Me neither. But we can try.
Yeah.
Got it.
And the sponges would
Yeah, I think I'm gonna target this one first, and then there's interest in trying the dye, the green dye on these sponges that are in this little cave. And hopefully trying to measure SOLARIS. I don't know how easy it will be to get the arm in there.
Oh, you want to get SOLARIS in that as well?
Yeah, but we can start with the corals.
I might as well get set up for all that then.
Yeah, okay.
Because this is not a fun stuff to set up. Might as well get done. It's pretty cool.
Oh, that's another crinoid flying.
You get scared off the other one?
I might have touched
Oh,
it.
the arm is that a separate arm?
Oh yeah.
Bye bye. It's crazy when you sample them how easily they fall apart.
Don't know how we're going to get SOLARIS in there, but we will try.
That's what I was thinking. Oh, that's a cool crab in that sponge.
Good God.
Fast crash.
There's a squid there too. It's like a dark.
It's pretty interesting
Squid.
when we're working like in both below the photic zone and
I'm sorry?
it's interesting where we're working because like the transition is really stark between the mesophotic and down here, but it's not very far away.
No?
We keep going back and forth to change the way I'm perceiving things.
Yeah, just, it's gonna, might be a little bit
think that's easy on top, I'm pretty sure.
I guess for you, Luke, if there are species that occur in both, like 150 and 300 meters, where
Yeah.
the polyps are different,
I think we're stable.
you know, almost
Yeah.
the same species.
Yeah. Have you seen any species that occur in both of
Is that
those?
what
Are you, how is Solaris?
Real quick, we'll start doing the background now.
I'm gonna go back up.
Yeah, we can probe, but
I think we're gonna try
we're doing the background, we'll just hold on a minute.
coral
Right?
and
Okay. Coral first.
Yeah, we can try the coral for, what do you think would be easiest?
Coral first.
Coral first.
Anything interesting around here? The coral.
There's a cup coral, yeah.
Hey J-Rod, can we get a, when you're done with that one, can we get a good picture of that coral on top? The, has like 5 arms to the left. Sorry. It's hard to describe this, but.
Yeah, it has like thin branches.
Yeah.
That look good?
Yeah, that one. Get a good picture.
Oh,
Not this one, sorry. We're just taking a picture while you get back on. Highlights. Cool, thanks.
There you go. You wanna
That's
see the
an
po-
interesting
you
one.
wanna see the polyps a little bit better?
Sure, yeah, yeah.
Yeah, if you can. Not sure how far anyway. That's great. Yeah, I think that's a Thesia.
Thesia?
I think so.
We could grab it, just for diversity. Solaris is ready.
Solaris is ready then.
So this will be event 12, correct?
Benjamin.
Okay.
This one, right? What side do you want to start on first?
Can we see, I think, the side closest to us inboard? Because that's where the polyps are.
Polyps are out.
Polyps are out, yep.
Good, good sit. Zoom it in a little bit.
Whoa.
Try to touch it.
Yeah.
Nothing.
I can't tell what all is on that. Is it a hermit crab and some others?
Yeah, it's a hermit. There's like,
So those are
that's the Plecophoron.
Oh, okay.
There's 2 of them.
Yeah, yeah.
1,
3.
2.
There's on the bottom too.
Oh yeah, 3.
And
Pycnogonus.
there's a Pycnogonus sea spider.
Yeah, I saw that.
If we get it, we can send it to Kevin Klostock. He'll be very happy.
Pycnogonid would probably be smart enough to move away.
The Plecophoron?
No, the Pycnogonid.
Oh
you're
yeah.
talking about the
I was talking about
Yeah,
the Plecophoron.
light purple Pandarus is it.
You get anything?
No.
Big nothing? So the current is coming
Oh.
across, right?
The crab.
What if we go with the wand on this end?
On the other side?
So Pandarus
Not just the end, but kind of like on the edge, so we see if we can. Yeah, kind of like the tips on the left, right there. What?
But it looks similar.
Not touching it.
be there the mice and they were trying to make it photos. They're like, get closer, 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 but don't touch it, closer.
Nothing, nothing.
We should probably be careful with the photogrammetry.
Yeah, we don't want to touch it.
Like, closer.
Just right on it.
Wants to touch me. That's it.
Yeah, that was really good. I guess we're not getting anything, no?
No.
Okay.
Should we try this one?
That'd be awesome.
Which one do you want? Both? Because we have
There's the one
this
underneath.
for
She wants like the cup shaped one.
Yeah, that'd be
Okay.
awesome.
Yeah, we want to. Can you think you can get there?
I think so.
Okay, give it a try. So that was a negative for events. While it's open.
Have you guys seen any of that production of the glass houses?
In California, yeah, it's like super high. 13 minutes.
Okay.
We didn't see any signal with that same species yesterday. We broke 3 of them. Were you here for that?
No, I usually don't.
Yeah, just circle one in, give it a second.
Yeah, I'll be very careful with that.
You're going to the one on the right, correct? The smaller one or the bigger one?
Whichever is easiest, I think.
This is sponge that I don't have a picture of. That's from America. I don't know what the point.
Can I rotate this as
Huh? You can rotate.
Like this is the first sponge that I made.
Well, I mean, yeah, we fixed that, but if you want to, I would rotate it the other way, so you're not blocking your view. Come out a little bit, come towards us.
But I took pictures.
That way you don't hit that yellow sponge where you're
Yeah.
come out more.
But we didn't
Come
get
out
more
more.
sponge too much.
Now, okay.
I made it up.
It looks all right. Everything looks good. Otherwise, I'm watching.
See the outlet? It's good.
Garbage. Well, just moved in your hand. You might be able to move it some more.
I'll try to remember the next time.
All the hoses still at 5.
It's like a blasted style.
You got a good view and sit and zooming in the science.
SOLARIS event.
12. 12.
That's 13, yeah.
Oh, 13, sorry, yeah. My bad. SOLARIS event 13,
Is the big
that's...
one? Okay. You're inside of it? You're gently inside of it?
Glass
Should I get closer to it?
He's inside the cone.
What do you think?
Can you all to the left a little bit?
Just hold it there, or what?
Just rotate all to the left a little bit.
Oh.
There you went through it.
Yeah, just hold it there.
Well, if you look at everything from that.
It's off the axis.
Oh.
Oh gosh. Freezing. Oh, sorry. That's me.
Yeah, still 10.
Gotta change my angles.
Yeah, so I guess we definitely see a signal after we bump everything. You see signal of sponge?
Well, yeah.
We may have a couple people in there, I don't know.
Gotta get closer or?
Maybe not.
You're a little far out.
It's a little too far out.
And sit,
What do you think? You wanna hold it there for a minute?
We can, sure.
Yeah, if you can shift it to the left a little.
Not shoulder left, jaw.
Just hold it there.
It's pretty good there.
Yeah, thanks.
Yeah, hold it there.
Okay, I think we can pull it. Thank you so much.
No signal
Well, there was a signal when we.
when they poke. Okay. Thanks. Then we can put that away.
Just start bringing it over to the left. In that position. And work with it over there. Away from there. You got a lot of shoulder up to do.
Back in the same spot.
So what are we doing now? Sampling something?
Yes. Yeah, we want to sample that
That core?
coral that we measured.
How much of it?
I think I mean, it's a small one, but it needs to be in the quiver. So if it's too big, thing is that these ones we tried to cut before, and they're kind of rigid, so they just kind of come off. So it's most likely it'll come off.
Okay.
But I think it might be small enough that you can put it in the quiver.
You want to use one of the ones that are over there, or do you want to use one of these?
What do we have in quivers? Which ones are available?
Quivers. It's easier to tell you which ones are not available. 2, 3, and 5 are not. 17 and 19 are not. Everything else is available.
Okay.
So 1, for example, 4, 6. I don't know what's
1, 4, 6. Okay. Roger. Thank you.
Building and one, this one was well secured enough. It was coming out the door. Four. We used 2, 3, and 5, but pictures are.
Did you say 4 was used?
4 is available.
4 is it?
This is 4.
Oh, I think you didn't have, yeah. It was good time.
It was just drifting, but it wasn't.
Yeah, I didn't get to it in time. Nailed it.
So the temperature here now is 16.7 degrees.
So we should definitely make it at least 15.
So 15 is,
Again, the one we have.
yeah, 15, yeah.
What's
I
the
guess
time?
14 is kind of on the lower side.
Let's say whatever I collect. We're collecting this one for
Come up
biomass.
over that sponge and get as big of a crime.
No, this one is for quiver.
Let's say like whatever the next thing we can collect for biomass, that's what it is, and then make it call.
Okay.
It's something like, right?
Yes, that's the one. Thank you.
Santiago, what's the tide? Is it going down or up?
It is
Going down.
going down.
Okay, it's getting warmer.
That's a good note to make.
I mean, that makes sense.
A little bit further down.
No, it doesn't.
I say it's number 18639. Light purple, paramorecided.
Yep.
Thank you. SH
Association with SOLARIS event 11.
12.
12.
12, you're correct.
Yeah. Light purple primary seed SH18639. Correct?
Yes.
Solaris event 12. Temperature 89 meters. Temperature is 16.7. We're heading to a period of low tide.
I think it's optical vision, you're good. It's just optical vision.
That's weird, is it? Looks like it is.
That looked like it was on the other side of the strap that he was.
Seems like we're seeing less crazy zones here. It's we're a little deeper.
I think
There's a
so.
lot of surface.
Yeah. Interesting to check the
Should be freezing.
EK80 to see where the drip scattering layer is.
We all have this thing set on like 70 or 80.
Juliana,
Grab one
can we
side.
pull up the EK80 on one of the screens temporarily here?
On one of the straight. Yeah.
Yeah, I wanna just wanna see what
Screens.
whichever. Maybe that one back there?
Yep.
As soon as you're getting redundant. Maybe there.
Use your elbow then if you can. You're hitting closer to another.
What? Quiver 4, yeah.
Oh. Outgoing tide is a current from the north, which would be water moving from the Atlantic to the Caribbean. It's getting warmer, which doesn't necessarily make sense. That's pretty out. But maybe the water down here is coming from elsewhere. You know what I mean? Like I would expect, at least at the surface, that it would be getting colder. If there's movement from north to south in the passage. But I might just be wrong. I might not know enough about where the water comes from. It should come from north. Or during outgoing tides. I get it because I have this.
Let's see, the air is rotating.
Well, I have a surface title. I'm not just basing this off of my own perception. I have a surface title.
What does that mean?
Well, it means.
Little arrows.
Oh, just the. I don't know what these are. Those might be the names or whatever. But it'll tell you the direction the water's moving, and then it'll tell you the magnitude of the currents at the surface.
What is the direction?
So outgoing, it's to the south, and it's kind of lower now, but earlier it was. Midnight it was 0.3 knots to the surface, to the south. So that's from the Atlantic to the Caribbean. And then you can see on the incoming tide, it's the other way.
Just gotta push that foot down.
It's like a 6 a.m. to whatever. But it's strange that's getting warmer.
Nice, excellent sample. Yeah, I don't. Yeah, I see what you mean, but I don't know the water structure of a meter size.
Yeah.
Yeah, I would imagine in the Caribbean will be warmer, but
Right.
It also gets compressed, right, here in the passage, so I don't know. It's complicated.
Yeah.
Huh?
It's complicated.
Okay.
Am I good to take off?
No, I think we wanted to try the fluorescein, so the green dye on the slurp, to see if we can test one of the sponges that were under that ledge. green juice.
You still got it, you still see it set? You can see the handle set there. Not gonna do too much, but I will. Okay, I got it. Sorry. Now eyeball.
Super careful.
Yeah, so we wanna do a squirt of the green dye
On the sponge?
at the base, out in the outside.
The big one, right?
It could be either one, yeah.
Something over there that's really pushing it off. Must be Watch out. Ready to watch out a little bit? You want to go in the little one or the bigger one?
I think the little one is fine. We have a good view of the base.
You may start squeezing now.
Yeah, looks like a good spot. See if anything comes out on the other end of the sponge. Don't think I see much, huh?
No. Hey, wondering if it's being diluted too quickly for us to see it, even if it is going through.
What's that?
The current might be diluting the dye.
They said they couldn't really get it to work on the last shift either.
I thought they had it working.
Oh, that's concentrated there. Oh.
But it's like noise to the.
I'm squeezing it as hard as I can.
Alright. Some of it's adhering to the sponge. Looks like.
Yeah, I don't see any comment through.
It's like a tiny little chunk or something.
Yeah.
That's cool.
Yeah. I think this is good.
Alright,
Thanks.
we can stop?
Yeah, I think we can stop.
This is the thing, the Thesia that looks most similar to that, but I think this is actually a fragment. Because I see this thing, it's not. It's hard to tell, I don't know about that one.
Am I gonna come off bottom?
I don't think it's Thesia.
Yeah, I think we wanted to get that skinny coral on the top.
Okay, I'm just really hot right now, I need to cool off.
Yeah, sure.
Okay, we still need this together?
I'm sorry?
Do we still need this?
No, you can put it away.
Is that quiver?
I'm sorry, what?
So is quiver 18?
Yeah, we'll
I don't know. Check with
I
Schmidt.
just really need to cool off.
Does quiver 18 have anything in it? Quiver 18.
We'll put a lid on it.
It's empty. Well, the problem with Thesia Santiago is there's no single Thesia.
What do you mean?
I guess I'm quick return. What is that here? Because the species that are described as Thesia are polyphyletic, and they were all described in the same paper.
I asked you.
So which one comes first? Which one's the true Thesia? The one that came first in the paper, or like the page number?
I don't know. I'm sure there's a rule about it.
Yeah. Because like Thesia nevia is described with these other ones that are not related to it.
Classic.
We're way past it. Still past it. Come up. Come up. Pitch in.
Santiago is
There you go.
question
You're
in
almost
the
there.
chat
Go.
asking if
Open.
you saw any sponges almost
Open.
Not aware of that. No, the previous watch.
Put your jaws around it.
I'm not sure if they did any pumping experiments. We did collect the squeezer earlier from one of the sponges, but I'm not sure they did the dye. But it's also, at least for shallow sponges, we know that they're more active during the day time hours than nighttime hours. I don't know if that's true for the deeper species as well. My guess it will be less so, but there might still be some periodicity to the activity. So it'll be interesting if it was still in the water during daytime to see if there's any difference from the same with the same species at a similar depth. So right now we're cooling off the ROV a bit. It got a bit overheated working at the seafloor. So it helps to come off the seafloor. Just stop trusting so much, right? Is that? Yeah. So when we are at the seafloor.
The T4s also cause a lot of heat as well.
I'm sorry.
The T4s cause a lot of heat as well.
Ah, okay, yeah. So yeah, so running the thrusters and the arm generate a lot of heat. And so we come up bottom and stop using them as much. And with the help of the surrounding seawater, we'll cool off in a few minutes.
This paper's actually describing this one. It's a little bit more.
What is the depth range today? That is it.
Yeah. It's not the right depth range, but just thinking about species. I mean, James, this looks similar.
Who described it?
Andrew Schuler and Peter. Well, they're just, this is the guide they made with the description of all the species. They didn't describe that species, sorry.
What's it?
This is what Thesia citrina looks like, for instance. It's very different. That was tiny. Is there a good picture? Yeah. They all pretty. Ooh, that's an interesting color there. That's,
I think that's one of the purple,
Oh,
light purple permaricia that we just collected.
just has its polyps out. There's stylopathies again.
Oh yeah, there's one stylopathy. Oh no, there's a bunch of stylopathies.
Santiago, if we've moved and you want to do other things, I don't think it's unfair
right
if I
back
get
there.
that for.
I can get
Sure.
right back there if you want.
Yeah, we're back in business.
I can be as soon as I get over there. Yeah.
Okay.
Okay. Just need to get a lot of water flowing over that. But moving's okay. Not right now. We're just over here. I'm not gonna be going back there, so.
So we don't see that sponge, the Coralistus at this depth.
Yeah, I guess not.
Or maybe a few individuals.
So another.
Wow. Can we do a quick zoom? Oh, there's a Paramaricia there, a yellow one.
Yeah.
See this fan? You think we can get there? It's hard to tell what the terrain is.
So you want to go there and not that other spot?
Yeah, we'll work here. Also, it would be great to get a closer look at what's going on with all this on the top of the rock. Some of them look like zoanthids, but there's a couple different things there.
Yeah. I think they're just different colored zoanthids. I think they are.
You think they're both zoanthids?
Yeah.
Yeah, I think you're right. I wonder if those were octocorals, all octocorals that got colonized.
Yeah.
There's a lot of them.
Maybe the color reflects the flavor of the octopus they ate. There's a little chrysogorgid there too.
Where?
On the back side of the rock. We're turning away from it now. Sorry.
No, that's okay. This is a very
Wow.
cool view. You take a finger.
Yeah.
There's a lot of swantids. So many species of swantids are parasites of octocorals, and so they'll come in and take over the skeleton of existing coral.
You want to set up to sample this guy down at the bottom, right?
Correct. So it's possible that up there it was all octocorals, like this Paramaricia yellow thing, like that, overtaken by swantids. Is that gonna be too difficult terrain?
We'll see how much force it takes for you to stay in place. You're behind that, behind rock, because I'm gonna need to leave that.
Oh, Tina for NerfNet on the cover of Science.
For what?
NerfNet.
Ooh, that's cool. You get pushed around by the current?
And then also this big headache, right?
Oh.
That small coral, the white one with the yellow pops, could be paracesis.
Not that great.
Oh yeah. It's just, it's not that, it's Paramaricia. Andrews and Kathy's tree.
Paracesis is a good idea. I like it. Everything's Paramaricia.
Yeah. Yeah, yeah, I mean, that makes sense, right?
What? If it's too difficult, we can look for another one. I'm sure we'll find more this depth.
Turn the head and sleep.
Well, I can't turn my head, or I'm gonna have to use lateral to stay there. That curves in, I'm trying to make it go. Yeah, I'm trying to go directly into it.
In fact, there might be another one right here.
Yeah, but you want a bigger colony though for this, that's right.
It would be good, yeah.
Just come a little bit lower.
We don't want that much. Like, most of them don't fit in almost there, the black ones, and soft ones, I just can't tell. Can you look at the 4K screen here? Gonna be able to reach it in the 4K. But no chance. Alright, get a little bit closer.
Yeah, I mean, I think it's safe just to say Paris today looks like this with a picture. We can distinguish which ones are different, but it's hard to say which
Just trying to see if I can hold it.
they are.
Okay, good guess. I like Paresis.
Yeah, but that's the thing is like, who knows where that came from?
Okeanos is good.
Yeah, I mean, someone just said, oh, it looks like that. That's what we're doing too, right? We just say, oh, it looks like that. We have to go back.
We can check with Andrea.
Andrea, she thinks this might be Paris. We talked about that here. Because what do you have to do? You have to look at the SOLARIS, right?
Sequence it.
Well, yeah. But besides that, to get to the species description, you have to look at the SOLARIS, don't you?
Yeah, at the moment.
For the most part.
I just don't, hopefully you have enough room.
For species, yeah, for sure.
Your rocket
Genus, is there, you still have to look at SOLARIS?
I don't remember what
All you
it.
have to do is pretty much go up to it.
Oh, look up some species description.
Yeah, definition of the genus.
You go to Paramount.
I mean, once you learn the fauna of this area, these are all very likely the same species. So once you identify
Yeah.
it, you just by the growth morphology, the quality can identify it.
I agree.
Are you
The hard
good with the
part is
background?
getting it.
I'm sorry?
Are you all done with the background?
Let's see.
And
How's
you-
Solaris doing?
And do you need some images of it first?
Yeah.
Mm-hmm.
Are you doing a calibration or? You need around 30 seconds?
Yeah.
To measure the background, right? Okay.
J-Rod, if we want to get a picture, that'd be great.
Yeah. Get a good frame grab of this colony with that nice black background.
Got two.
Are we going to sample that?
We will attempt, yes.
Two ophiuroids.
There's a hydroids on the rocks too. Like that.
We just dropped the ISO at touch.
Yeah, give me a second.
Okay.
Try to get the rocks out of here as well.
Perfect shot. Nice. That's sweet.
I think we're good.
Solaris is good to go.
Yeah.
All right.
Yeah, can we just, yeah.
Yeah, gotcha.
We'll put this would be perfect, like a slideshow or something.
Yeah.
Desktop background, black on the right.
True. Work on the right.
All your icons on the right, the desktop.
Yeah, exactly. Ooh, look at that.
SOLARIS event. 14.
Ooh, right there.
Next to, what is it?
It's cameras, yeah.
We good?
Yeah. Yeah, we got it. Thanks.
All right, let's probe with the Solaris. I was going to suggest try.
Yeah, we're just trying to get our bearings right here.
Yeah, sure. Just gonna say maybe try this part of the colony that is not moving as much.
Yeah.
Oops. Keep kicking your water bottle. Sorry. Beautiful.
Once you reach, that's completely up on the shoulder, so it's all gonna be elbow.
Yeah, but you got plenty of elbow, you just need to shoulder right a little bit first.
So we're gonna.
Shoulder right, keep shouldering right.
Polyps open, yeah.
Keep shouldering right, and then use your yaw to come up.
Polyps are open. This is Solaris event 14.
Yeah.
You know what I mean?
Yeah.
So you know.
My yaw's all the way.
Yeah, you go the other way. If you would have stayed down and used your yaw,
Yeah, I yaw the opposite.
yeah, on the other way you can, no, that's pitch. Bring your pitch back.
Oh, yeah, yeah.
Okay. Cool. Then you agree with it, yeah, use your yaw.
Yeah, I think we have a signal.
Do you have a signal? Can you hold it there for a minute? Please.
We did touch an opioid on for a second.
Where?
Did we?
Yeah, the tip. I don't know if that. It didn't hit the tip though. It hit like the wand. It just wrapped, like it slid around for a second. I don't know if-
Just now?
Yeah, a second ago.
Really? That means that.
See how they're all how it's all coiled up now. The bottom one.
Oh, and the polyp's closed.
Oh.
We just wait a minute. Maybe it will open up again.
Well, maybe the opioid's an alarm for the.
That's a good sweater
Thought
shot.
about that.
That'd be pretty cool.
Yeah, I'll get a. Yeah, that's a good shot of it.
Solaris working.
And the whole instrument, yeah. I'll get that one too. Nice. So back to background.
Is that you moving or me moving?
Can you zoom in on the colony, please? Just want to see if the polyps are open again. Seems like it. Oh yeah, they're back open.
They're open.
Yeah, so you can try approaching again with the probe. Yeah, I think on the lower part of the colony would be better.
Where the current's coming at. You want more zoom? Or you good with that?
Give it that percent. This bit are like more than this more.
Like, see, like you're more, yeah, that going
Yeah,
over
like
there.
there?
Yeah,
Right
see,
there.
Yeah, you were over at the big branch, now you're
Yeah.
behind all the.
Just hold it there.
Okay.
Oh, you're still a little bit far away. Okay. Yeah, you can get closer.
That thing's gonna blow into them too, like that.
That's perfect right there. Just hold it there.
Hold it for, 30 seconds and see.
So the wand grazed the opioid, and curled in, and then all the polyps on the coil closed.
And that's when you get the signal, right?
Do you want us to leave it like that and touch it with the other arm? You're not touching it with a nuzzle, but
Yeah, I mean, we kind of, what we're most interested in is kind of the steady production of it. You get nothing? We're pretty close, right? We're like well, it varies, but we've been like within a centimeter of it. Almost touch it.
almost. Right there.
The current is very strong, so maybe it's just blowing away any signal.
Yeah, I need to reset.
Pops are opening up here.
What do you say? You want to call it, or?
Yeah. I think that's nice.
So there was a signal when you got disturbed, as we've seen before, but current is very strong. All right, that was pretty good. Thank you. We can get ready for sampling.
Can we bring down some brine shrimp and feed them?
That's what, so I was talking about the experiments upstairs to some of the ROV pilots, and they thought I meant we were gonna do it underwater. And I was like, yeah, we're gonna.
Oh, the feeding on the water?
Yeah.
Take the brine shrimp for an adventure. Just
Seems like we
replace,
could stimulate the
You
replace,
could, I mean, it seems possible.
right? You can replace the nozzle with the
Pretty
first scene,
cool,
just
yeah.
like full of brine shrimp, just like
Yeah.
here, blast.
Got a view of the nozzle tip right here instead.
So this is gonna be SH1863
Deal with it afterwards.
18640.
640. Okay, got it. Thank you. So we are 401 meters. Temperature is 15.1 degrees.
Where are we gonna be putting this at?
Biobox
Either two.
B2, 2B.
Or not to be.
When did we say the low tide was? 3 a.m.? 4 a.m.
Yeah, the tide is slack at the surface.
How much of this do you want?
Yeah, I think what you're going for is good. It's like a branch there.
Do you want
Yeah, that looks good.
more of it than that, or
Maybe a bit more, yeah. You go up deeper.
if we goes up, you can get the
Oh yeah, it'd be great to get the off your right.
yeah. So I mean, yeah. If you get that higher branch.
Wow, current is ripping now.
Yeah, it is.
towards down a little bit. Down.
I think that model on the is
Come down a little bit more. There you go. Go to the right. There you go. Now shoulder line.
You can see the surface is
yeah, it's really
You're getting there.
almost a knot.
Maybe
Well,
a little bit more zoom.
0.7 knots.
You're almost there.
Look what's happening at 250
See your
meters.
sit. You're
Yeah.
so
So yeah, I think that
you got a little
the
bit more
assumption
pitch out
that,
that
you
you
know,
can
north
use.
to south service current can make it warmer is not valid. Because look at that current difference.
Yeah.
You're what?
They have enough weird position with that.
Well, that's where
Yeah, you
where
definitely
it's hitting the ridge. Well, I guess it's hitting
You
up
don't
the
have much
shower.
pitch down. You got to
Oh,
put your
I
elbow
see.
down. Good. Get in there.
And then reset a little bit. Am I quickly to learn?
Yeah, it must be really complicated.
You got it here. Say, bottom right there.
Oh, yeah, yeah. Sure.
So much vibration.
You got good zoom for it.
Yeah.
Go in a little bit deeper than that. Just a touch more. I think you got it there, right?
Yeah, close.
Yeah.
Yeah, it looks good. Excellent. Oh. You think that will fit in the bio box? Yeah.
I think its foothold would be a lot stronger than that. in this
I know, right? With this current.
Alright, parts from that.
There's a big Nogani on it.
You want a 2 or in 3? 2.
I guess whatever it is, because this is a big one.
Yeah.
You try to put it in both threes.
No, we want to be able to collect.
Let's go into 2.
Or more.
Once we're done with 2, we don't have rest open again. I'll take it that way. Get half compressor in. It's more empty than that. There's a fish in there.
There is a fish in there. I'm getting a pet.
This thing's from Bermuda. Doesn't say how deep. It says rarefied. They're a little deeper than that now. They call it black gorky.
The base of it's stuck on that
Checks out.
basket handle.
I mean,
you can just go
like
ahead and
this.
if you want.
Oh no, I mean, we all know it's a sequence of everything and make a really big tree. With like 5 species.
Big Nogani is at the edge of the bio box. Oh no. It's in the branch of Phillips image.
I already said it. Yeah, just got some further of it as we can. That top piece that's over there can be the primary one to try to get in there.
Yeah, so we tried to cut a branch of this coral, but it unfortunately came off its attachment of the rock, which we were a bit surprised because it seems to be able to withstand pretty strong currents. But if we leave it on the seafloor, it's not gonna reattach itself. It will most likely die. And so rather than leave it to die here, we'll try to get the whole specimen because this will be a very nice specimen with a lot of scientific value that we can make a lot of measurements and learn a lot from. And it will make also a very good specimen for a museum collection.
You can get more precise. A bit deeper without it. Alright, so if you can right now, start trying to shoulder right as y'all left.
Yeah.
Like just keep doing it nice and slow. I'm gonna slowly try to bring this lid down on your jaws.
Yeah.
Alright. Keep doing that same move. Alright, just y'all. Coming down hard. There you go. Try to slide your jaws out.
Santiago.
There you go, good job.
Yeah.
Very good.
I just had to start a new Facebook Live because we
There
did
you
the
go.
max time, so I started a new one.
Okay.
Do you want to introduce the dive again?
Sure. Give me 1 second. Did you see if the branches? There are no branches in the seal.
There's one tiny branch sticking out right there.
Okay. Do you think that will be a problem for the seal of the bubbles?
Nah, that's a pretty good seal. That is just that little guy right there that's sticking out.
Okay.
Everything else is in.
Yeah, very confident. We just wanted to get super warm at the surface.
No, it'll be alright. It's a double rubber seal right there, so it's crimped down on top. Really good.
Great. Okay, we call it a really good sample. And that was a 0635. And so for those of you just joining the live stream, we're in dive 508 with ROV SuBastian, and this is Encore Expedition FK 230417. We're on the west coast of Puerto Rico, working in the Mona Passage on the Desecheo Ridge at a depth of 400 meters. And we are characterizing and sampling the diversity of corals and sponges in this area, taking measurements of superoxide production with the SOLARIS instrument. We are collecting physical samples of the corals that we measure, and we're also collecting we're filtering water with the ROV for environmental DNA. And so this is science lead for this watch, Santiago Herrera. I am a professor at Lehigh University, and we have a team of scientists on the room from the Woods Oceanographic Institution, as well as students from Lehigh University and researchers from the Smithsonian Institution, in addition to our ROV team. So we're gonna continue surveying this area and looking for more corals to probe and sample. So far we collected 2 yellow Parmeresia octocorals that we're gonna try to keep alive at the surface and do experiments on board the ship. And we collected a 1 light purple Parmeresia as well.
Just bring it in tight now. No, you kind of want to get the tip of it, like somewhere right around here. You have to shoulder left a lot. Keep it out of the
There's some whips.
Yeah, some more whips.
They have to be tight in with all the joints and we've got to be happy with it there.
Good number of yellow Ferreira sponges and some other sponges that we don't know the names of at the moment.
because the current's just ripping my clothes over.
You happy with it? Yeah.
Yeah. You can bring it in if you want, but
I saw the operations there. So, yeah, so we put that option measurements out between the, so we can measure the dial.
it's just legitimately pay out of the cap. That's all it does. But that's just a visualization of what's happening.
Yeah.
So you see these large boulders around likely have rolled downhill from higher up in the reach. Make a pretty good habitat for corals and sponges.
Is that a Parmeresia in the crevice?
Yeah, it looks like it, right?
Down in the back.
Yeah. That little yellow one? Well, that's gonna be really hard.
No,
I'm sorry?
I was mentioning one like
There's another one?
that. I don't think we can get in here, but we might want to look at this one.
I don't know. Oh, in the back.
Yeah.
All the way down.
That
No,
might-
just-
that
we
might
can
not-
just zoom and
It
see.
might not be bad.
I don't know what it is.
My guess is that purple, light purple Parmeresia. So we've seen that at this depth range between 400 to 320, 340 meters. Sponges are very abundant, and octocorals of the family Parmericidae are also common. Very few black corals. We've only seen this bottle brush species of Stylopathes, black coral. And this one may be a black coral too, but
Yeah.
get a closer soon. Yeah, that's definitely black coral.
I haven't seen this yet. I don't know what this is.
You don't know what this is? That's the first.
Could it be a Leopathes?
No.
No?
Let me get the, let me get the
branches
lighting.
like antipathes. Yeah, like they're not pinnules. It's overlapping flamellae and big molars.
You sure it can't be Leopathes?
Leopathes?
Yeah.
Impossible.
Okay.
Impossible.
They're not flamellae.
Inconceivable.
What would make it not Leopathes? Just
Oh, it's flamellae. There's no flamellae Leopathes. It's weird. Can we collect a piece of it?
Flabellet is like fan-like.
Yeah.
I learned that on this trip too.
It's really weird. It's worth collecting.
Should we collect it?
Yeah.
We can try to collect it. Do you think that's?
I think it's really weird.
Yeah, that looks like a decent spot.
You need a full full setup. Like, are you gonna need SOLARIS or just
Yeah,
a
we can
sample?
try it. Why not?
Got big
We have the instrument.
Big open olives.
No, no.
Yeah, let's do the full complement.
This is a lot smaller than I thought. I'm looking at the
I think you might be able to get there.
I'm sorry?
I thought it was like huge.
What do you think?
I'm gonna see what it could
Do you have the lasers on? Oh, there they are.
Oh, okay. It's not big.
Well, it's like a 30 centimeter across.
Yeah,
I think we only want
parts, like,
Yeah, they don't do well. And it's gonna be like hanging out.
because like it'll take down the whole tank, like part of it starts dying.
Yeah. Is he still up around?
They're going to bed. Like they set up the fixative so that I could preserve for them. And then, I don't know, they were just like winding down. They were asleep.
Nice. It must be like, start getting to sample all the good corals.
Yeah.
And your audio's back.
We're back. Great.
Back for someone to take full comments from Dr. Desecheo.
So we just did Solaris event 15 on this Antipathes flabellate colony. Flabellate meaning that it's fan shaped. And we did not observe any signal of superoxide. That's been consistent with all black corals that we measured so far, or most of them at least. We're at a depth of 401 meters. Temperature is 15.5 degrees Celsius. And we're getting ready to sample this same coral colony. So you said quiver 1 is just sealed tight?
Seal, it's the whole quiver's coming out instead of just the
Yeah.
cap, so that finishes up.
Maybe it had a little bit of air or something in there. So 6, 7, and 8 are available. The back row.
What's the temperature here?
15.5. We just had a change in the watch standards for Solaris. So we had Lena and Erica step out and William come in. And Eric
Eric was up in the Masters.
coming up.
Yeah.
He was just you.
He's excited about photochemistry as usual.
I can't find any description that's like this. This is really weird.
Could be a new species.
I can't, nothing, yeah, I can't find anything that's got overlapping or, you know.
I'm sorry. I was just wondering where she was. We don't need her, but they will, when we come up, it's gonna be sweeter water and probably miskins. Just trigger
I can try to get you it closer, but
Nicole,
it's not fun.
what miskins have in trigger? I'm sorry? Did I call her Nicole? Sorry. Sirmalini.
I don't know if any, because here is nothing.
No, I don't think anything.
Interesting. Okay. Yeah, we should.
Just for preservation's sake. How much longer do you think we'll be able to until we fill the box?
How much Solaris? Oh, good morning. Hello. How much Solaris reagent do you think we?
So we're about halfway through with our SOLARIS reagent. Thankfully, thanks to Williams updates, the software is working much better now, so we're having to restart a lot less. So now we have an accurate track on how much we have.
Excellent. So thick.
Pictures.
You want better?
Yeah.
I'm just saying, you got a screenshot.
Yeah, I got one.
Get that one.
You might want to grab some more if you need them, like for. I can get highlights of this.
That's a good one.
It's really nice.
Maybe a full frame one, full colony one.
We did get a full frame one.
But this seems like a nice lighting now. I don't know.
Yes.
Scottish monkeys was.
For scale. All right. I think we're good to
Alright.
Exactly.
to grab.
Yeah, if we could grab, that'd be great.
It's funny how misleading the size is.
Yeah. It's like a perspective issue.
Sorry, I am not very stable. We're gonna have a little target. Try to get it through a little bit better.
So let's start off with the one that try and do all
He wants as much as you can fit inside that world for me.
I think you got it mostly.
It's a nice job.
Nice.
These guys could sample crazy.
Good.
Beautiful.
Nice.
So this is gonna be samples SH18641.
Exactly.
Correct. Thanks, Marlenia. And we got quiver 8 open. Juliana, is it possible to bring the color room up to 15 degrees Celsius? The the call lab, yeah. Thank you. You think that's what, 15 or 16?
You don't have to do that.
So it's about like 15.6 or something?
Yeah. Well, right now it's 15.2. It's gonna like 15. Yeah, let's say 15.
15?
Yeah, 1.5. Thanks.
That part is that would not work, so that part is going first. You have to, when it's close to supposed to get it in.
It's quiver 6, right?
That part
This is
and then
quiver
slide
8.
towards us,
Right?
and go down,
We'll double check.
and then get that other piece in.
Yeah, you can see it on this
Now
camera.
start swinging it.
6, 7, 8.
Use your yaw.
You see the lid?
I can see the
6.
7.
You can see the 7? I can see the 6.
Do yaw and shoulder left at the same time. There you go. Yep, just like
Oh yeah,
that.
and the 8 is the one that is resting here.
And then
It's the 8.
Pump open. And slowly start coming up. But don't open too much or you'll actually grab the order. Alright, you can come up a little more. Open. Go.
It was already at 15.
Oh.
Oh really? So I thought we had it set at
It
14.
definitely was 14. Maybe someone else.
Did you? Was that the setting or the reading?
The setting.
Settings 15.
Yeah.
Oh.
because it was originally orange.
Yeah.
But I don't know who else would have changed it.
I'll make sure it was still set instead of just.
I can check it,
15.
I'll check it out for.
Okay. Unless there's like a slight offset between the setting and the actual.
That could be. That could be, because before we had it at 20 and it was like maybe 19.
That's true.
Yeah, good job.
Nice. Did you buy those quivers lid stoppers, or did you custom make them?
It's custom made with, I mean, the black portion of it is actually just a blank for if you replace your toilet. That's what you put there.
Nice.
And close it off until you put the new toilet on. And then the metal rings we fabricated because the plastic, of course, us pushing down on them was breaking them. So added that, and I changed out all the hardware because they don't use any stainless on those plumbing things, so we changed all that out.
Right. They don't need to use them underwater. So these are stainless.
Yeah, they're stainless like that. The ones that are rusting is, I don't know, I guess it's just cheap stainless.
They gave you some 316 and some similar stuff.
Yeah, we had them made in, the plates made in Australia. We went to a Home Depot and bought every one of these blanking things that we could find. Whereas actually, I guess they don't have Home Depot. It's like Bunnings or something like that. Nicely done.
Yeah, they're nice. I like those metal tops for pushing down. Really good.
These look like that.
Some of them are tricky, man. It's good. It's good. Fun.
I'm gonna try
Great,
to get out of
great
this
sample.
hole.
Thanks. Let's keep looking around. Be nice to get a couple more of those yellow Parmericias and see what else we find.
Yeah, I'm trying to get
Set
all
point was 14, it's now 15, and it's reading 15. Great, sorry.
Thank you.
Yeah, so it was 14. Got it. It was for some reason cold.
I'm happy to be out of that.
I think that's good.
So we put some in before, but just a lot of reference and we put some in before. Just 6 and 7 and that front box. Wow.
Alright, can you try and stow? Alright. How far are we gonna go?
Or was that just for the clay? Yeah, so you could.
Oh, definitely more out like straight up in the camera. Yeah, really trying to get that tip down close to the rail.
Oh, but you want, I saw it, I thought you always wanted it up.
No, no.
So you can see the railing.
No, as long as it's above the rail, I know I'm hitting the rail before I get that tip. Is the best thing.
What's that?
Okay.
Oh, Santiago.
What's that guy?
Yeah.
What? Oh, that's another one of those antipathies?
Is that the same
yeah.
thing?
Yeah, it'll be good to measure that one and sample it too. So the second species is black coral in this depth range, so it'll be good to have
Yeah, I'm probably find the third here too.
a pair.
I thought it was a purple paramercy for a second.
I was checking it, I think those may be deeper,
Oh.
like in the 500 meter depth range.
Here we go. Right? This one might be. I'm gonna say that.
Oh, there's a Santiago. There's a Chrysopodium.
Oh, yeah.
We've seen a few of those. We might want to
Sure.
consider them. Have we
Sure,
seen
sure,
more?
sure.
Oh, there's two here.
To what?
Two Chrysopodiums.
Where?
Right here.
Oh, yeah. That would be easier. To measure.
In the spa.
No, those are paying.
Oh, yeah. Sorry. Spa officers are moving around. Maybe ruffle their feathers a little bit.
Yeah, it'll be good. We wanna
Have we
get
tried
position to measure this fan.
On what side of that?
Good question. Current's coming at us, so probably from this side. And then there's another, you see this fuzzy thing here? I also would like to probe that one. Ooh, look at that whip, bottle brush thing.
What the heck is that?
That looks like black coral, but different, right?
Where is that?
On the left on the screen.
Oh, it's big. It could be.
Not Stylopathes?
Come in more.
That's huge. I don't know.
Or it
How
could maybe like a
SOLARIS.
sponge.
How are you guys doing?
Yeah, so we're good. So just to help conserve a bit more reagent as compared to yesterday, if we could get into precision first, then I'll take the background at the site, and then we move the probe in. Does that seem like an okay protocol? It'll take a little longer than before, but it'll help us have a much longer dive.
Sure.
Yeah.
Give us.
Okay. Are we in decent position then in terms of the ROV?
Not yet.
Getting there.
Okay. Okay. Just let me know once we're seated and I'll start on collecting the background.
Sounds good.
Great. Thank you.
You think it's a sponge on the left? Like a glass sponge?
Which one?
The bottle brush?
No. My guess is a black coral from this view.
Yeah, we see a black hole.
But we'll take a look at that after we're done with this
What's our depth?
couple corals.
400. Alright, Santiago, I'm gonna start pumping some EDNA samples.
Okay, we're gonna start filtering water for eDNA on pump
Number 11.
11.
The pore size is 0.2 microns.
0.2 microns. What's the sample number?
Give me a second. 13547.
SH number.
Santiago. Sampler's working at 400 meters. I didn't put that together.
That's what I was.
Yeah.
Well, we had. Remember you had issues with it.
First dive.
First dive.
Talking to me for
Stop talking at around 300.
Screenshot of this one before we go into.
This
Yeah.
is definitely the same species as the other one.
Excellent.
What species is that we're thinking of?
Antipathes.
Antipathes. A genus.
Another one.
Okay, before we get too close with the arm, we're gonna need to get the background in.
Yeah. About how much closer do you want us to be?
That's a good distance away.
Okay.
Alright, collecting background signal now.
Oh, yeah.
Ooh, that's a nice cockroach.
Oh, yeah.
If Claudia was awake, she would want.
Why haven't y'all been getting cut close?
Huh?
Why haven't y'all been getting cut close?
Didn't they get some in the last?
They tried to, I don't know if they succeeded or not.
I got a really good method.
You want to suck it up?
No,
I think they collected some.
I think we're gonna try to.
Let me check. Sam mentioned it.
Oh, that sounds right. I don't remember.
They collected a shallow one.
We are good from the background side, so
Oh, this is-
Solaris is ready to approach.
The black one on the underneath the overhang also. Up in full dome.
Oh, well, there'll be a train guard with it, right?
Yeah, sorry, I got ripped off.
Right, it's all good.
Do not know how that tech turned out.
Using the phones, I have like 8 samples today.
80?
8. Oh. And 2 Niskins, so maybe 10 samples. Oh god. It's good for my watch schedule though, because it'll probably come up in the middle of my watch and I just you have a good data set. I like the sampler.
I'll see. If that is working. We should send an email to Annette later today. Give her an update.
Are we planning to go a bit deeper here at any point? 800 or
Yeah.
it'll be good testing it and see if it works at 800 meters, then A plus perfect score.
Yeah. Look at those fish. The friends. I feel like I'm fully into the watch schedule now.
Yeah.
I feel like I'm fully into the watch schedule.
I feel so much better. I slept really well last night. I finally feel like a normal person.
Yeah. I know I finally slept and didn't wake up last night.
Yeah, that's really
I
good.
slept a little in, I was like, oh. I did my normal thing, which is like set a bunch of alarms, wake up, and then fall back to sleep.
Come on, Solaris.
Didn't do it. It's a cool thing, huh? Yeah, 2 of them.
Yeah, there's 2. There's a pair. Oh, 3.
I think that's a shadow. One's a ghost.
No,
No,
there's
they're
3.
six, they're six.
I see their eyes.
Yeah.
And that's Solaris event 16?
Yes, event 16.
Solaris Event 16. On this Antipathes coral. Black coral. So we didn't get any signal on the last one.
We do not have any signal thus far.
Okay.
I guess that's good, yeah.
I'm sorry?
It's good that we won't get a signal.
It's good.
I guess if they all don't get a signal, that's good.
Yeah.
Consistent. Yeah, it would be weird if we like, well, we,
Well, it's, I mean, it's more interesting if like some produce and some don't.
Yeah.
Try and figure out what.
Because there's no good or bad, it's just interesting.
Yeah.
yeah.
If they all did the same thing.
If we get down to 800 and find alternatives, because that's a real big columns. I don't know.
But we try to measure that alternative patties, yeah, remember?
Did we, on the first side, did we try?
Yeah, they stuck the thing in, but it was ripping. It was like, oh, super strong.
Oh, right.
Is that the one? That's what you were talking about? What is that? There's like a glass sponge.
Yeah, that's a sponge.
Oh.
There's,
Do you have interest in knowing the polyp
Oh, I see
size
that,
for
yeah.
all of the things we collect?
Sure.
I could do it.
Sure.
And then you have that data for the.
no.
That would be great.
It was like.
I don't think they're getting anything, right?
Nothing on our end.
There was another.
It's been pretty close several times. That close to the colony, I mean.
Yeah, because it's definitely wavering back and forth right next to the road.
Yeah. Okay. Good data. Really
Wanna
good.
call it no signal?
That's
No
why.
signal, yeah.
All right. So that's the end of event 16, no signal. Before you put it away, there's this other bushy coral that we think is a Chrysogorgia or Chrysogorgid on the top right screen. Let's take a close up first and a frame grab to confirm what it is. Yeah, that looks like a Chrysogorgid for sure. You got a frame grab?
Got it.
Got a frame grab?
There's a little squatting in there.
Yeah.
There's a spot lobster, yeah.
Yeah, it's a price of
So this is.
Squatting too high.
Yeah.
It'll be good to measure this. We've seen several of them. They don't get very big, but they seem abundant.
They have a lot of polyps in one place.
They do.
That's good for us.
You know?
So do you want to call this event 17?
Yep, exactly.
Okay. Are you ready to go in?
We are ready.
Okay, great. Let's go for it.
Some of these can be. So there's a pseudo Chrysogorgia, which I think is more closely related to Metallogorgia.
This?
Yeah, if we see a black skeleton, that might indicate it's pseudo Chrysogorgia.
It looked black to me.
I was just looking at the tree, and something identified as pseudo Chrysogorgia is closer to Metallogorgia, which would be interesting. I mean, if they share a black skeleton, that might be a good indication. But I don't know how right I am about that.
How's that? I'm gonna make it a little bit brighter so you see it like that.
Okay.
Alright, and a reminder, this is event 17.
Yep. Thank you.
Event 17 on, what do we call them? This Chrysogorgia?
Yeah, we can call it.
Pseudo Chrysogorgia? You want to call it pseudo Chrysogorgia?
We can call it Chrysogorgid.
No, but let's differentiate it. What if we find another?
Well, why don't we just call it Chrysogorgia black skeleton?
Okay. Chrysogorgia black skeleton.
Yeah. That's its morpho species.
I'm not too afraid of being wrong. We're gonna be wrong in most things.
Okay.
As long as we're consistently wrong.
That's
Yeah, we just wanna know which things are the same and which things are different.
That's right.
It went for a long time.
It was an active.
Alright, we are reading currently at baseline.
I feel like that's a really good credible quote.
Polyps are all facing you.
Whoa, the current is strong. And so we have a strong current coming from the west, almost.
These ones, I don't know if you want to notice, but they always have a squat lobster.
Squat lobster.
They're doing the squat, yeah.
Interesting. I want to switch around.
That's kind of, yeah, it's a difference between most other corals. They always have it. Some corals have them, but these ones have like the seemingly obligate As you can see though, the spot lobster is not very active.
It's just squatting.
Yeah.
Oh, he's just hanging out.
It's interesting because like some other types of spot lobsters, you know, they run away.
Yeah.
But in my experience collecting these, they don't even bother to move. Less than an ophir or even.
Can you try to get on this side?
Kind of helps me, you know, that makes me think that they really are like active for the there. The ones that move around on the rocks, they're
One of
really
those
hard to
things
catch.
that-
Yeah, they're really hard to catch.
Touch it and
We've been pretty
polyps close.
consistently at baseline. I guess we can wait to see if there's a spike from the polyps closing.
Yeah.
Appear to be the case though.
I don't think it's so diluted with this guy.
Nothing. All right.
Okay.
No signal. Okay.
So we're gonna sample
Event
it.
17?
Yeah, we're gonna sample both corals that we measured.
Would it be possible, just for logging's sake, to sample the black coral first, Santiago?
Yes, let's do the black coral first. That'll be best. I agree.
Then we won't get confused.
Uh huh. I agree. So that was the end of event 17 with Solaris. We're gonna get ready to collect physical samples of these corals.
Okay.
So we have quivers 6 and 7 available.
Got a pretty high flow rate off this pump, which makes sense given this is our deepest here.
Less stuff.
Yeah. Less craft. Less vital linking craft.
They're, what do you think it is to the left?
So what? You talking about the big?
At the thing, that might be a black coral or a sponge.
Oh, that's a sponge for sure.
You think it's a sponge?
Which one?
Like way on the left.
That guy there.
This? Nah, that's a black coral. Oh yeah, black coral.
Right.
Oh yeah.
Whoa, look at it.
So fuzzy.
That's like a proper feather duster or whatever. Pipe
I
cleaner.
got fooled by those things so much when we thought they were corals.
Because we
They were
collected
just
a lot
like-
of glass sponges that time. Cool. It's a nice location.
What's that? The one right here, this one. Yeah, going back in order. Sometimes it's better just to try to get your fingers to work through it. And then.
Would it be easier to grab it from one of the sides?
Oh, it's just so.
It's like all attached.
Yeah,
to
so
it.
he's trying to get it like, or push a little bit right there. Keep doing it, just.
I think you got some fusion there, Jeremy.
Right? It kind of looks like it is, but they could just be
just like
kind of interlocking with each other. I don't know. Yeah. And they could be like non-perfect fusions where like sides of branches are fused, but they're not into each other. I don't know. We shall see.
Rotate clockwise. Bring that back right down. So there's a
Polyps are barely retracting with all the.
They like us.
Yeah. It's a good scratch.
I can't even see the main branch on this thing.
Yeah, it's just on that side.
Yeah, there isn't the main branches there.
Don't do this.
that necessarily? Yeah, that's fine. Whatever you get.
Nice sample. Excellent.
Perfect.
It's not right.
Yeah.
You can really see which ones have the filters.
Those ones have filters?
Yeah.
Have they flow?
Yeah, they don't allow any flow.
Works
well.
Oh.
Been mixing them up too, I've seen those.
It's very good. It's nice to have all that data. Oh, you opened number 1. You did it.
Sample number 18642.
Oh, thank you. SH18642 confirmed. Antipathies going in quiver 1. We are at a depth of 398 meters. Temperature is 15 degrees.
Just living the life. Get black ones. Or watching people get black ones. You guys are beginners.
Nice, nice, nice. It's a good sample.
You like this depth?
Yeah. Sponges are great now, but this is 300 to 200 has very few corals. Got really good samples.
Mhm.
You have like, I don't know, by the end of the cruise you might have over 10 of those pore size.
10 million?
No, like 10 sets of 3.
Yeah.
30.
I think you think.
I think that's the minimum to expect. That's really good. You'll easily get 10 sets of 30. You'll easily get 10 sets of 30. I think I might already. I'm already.
You're done. You can retire. You can get out in the morning.
So that's only, I mean, that's only, well, that's kind of low. It's only 30 extractions, but filters need to be divided. I don't know. What's that?
They eat any samples going to the Darwin station.
Oh, cool. Oh, they're on there? That's good. They're gonna have a journey.
Hmm?
They're gonna have a little journey. Yeah, they got a good number of samples. That might be a really nice project. Especially if Dan has token. That would be a really good data set.
Yeah, I don't know how much token versus
The outlet video, you know, it's gonna be a lot like token is easy to annotate. That'd be something that
Milan.
Milan can work on, yeah.
Nice. We got another good sample in the bag.
Just need one more of those and we're done with that one.
Done with the cruise, that's it?
I'm sure we'll have one more around here.
Oh, it's sticky. Jeremy likes the closest.
Yeah, they're tough. They're tough. collaboration.
I'm sorry?
If it's a species, we do a little collaboration and species description.
Yeah, it'll be fun.
Kind of looks like this one.
Do you have any species in
I do not
there?
like this squirrel.
Yeah.
Fly, fly, fly away. Right now. Yeah, I'm trying to get it in there right now.
Do you know, alien Xlei at the Encore?
Sorry?
Do you know alien Xlei at the Encore? It's like, it's super abundant in Encore, plankton. Oh no. So I took it. It's not toxic. It's just, it makes like the milky blooms, or it can, I guess it can be, where you can use like the sulfate. Yeah, not like neurotoxic, but anyway, so I took a class from Chisholm. So they did because it has to be, the species description has to be agreed to.
It's for this, but you're gonna have to exit those.
Race genes description.
It's frozen. It's frozen.
Yeah, that's it. Just take it.
What are you showing?
Take it.
Thought it was like a, like a bio box person. He looks happy sleeping. It's stretched out.
It's not like it's in, but it's
So relaxed.
stuck to the soft drinks and all that stuff.
Looks like a base.
Ooh.
You see what I mean? Just not like, let go. Trying to get it in line. I'm not
This time yesterday, it's been really bad.
Yeah. Yeah, a little more smooth.
No.
One of my things that's the worst I get.
Get in there. Coral, please.
It's in there.
Majority, yeah.
Just that bit.
I think it would be cool to do like on the SOLARIS, not on the wand, but on this sampler held part, like a little current meter. It just hangs with the current. Yeah. It's flying away.
Oh, that's good.
So I'm not sure. People start reviewing this
Push it forward
one.
a bit. Grab it, grab it. Close it, drop it.
Well, she asked me.
Bend. How many times I can
It's
say
actually
close your
kind of
jaws?
nice because it means like the rents are shoveling it in the ethanol.
Stop moving so much.
Okay.
They went too far away. If I say wind next time, you don't close your jaws.
Okay, fly.
I'm coming up and bringing it to the quiver.
I think we'll call this Antipathy's Escape Yelling.
Yeah, that's a nice name.
Alright, come before
You
us.
have to
Right.
describe it.
Alright.
Keep it in mind.
That's right.
Right.
Red in the hole.
Ah, so close. It's right
Almost
there.
made the 3-pointer.
Oh,
Yeah.
Off the ground. Grab it. Coming up, coming up, coming up. Go, and don't go to it. It's pinned down right there.
Is it in?
Okay. On your left hook, grab. You got that handle.
You don't want to pop over the
Down. That's it.
I'll give you signals. Not too far away. That's fine.
Yeah, but it's just next to
You ready? You ready?
You missed the corner. Yeah, we can see that.
Porch coming out. Porch coming out. A little bit more. There you go. Coming to you. Stay there. Go. Corner. Grab it. Grab it actually. It's there you go. Press. That was big. Coming up. Nice.
That's
Looking good. You're right over it. Just go a little bit more outboard. Start coming down. A little bit outboard. Outboard and pull that. There you go. Good job. All right, let's try it. Down over here. All right, let's press it up. Pull it hard. Start releasing. Gotcha. Start it up right now.
Took some mystery cold medicine or the first aid box.
Let's just show this in a bio box, I think.
Yeah.
Oh my god.
That would be amazing.
I can see the fused branches now.
Yeah, they're causing problems.
Just put it together with the stilaster and bio box 1.
I think you he has a pretty good handle on it to go in on the northern tribe.
Do you want to give it another try?
I'm totally whatever you want right now. This thing.
It's. I'll just say just put it in a bio box and just forget about it.
If it's like coral waves.
Do you want to find another one? No. Maybe in an hour or so when we have recovery.
Emotionally draining.
box 3.
No, it's by box 1. Just share it with
Box
the.
1, that's way easier.
That's one of the stairs.
I can save you a little piece so you can keep it for some memory.
Are you sure? Can we do Biobox 1B?
Biobox 1B.
Yes, please. Because Biobox 1, it has two pilasters.
Okay. 1B.
Yeah. Alright. Last chance to escape.
1A is already confusing.
Duh.
Yeah.
And it works to make sure you're down in it.
Oh, this corner was pain.
We'll do that same thing we did last time. Alright, check. Box starting close.
It's trying to float away. The dang thing.
Come to the port a little more, yeah, just like that. Alright, start. Bring it down. Alright, go ahead now.
Ah, yes. Great job.
No more of those.
We need one for this. If we find it. That's funny.
Like 20 minutes.
Don't worry about that. No, that thing's gonna come right back off.
People are really good at that. People enjoyed that video, the live view, or chat.
Alright, we'll look at that.
Getting some claps.
Action packed.
Okay, so what?
Claps from the live chat.
You think we can get back to the same spot and grab the little bushy one? Oh really? Huh. Yeah, so our heading is west. The current is coming against us straight ahead. What time is it? It is 3:53 local. And the tide, low tide at 4 a.m. Oh yeah. We're gonna have some strong currents for a bit here. Can we check out those? You see those fans? There's a couple large fans. Yeah, I guess this one. Just a quick zoom. That looks like a black hole. Oh, look at this one, Luke. The bottom left. I think that's the light purple primary seed. It looks a bit different. When we're just growing.
Well, the big one was. That big one looks like a proper pyramid.
Which one do you want to go to?
This one? This one.
We're probably not going to get Chris Quake aggressive yet.
No, but there's another one right
I
there.
can get back to that crescent virgin.
No, I think the purple one's better, right?
Let's take a look at the purple first. Just not sure if we've seen this one before. Are we deep? Oh yeah, we dropped 20 meters. That would explain.
Back and down, yeah.
Yeah, this looks like an actual primary seed.
Yeah.
Beautiful.
You see them chemicals just laying down. Yeah. From the folks, she's getting rid of behind us.
Oh, there's a little crescent virgin behind it.
You see one?
Oh, yeah.
Oh yeah, there it is.
How's that fish not having any problems?
He's chilling.
Four 400.
110 meters.
Right.
I'm still working.
Let's try to measure this one. Might be a good candidate for ROS. We haven't seen the species so far.
Are we in a good ROV position?
Oh, I think we're.
Bring it in.
Yeah, let's bring it in to do SOLARIS and sampling, please.
You're too far away.
And just let me know when the ROV is in position and I'll start running the background.
Give it 20 minutes. If I go push up against that rock right there.
It's like if I make a correction, I just drop it in all caps.
You were what?
I had to make a correction because I put an SH number for the crescent virgin and took a picture of it. So I just made a correction by typing in all caps. I think that's clear enough. There's not I don't think there's an easy way to go back and do something.
No.
Okay, looking at Alan. I'm looking at, yeah. It'll be the third and the last.
No.
Did you find another one of those things?
Yeah,
Are you giving up?
Yeah, J-Rod, it's that
I
one
think
tuck.
J-Rod's about to walk
It's that
in.
one tucked in the crevice.
That's been wanting to get here anyway.
He wants this one right here, the one tucked in the rock.
Is that why you're leaving?
You should cool down. I just got your axials on, so you should get pretty quick. 55. Yeah. But you should be close enough to the front.
sniff the front or back?
The front, I think, because the current is coming from the back.
Alright, starting the background
You just
measurement
watch
now.
yourself every now and then. See other focus right there? Every now and then, something like a current or something hits those floats, it will just yank you. So just be ready to clear out, get that nozzle out. I'll be
be right back.
So if we could hold Solaris here right now so the background signal can stabilize.
Go, Jack. Is that a lobster thing? Sure thing.
We'll just need to hold here for about 30 seconds.
What should we call this? Purple Parmesia? Parmesia purple? Parmesia purple.
This looks like Parmesia.
Parmesia.
The real Parmesia.
Okay, Solaris is good on our end. Thank you for your patience.
And this is gonna be Solaris event 19, correct?
Yes.
Okay.
Is it ready to move in?
Yes. It'll be good to aim for this lower part again. Just moving less, swinging less.
Wait for that. Set.
I'm gonna watch this vehicle because I keep pulsing. Yeah, I'm able to swing that far.
And zoom out a touch from your. Fully zoomed in and zoom out your satellite.
Yeah, I'm gonna try and give you some signal. So I can see my foot. Still behind it?
Yeah, not too close.
Yeah. I'm afraid if I extend my foot, is there any chance you could shoulder down and be all right?
What child would they want?
Yeah, we need to open up the angle or back up further away because it's
Okay, put it away for
alarms
now.
all like jarred up.
Roger that. Put it away for a safe place. This is fine. That's what I can do with my position.
I believe we stopped the thing that was changing operations.
Oh, let's give it a test. I haven't done it in a while.
See what happens.
Have we been able to save the frame rate images used before?
No.
Or just save them locally? And do that and just set up, just fix the permissions.
Okay.
Can we just add it one more
I'm
time?
gonna ram out a little bit.
Because we stopped the sync.
Okay, I dare not ram out much further than that.
We need
think that's enough?
It's just kind of hard to get to this side of it. We're being so close. It's like the arms fully in.
Yeah, I
Supposed
ran
to
that.
be all I've got.
I got about 4 or 5 inches.
Is there another position we can go in and try and
I'm not gonna restart.
I got it.
try?
I went out a little bit.
Just because I'm very limited. Like I'm all the way as close as I can get to the sun.
Alright.
So you
Because I
take
ran
keys
down
now?
this one too, so we're further back.
Yeah, the process is stopped. We'll still have to eventually figure out what processes, what specific processes we're mixing up, and then we will have to try.
Okay.
Sorry.
Are you gonna read through this process one at a time?
I'm not sure how much we're doing that, and then we'll just talk over. Really. Make it done for this summer. And then hopefully that's.
Well, they can do the same species, different color,
Yeah. I'm still
unknown.
way past where I need to be. I'm running out of. All I've got is azimuth.
Yeah. Yeah, we'll have to do something with it.
I mean, like, I can keep coming like this, but. Still maybe.
That should be able to move forward now. Okay, going shoulder right.
Stable.
Yeah. Stay close to me.
Getting close.
Obviously limited views because of the way I've had to move my arm.
Mm-hmm.
It's hard to tell if it's wrong.
It's very close, I think.
That looks very close, yeah.
We're not seeing very much of anything.
Touching it there, yeah.
We just hold it. Oh. Yeah, just hold it there.
Might be getting a small increase in signal, or there might be debris since you don't have the filter tip on. A little. I think it's safe to say that we aren't seeing too much.
Nothing? Do you want to wait or call it?
We've been measuring here for a solid 2 minutes since we approached. So
You can try a
I
higher
think I'm
point
comfortable.
on the column.
All right. No signal. Well, thank you. No signal. What did you say, Luke?
No, I just wanted to maybe try a different place.
I don't think we have too many options
Yeah.
with the arm. All right, we can. I think we can put Solaris away and get ready for a sample.
Cody, SH18643.
Thank you.
We call it Paramauricia purple.
That's right. Paramauricia purple.
Yeah, give me. Get on a quick bit some more. So I'm gonna.
SOLARIS event was 16.
This was 18.
18.
Help me with it, is there?
Let's see. Yep. What I don't know is we're gonna reach this.
Maybe a stretch.
If I ran in a little closer, which I don't think was the reach.
Maybe we should just try and reposition a little more, turn kind of 12 bodies and shuffle before.
I'm sorry?
We're going to have to reposition
Okay.
somehow. I don't know what we're gonna hide. Here we go. It's going to a quiver.
Yes. I think we have one open. I think quiver 1 is open, right? That's the one we were trying to stop the antibiotics.
Carelessly hanging, but we'll be quick about it.
No pressure, you need.
No pressure.
Hey. This should be easier than the black coral. We just did. No floating. I think there was, let me see. I think maybe this branch, tried that. Might be a little bit wide to go in a quiver. Might have a similar problem that we have with the other problem. But I think you see this.
Get a bit further past him.
Yeah.
Need some zoom on this one.
There.
Hard to tell.
Zoom in the top, zoom in the top one then.
Yeah. Yeah, I think the top one will be easier.
It's as good as any. Yeah.
Sure. Give it a try. It's not big. I think that's on a bio box. Actually.
It's fixed now.
It's right on the blade.
Yeah.
Come on.
There you go.
Very well.
The arm is grabbing.
Yeah. You pull it. There you go.
Oh. This guy's alright now, he's just got the juices.
I think that may be best suited for the bio box.
Whichever you want. Which one?
I'll drop the arm.
Which ones are is empty?
Bio boxes, 3,
Bio box 3.
8,
Coaching out. My box 3. Opening my box 3. Start closing a little bit right. Should sink.
this one, 3, A or B?
A.
Hey. All right. Great job.
Temperature?
The temperature is 15.6 degrees, and depth is 410 meters.
That marks 3A.
You said 50.8?
I'm sorry?
50.8?
What's 13.8?
15.8 is the temperature.
15.8.
15.5.
Before we move on from this site, is it okay if we pan the cameras toward the SOLARIS main body, just to get an idea of how much reagents we have left?
Yeah.
Kind of that central cylinder area, not on the wand. So yeah, panning even farther over.
I think it's talking about
farther
the
to the left.
mounted on the ROV.
Yeah, we were trying to reposition it last night so we could get a glance at it.
So like an actual ball.
Mm-hmm.
About a couple of minutes.
There it is.
Lights. And light.
So this one's.
No. Just let me get it. It's me that.
Some more lights back there or something. Still getting.
Like been out. That way. There you go.
I can give my sec. If you keep the camera and stuff there, I'll show you the lighting.
What are you trying to do, Nick?
He wants to look at the let's have a look at that thing.
Click your focus button. Or here, I know, I'll get it there.
It's cool that it has a lens on focus.
You zoomed all the
Oh, the
color.
Oh, better.
Much better.
That's good. There we go, it's on the other side.
Ta-da.
Pull the camera in a little bit.
Okay, I think we've gotten a good enough look at it.
Pull the camera in.
Next time when we mount, we'll just need to rotate it further so we can see those other bags a little better. That's a great view of the sides,
There you go.
Okay, thank you guys so much.
So fluid.
Looking good?
Yeah, we were hoping that we would be able to see these two bags that are kind of hidden by this post, but that's just because we thought we rotated it in a way that would allow us to do that, but apparently we didn't quite get the angles right. So, notes for next time.
Okay. So all these
All right.
covers off of this one, I think. That's what we're doing about.
You guys are doing a lot. I will see y'all tomorrow.
Hey.
Yeah, so that map here, right?
So this one, number one here, what's that lid off of there?
No, sorry, that's right. So we weren't gonna use that because the low cover was coming out, but then we got a awkward coral that didn't want to go in and kept escaping. So that was the last one we were trying to put it in. But we ended up putting it in the bio box
Where are we
instead
going now?
because I kept trying to get away from us.
Okay,
So
what's the
one
next one?
is
Yeah, we're gonna
available.
keep looking around, making our way to On the way to the waypoint and
It's also plugged in.
hope to get a couple more of those yellow
How do y'all like them over there?
yellow
Better or
fans
worse?
that we sampled earlier
No,
on.
matter.
It's alright, I mean, it's not been any issues so far, but like I said, I didn't use one before, now.
Okay, so we're gonna keep moving to waypoint 20?
Yeah, that's right.
Howdy, man.
Howdy, how you feeling?
A lot better. I heard it's like crazy current. Oh, yeah.
Hold on.
Little supplement. Alright. Nice. I see it's showing a little bit.
First, we have two calibrations, right? So
Yeah, we're
but we're not planning on putting that up super soon, right? Because we still have.
we're at the peak of the low tide. Oh wow, look at that fracture on the see-through. It's pretty cool.
We should have almost covered enough here.
I have to creep up.
Well, we've always ran out sooner than we expected, right?
I think I saw another one of those purple fans. You see it?
So
Yeah, but I would say, I mean, I think probably
Like
our
try
estimate
pulling your lowers for the sound.
header probably comparable to these, right? I'm just like, I wouldn't take this number like as if it's necessarily accurate, you know? I think I should confirm that our experiences based on our intuition, we've always ran out sooner than we expected. Now, maybe that's because we haven't had this good quantitative number, but
was that on the previous expedition that you had that, or is
no,
it?
that's not, the times previous, right? We ran out, it was still way sooner.
Right, I think that was because we had to keep resetting the file and we didn't have any sort of track. But so this value is based off of the flow rate here, multiplied by time, and we know that this flow rate is actually slower in actual practice. So if they can reset this file at all, this number should, in theory, should be lower. So, yeah, that'll be
But
good.
in practice, we've always done that short of these things,
It'll be interesting to see.
but when, I guess the question is, when should we have their protection? Because if we just wait,
Right, exactly, so I think we should probably do one right at the end before we come on, just so we have it final fragments, and then, maybe not even all that, because sometimes we have one in situ, right before we come on, I think, but, I guess one, so, I guess, Hey Santiago,
Yes?
do you have an idea of how much longer you want us to be down here? If we were not limited by reagents, is there an estimated time limit that you're thinking? Is it just until we load up on samples in all of our boxes?
I think so.
Okay. So at this rate, would that be like another 4 hours or so, do you think? I unfortunately don't have one of those sheets in front of me that's...
Oh, here, I got the sheet.
See anything?
Yeah, it's only like half full.
Okay. Yeah, we were trying to get an idea of when we should do our calibrations, because that's the one region that's most limiting us on this, but as long as we appropriately stretch out that time frame, we should be fine. So,
Yeah, I think if you have enough reagents, we can keep working.
Okay.
See when we will come up earlier. 5 days
lurking in the
later.
bottom of the field. Okay, great. Thank you, Santiago.
Yeah, of course.
Yeah.
We're all gonna run out of reagent.
There's more coffee upstairs. I'm drinking
Here
more
outside,
reagent.
so I'll push
The
that.
human reagent.
Yeah, well, seems like we were approaching the waypoint, but I don't see anything worth stopping for, so we can continue to moving towards waypoint 21.
Roger. Just trying to move forward.
Right, yeah, I see. Straight against the current. Right? Oh, wait a minute. There's one of those yellow ones straight ahead where the lasers are. Spoke too soon.
You want to Solaris and sample it?
Yes, please.
Alright, just let me know when the ROV's in position, we'll start collecting that background.
Event 19? Roger.
So I can move with both arms.
I'm sorry?
Hammers, yeah.
This yellow one? Yeah, farmers.
You think?
That's perfect. That'll do it.
Alright.
Let me bring you in closer.
I'm starting the background.
Alright, start background on Solaris.
Let's get out of the wind for a while.
It's a lobster coming.
Okay, SOLARIS is ready to approach.
You ready?
Yep.
Alright.
Okay. Here we go.
It's feeding, right? Station.
Has Solaris been pretty stable all night?
Yeah, it has been behaving. We're very pleased and proud of SOLARIS tonight.
Oh.
Try to put the one on the downstreams on the right side of the coral. Yeah, like you're doing there.
Put it on the right. It's gonna try and touch it.
Oh, we have a nice little response coming in.
Have we touched it?
Yeah.
Looks like the tip's a little low.
Yeah, the tip got.
Yeah, we lost the filter cap along the way, but happy accidents.
Yeah, just gotta be careful not to poke things with the tip. We don't wanna entrain any particles from animals or anything else.
Equals after touching.
Yeah, you guys see nods are touching.
Did anyone see if the
They were open, they closed.
Okay.
This is kind of where y'all want it.
Maybe they'll reopen
It's a tough angle.
The current is up to have a gust.
It's crazy, right?
Steady.
So you just got the peak probably when the pulse goes like.
Exactly. And then it's been essentially baseline since.
Okay. Are you okay with this?
Yeah, just give it another 30 seconds. It's boosted up a tiny bit there again. So I just want to see if that manifests into anything more substantial. Okay, I'm happy on this end.
Okay. Thank you. We can stow it away and get ready for sampling. So this is gonna be Paramaresia sample SH18644, correct?
Yeah.
Thank you.
Okay, where's this going?
Buy box.
Can we get a quick zoom on the coral
3.
before we hit?
Oh yeah, let's get a.
Available.
Let's get a.
Yeah, the last one.
You don't think they'll fit in
Yeah. Get it closer. Get the whole thing in frame and in focus.
It's great, thanks.
And am I trying to collect this whole colony or just part of it or?
You don't think they'll fit in quicker?
No, we want it in a bio box.
Okay, that's easy one then.
Let's see. I think it's small enough that you might end up taking the whole thing.
Make a picture and see.
We need to do a servo valve set together. Dropping.
Yeah.
Nice one. You said 3B is open? Similar?
Watching out.
Yeah, 3B.
Okay. Bio box 3B is available for this one.
I was going to get ready to run a SOLARIS calibration. Are there any other samples in the immediate area we were planning on grabbing?
No, I don't see anything else. Yeah, go ahead. A good time for a calibration. So we're gonna run a calibration on the SOLARIS. It'll take a few minutes. Did you stop your EDNA sample, Luke?
Yes.
How much volume did you get?
So far,
You ready? Okay, the box we open
62 liters.
Oh, it's still going.
Yeah, I have a little more.
Oh, can you stop for the samples?
Yeah, I stopped looking. And then after that, so probably before the next sample, I'll be finished. And then just do some biscuits before we come up, unless we change. If we change habitat or depth, I'll probably fire them earlier, but I don't think we will. As long as we're around 400 meters. It's ish. Unless you want to go shallow.
Well, the only waypoints. We're going to bring us to 370, 340.
Yeah, a question. That's the 3B, right? And left of it, it's the 3A.
Yes.
Yes.
And there's supposed to be a purple for Mauricia there.
Yes.
Am I the only-
Is that a question?
Is it there?
Yeah.
Okay.
It's in the bottom.
Okay, then bio boxes are full.
Oh, bio boxes.
Okay. Oops. Okay, so we're gonna keep going.
Yeah, let's keep going.
Right before you lay it out next time, man, I'm just gonna do a quick circle valve offset. You look sleep in.
Next time we get a chance, I'll make sure it gets the whole thing, and then, I don't know, there's no way I need to put the reason it was lagging, as we're supposed to.
Let's see a sedimental slope here.
There's no way to really have it so it doesn't lose memory without it.
So we're gonna continue moving along towards waypoint 21. We should be at a depth of 370 meters, so expect to gain 30 meters as we go up hill.
So it's only like 7,000.
That's another purple one, right?
It's super low.
Can we take a look at this one, this purple one, please?
I haven't seen the sticker pads there. That's true.
Ta-da! Yeah, it'll be good to sample. Are you still doing calibration?
Yeah.
Hold on.
It'll take us probably another 5 minutes or so.
Another 5 minutes?
I should just hide this so nobody uses it. It's better to just let them.
Yeah, I guess take the time to position the ROV in a good spot. Do some imaging.
Yeah, we can, I mean, 30 seconds, so we can fire some instance too.
Oh, we can fire instance. Do you want to do that now?
Let me just finish this so I can.
Yeah, but I mean, you want to do this here?
Sure.
Okay.
That's a good time.
Yeah.
Hey, do what you gotta do.
All that stuff can be used to
So we just write down 11 hours.
Well, probably one chest would be
it's not this.
Okay. Yeah, we can do distance.
You stop the sample?
Yes, so, for the record, samples SH13547, 13548, and 13549 have finished. So that is another series of 3 different pore size filters, 0.2 micron, 1.2 micron, and 5 micron.
And they were all 75
Yep,
liters?
approximately 75 liters within 200 milliliters, 73 of them.
Pretty accurate.
It doesn't do that much bring your pitch up.
Pitch up?
Yeah, that's when it will crunch. Freeze.
Oh, yeah.
Yeah.
Do you want to do this?
Yeah.
5 and 6?
Alright, anyway, so we're doing Niskins?
Yeah, just bottles 5 and 6. Thank you.
The racket as well.
So, Melania, this week's 5 and 6 will be number 13551
13551.
and 13552.
Okay.
Yeah.
Yeah, that must be related to how sluggish the shelter is. We're good to fire these both, yeah?
Yep.
Five fired. See it?
5 fired. 6 fired.
Alright, thank you.
I love the trigger system.
Yeah, it's
Yeah.
awesome.
Alright, Solaris?
We'll need a couple more minutes on our end.
Okay. We can,
Calibration is done, but we just gotta document it and then we'll get the background signal.
we can
We can
get
the
some
background.
good images.
Go ahead.
Okay.
You want this one?
Yeah.
Fortunately, it's not red.
Okay, we're sampling another coral. So that would be unfortunate. We're calling this event 20.
Okay.
Is this another, this is another, it's black.
This is actually an optical. It's purple paramaricia.
Oh my god.
It's like the one we sampled
Yeah, it's just in a bit of a different light.
half an hour ago.
Thank you for correcting me.
No worries.
2800. We're ready to probe, yeah.
Yep, we are ready on our end.
You got a frame graph, Luke?
Yeah, I did.
Okay. So SOLARIS is ready to probe, so we can get that.
Fix the lights with it. Close stuff. So you're ready to probe?
Yep, we are ready on our end.
Okay.
This is gonna be SOLARIS event 19?
Event 20.
20? Oh yeah, 20. This is a purple paramaricia. It has open polyps, so we'll see what we can get.
on the sea.
Yeah,
You got a good view on the sea too.
that's pretty close. You get closer, but I'm not bumping.
Yeah, you wanna. It's hard because the downstream is it's on the other side, but
You guys getting anything?
We're not seeing much of anything at all on our side.
Maybe I could do like this.
yeah, can you go from the top?
Happy? Wanna zoom in?
Yeah, you can do it a little bit.
That's perfect there.
Sure, that's great position.
Just hold it there.
She's
And we're reading approximately baseline.
Nothing.
I'm reasonably happy with that if you're happy with something.
Happy. That's good. Great. That's a pretty good drawing. Amazing. The polyps did not close at all.
Oh, we're gonna collect this?
Yes, please. Want to put it in the quiver, so don't get the whole thing.
No pressure, is that? No pressure. Motor's getting on the area.
No pressure. Why?
No pressure, but my motor's getting on some area.
Oh wow, we're really hot.
No, it's not too bad. It's hot, but so you better take that quiver off.
Quiver 1 is open. Right?
Quiver one's open too.
I thought Quiver 1 had something in it.
No, we tried.
Isn't that the haunted one though?
So maybe you can go for this branch. Oops. This tail branch here.
Picture and sit.
This is gonna be SH18645.
SH18645. Thank you. Purple. The primary is here purple.
Is that gonna be enough for you?
Uh,
A little more.
let's see. Can you close?
I know it's hard to tell.
Yeah, just close it a little bit and then we can see.
I'm gonna go a little more because I think that was gonna be a tiny tank. Oh no, it's 4 starfish.
Yeah, that looks good. Yeah, that looks pretty good. Perfect. So we're at 409 meters deep and the temperature is 14.9.
I'm gonna, no, I guess I don't need to watch out on this one. Probably a better position.
Kind of a subsample of both here,
Yeah.
free subsample.
a little bit. I'll let you know if you're shoulder too far down.
What's that about?
I'll let you know if you're shouldering too far down, but you're looking good right now.
Alright, on it, quiver, here we go.
I think this quiver is cursed. Nothing was
There you go, Schmidt. Some of it went in. There you go. Keep dropping in. If you don't touch it probably, if you don't move. There you go.
Ta-da!
You think it's just the
Excellent.
black rolls that are cursed?
You think the black orals are cursed?
Yeah, they float. Well, you know, is the name is
No.
anti-disease.
They're trying to get
Yeah.
this,
Happy
Yeah.
for hot
Get-
fuzz
get it.
disease.
Is it really?
This is the secret to
Yeah.
good
Used to
health.
ward off evil spirits.
Oh, really?
Sorts of ladder ghosts.
Maybe.
So what is telepathy?
Yeah, because they're all jacked.
Don't push anymore.
What's that?
Don't push anymore. No case.
How is it pronounced?
Stylo?
Stylo is the, means like a pen.
Oh, yeah. I guess that makes sense.
This monkey is big too.
Pen disease.
Hmm?
It's like stuff
Pen
around
disease.
something.
I mean,
I think it's a sail.
not really. I guess Stylo. I guess
Because
refers
they
to
are like a,
branches.
you know, like a, how do you call it? No,
Pen disease.
the feather pen.
Or
Quill.
telepathy.
You know, this whole thing that.
Yeah.
Oh, what's up with the what?
Bungee stuck on something.
Yeah.
I could mess with this while you're flying, if you need to come off.
It's not gonna matter because I'll be thrusting more and be using the minutes that's wrong with this. They'll just get hot. Might as well stay. I'll just keep watching it.
Yeah.
Let's see if I can figure out why this is stuck.
Bet you I know where that is.
Oh, you got
Ah,
it.
there we go. That's better. Moving all over with the sip.
It's far.
No, just the fact that it's still plotting on half the screen.
Roger that.
With the sip.
Yep.
This is somehow converting something into seconds, but there's a factor of 30 here. I wonder if it's supposed to be 60.
You know,
What this number means. Is it used for scan?
If you wanna top the SOLARIS a little bit.
froze, locked.
That's the square.
How's that?
Right. We're gonna keep carrying on to 21.
Carry on. Thank you. Great sample. Some more big boulders here. Solantids.
We started yesterday. 3-7 we started yesterday.
Something big straight ahead. It's probably a sponge.
And I have to start climbing up to get to
Mhm. That's correct. Ah, some big outcrops.
Santiago, what is sponge in Spanish?
Esponja?
Where's all entrance?
Yeah, there's a whole bunch of swamp. It's interesting, huh?
easing off a little bit.
Yeah.
Moving forward, so it's
We're almost an hour past the peak of the low tide. The peak of the low tide, the valley of the low tide.
That'd be one of my back
I'm sorry. Let's take a look at that on the side of the rock. Fuzzy, the big fuzzy on the side. This.
Huge.
That's pretty big. Oh, that's some Jeremy coral.
That looks like a black coral.
Oh, that's ours.
It's a big Jeremy coral.
We need only one more, and then we're done with this one.
Some yellow ones next to it too.
That's a big antipathes, huh? I guess we can try it.
Let's see.
Yeah.
Well, it'll be good to get a. It's actually maybe a decent spot for ROS because it's a little bit sheltered.
It's interesting how it overlays.
Is the ROV in a stable position?
somewhat.
Okay, well,
Be quick.
I'll start collecting the background signal now then. Where is it?
That's how you collect these samples, right? On the fly.
Yep.
Thank you.
This is a good one to take some video footage, looking at all sides, because it clearly forms planes on top.
I'm sorry?
Go ahead and do your video, whatever you want to look at. It's not
Can we just get a full frame first, Santiago?
Yeah.
Take it first.
Is it possible to just like shift across it? to
How's
the-
that?
Can we iris down a
Got
little
it?
bit? Can we iris down a little bit?
Oh, I don't control the iris.
Oh, okay.
There you go.
That was one?
Yeah, that's good. Thank you.
We are good from a SOLARIS perspective.
Look at that crab.
Yeah.
Oh, we're good.
Yep. SOLARIS is ready to measure it. Everyone else is.
Whoa, I'm sorry.
Ready.
Yeah, go for sure. I was thinking what you did last time with coming from the top and putting it like that was pretty good. I don't know if we can try that again in this coral.
This will be event 21.
Event 21. Yeah, Solaris is ready.
So once you use that arm, the temperature's gonna start climbing up.
What's that?
Once you use the arm, the
Antibiotics.
temperature's gonna start climbing up. So let's go ahead and get to it now.
Biggest antibiotics.
Probably is.
Probably is what?
That's gigantic.
Gigantica.
You're done, named it.
Huh?
That's it, named it.
Named it?
We know where to do.
Oh, there's a cookie cutter sea star in the back. You see it?
Alright.
You know, when there's too hot.
They're almost too close to us.
Okay. Oh, whoa.
No, cookie cutter. Cookie cut? Cookies and stuff?
Down here. Well, there's a cookie cutter shark.
Is that
Yeah.
a cookie cutter shot?
No.
No. Wish we did.
It's a sister. I think it's called cookie something. Cookie cut.
Open your iris a little bit.
Just bump up the ISO there.
Cookie sea
It's
star?
already
Sweets and treats by Sean.
Alright, it's pretty close, but not touching.
That looks great.
Yeah.
Those are
What
such
was that?
natural values. Or we are reading approximately baseline values still.
really black though.
I mean, there's tons of polyps, and it is really close.
Yeah.
I don't know what else we did. Shake? No.
That's perfect measurement.
That's such a good position.
That's an apricot right there, the little like
Maybe I did it all.
with an amphipod.
Alright, I'm pretty happy with that.
Call it no signal. No signal antipathies.
Okay, he's done. Are you done?
Yes, thank you.
We're gonna collect something.
Yes. We will attempt a collection.
Does it also just just go around the coral once in the video? It's clearly colonic.
Careful with the wand. Careful with the wand.
Oh, we can do it after.
Yeah. It's just, it seems like they're in a pretty good spot for sampling.
Yeah. Yeah, you can grab that top plane from the Rico.
Should be well underway now. Froze. Washed.
So you want a small sample.
Small sample. Where is this one going? In the quiver?
In a quiver.
Yeah. So 18, I think, is open already, I'm seeing.
Looks like it.
Yep, I see it. It's on the
Is-
bar right there. Okay.
Simran, can you confirm that there's nothing in quiver 18?
Yes, there is nothing.
Okay. Thank you.
Tricky getting
And
that
this is
cover.
gonna be SH18646.
SH18646. Thank you. Antipathies. We're in a depth of
Well, we can do 6 or 7.
409.
Can we put it inside for on the covers inside?
Yeah?
Because the cover on that outside cover went on the outside.
Yeah, do the easiest thing possible because this coral
Okay.
will crush your soul.
Well, this is the sticky one.
This is it.
Yeah, this is
The peanut butter coral.
Sticky and floaty.
Right.
Poaching out.
Yeah, go for it.
temperature is 13.5. Be cooler.
So which one is it?
6 or 7 are both going to be included in my sheet. Is that correct?
6 or 7, yes, they are empty.
I'll be right back.
This is 186546.
Yes.
646.
Yeah.
Santiago.
I think you might have to remember the rest of them. What's up?
What do you want to pick? What do you want to cut?
We just want a small piece, so whatever's easiest to get a small little cluster.
You want to top? See that small area?
Pushing it on.
Problem is sometimes it's pretty wide.
He's gonna cut that corner.
Yeah, the other thing is the branches are all kind of stuck together.
Let me see where I can get it.
Yeah, that's the challenge. This one it's hard to get inside it because the branches are all kind of stuck together like a spider web. Maybe even this little piece right here. Hey, Jeremy.
Let's just want you to take samples for your
What's that?
stuff. Are you just taking some samples?
Yeah, so
We
I'm
need
at,
a voucher as well.
right, but it has to go in a quiver. So
Maybe a little more.
a little more, okay.
This is the adult colonnades, Joe.
The problem is since all the branches are fused, it's going to be challenging to get a small piece that fits in a quiver.
Yeah, I know.
That also has big enough for
I
yours.
think I see one.
Okay.
If this is a new species, this is the type. So we can bring it here, so you can make the type.
Stuff is very sticky, isn't it?
Yeah.
It's fused, yeah, it's highly fused. I think it's an extra fan on that piece.
Is that enough? Or will we get more?
Whatever it is, it'll just do.
Okay.
Yeah, it's fine.
I mean, we can go again too.
Get
Just keep
another
that in there.
Yeah, that might be a good idea.
Okay. Yeah, if you get sticking at the bottom and get your claw out successfully, sure.
Top of it.
Is that bad?
You're on top of it.
Did it fall out?
Yeah, of course.
it did.
If that's gonna be too tough to get, we might just go for another one.
Is it safe to say we're not going to collect any more of these on this expedition?
First statement. I mean, three. A good number.
Three's a good number.
Good Lord.
I think just this one will be fine. Better be good number one.
That's enough for you?
Yeah.
Okay.
It's okay.
Nice grab.
Looks like it goes in this time.
Yeah.
You think it'll be enough?
Well, I'll just give that one all to you. We have 2 others with voucher subsamples this week.
Okay.
No. No. You're good.
Woo. Nice, nice. Barely sinks.
Yo. This shit was trying to come out of here. You gotta be kidding. This thing's like a practical joke cool.
Okay, that is quiver. 7.
Is that quiver 7?
I love it. That's pretty cool. Feel like I clicked quicker. Keep it click.
Yeah. Okay. What's next?
Nice.
What are we doing next?
So I want to do the image, kind of both sides we talked about.
Sort of.
Yeah, if you could just like get pan.
Fly around it or?
Yeah.
Is this gonna be photogrammetry?
I'm not.
Ideally.
No.
Actually, no.
That fly around might be a little difficult. Once I turn sideways, we'll give it a shot.
Is that that gift to start?
I'm gonna set it up for you.
3D.
Okay.
Yeah, do you want to load the 3D?
Yeah.
So when you're ready, can you push out the camera
Iris stuff.
and pull in your threads?
Yeah, that's all.
Actually, maybe you don't have to. Maybe
Let me pull in starboard ram. See what happens. Maybe you might have a better view.
Yeah, I don't see it. You're all the way zoomed out, so I think you're okay. You'll leave it out.
I'm just kind of worried if I try to fly with this current, it just kind of push me sideways. Especially if I turn this way in front of side.
That's all right. You do the best you can.
Okay. You want to get a video here first?
What's that?
You want to get a video here first while we're in there? This is a better angle than what we had.
Mm-hmm.
So there's a picture of that. Something about the color. Like, okay, I'm gonna fly away. The devil.
Sounds good. Go for it.
A little bit to get that nozzle out.
All right.
Drop the iris maybe a couple steps.
You want darker?
Yeah, just the top of this little like.
How's that for you?
That looks good.
Okay, here we go. Try. So zoom in while
Sure.
do your thingy. I'm gonna try using the set camera to fly around this thing.
Sure. No, you're doing great. Just come around more to the left, turn to the right, if you good. Stay low if you can. Looks like you're clear to the left.
Yeah.
Oh, that's a good crowd.
Nice.
Wow, we hadn't seen that.
Yeah, he's well hidden back there, huh? I think that's probably good, huh? You guys happy with that?
Yes.
Looks good.
Zoom in.
Well, good.
good?
I think we're good.
Okay. This probe's making me nervous.
What's making you nervous?
Probe.
Whoa, yeah. Maybe it should.
Okay. Proceed to 21.
Roger, 21.
Start heading up.
Another bubble.
Oh, yeah.
Where is Charlie? Good morning, I got another movement for you.
I start to see some light on the surface. They're eating those sponges.
Temps looking better all the time. Pretty much do yours for the minerals now.
Yeah. Whenever I turn sideways, it's kind of crabby.
Yeah, it's like I was taking a sample earlier and the pearls like this.
What that one is, look at that.
Whoa.
Blowing the floats out. That's a big mushroom. You see that? Turn sideways. I gotta head into the wind.
Some stylopathies. So we're almost at waypoint 21. That should be 370 meters, so it's probably gonna be a steep wall here.
Were you planning to go much deeper?
I'm sorry?
Were we going to go much deeper? Here?
Not on this dive,
Not this time.
but in subsequent dives here.
Okay.
We had like a target of 250, but we realized that the 250 is just only sponges.
Wasteland.
Wasteland. So I think 350 is that target. So that's why we're doing 400 to 300 or 320-ish.
Why are you asking? Because you're interested in the diversity.
Oh, I was just curious if we're gonna go much deeper.
Yeah, I think the next target that we have is 500. So we'll do like 550 to, or we could do like 550 the target, but push it 200 meters deeper. So it'll be like from 600 to 500. And then a deeper one would be like 750.
That's where we start the first dive.
That's where
right?
yeah. And it doesn't get much deeper than that here. So.
But it was interesting in the sand there.
Right. Yeah. Huh. What's that? Can we take a quick zoom at this?
Yeah. See this? See that there?
Yeah, but I don't know what it is. The one that is just down below.
Yeah, stand by. It's right in front of me.
Yeah, that on that camera. That's right.
I just need to get out of here a little bit.
More stuff I'm paying in. That looks like I might be pulling it pretty good this year.
Yeah.
Year back.
Yep. Okay. I just ran down. Here we go. Just like
I'm not here. Looks like the skeletons. Give me the traces. Mostly dead.
Maybe you think the white bits are new or just something running off the old?
Can I? Is it okay if I zoom in?
Go ahead.
Oh, there's polyps there.
Oh yeah.
Yeah, it looks like it could be my traces, but it is alive. It's just a little dirty.
Those polyps look like Jace's polyps though?
I don't know.
Can we count tentacles?
Do you want to count tentacles?
Yeah, can we see?
Maybe.
Oh jeez.
That's full zoom.
1, 2, 3, 4. I think 12 it looks like. Okay.
So
That checks out.
cool. You log that?
Yep.
All right. Did you get a picture?
No, I can't.
Let's do. I'll take one. Got it?
Yeah, I got them.
Okay.
That's cool. I haven't seen one of those yet.
No, I know. Thank you.
Proceed.
Yeah, we can keep going. So as the first stony coral we see this. Oh, this is an art watch.
Yeah.
Colonial.
Colonial stony coral. That's right.
I saw a couple in Desecheo, but we can take a look. Purple and white ones.
I haven't seen any like coral roll around here.
Just falls down the
Hmm?
hill. Just falls. You said you're not seeing any rubble.
Mm-hmm.
Yeah, it just falls down the hill.
Yeah, proceed. That looks like the purple light
Yeah.
purple perm receipt that we were seeing there earlier, that we sampled.
There's a wall.
This is, I guess it's more of an outcrop than a wall.
Yeah.
Wow. And there's the Swantis at the top.
Yeah.